stop your presentation, Sanjay.
We are starting the inauguration session.
Yes, I have stopped my sharing and
Leave further.
please go ahead. I can hear you very well.
Leave further.
हमारी मीटिंग खत्म हो गई।
So, should we begin? Welcome to the ICGB annual sets of conferences and workshop. We are starting this after a gap of 2 years, and one of the major motive of ICGB is to develop knowledge and then impart knowledge to the various scientists around the world, particularly the scientists from the developing world. And in this direction, our Director General Lawrence and Tinker and other directors of the various components of ICGB have been working very hard so that we can really communicate our science to various countries and also impart the knowledge to the students around the world, particularly the students from the developing world. And in this direction, we have worked very hard for the last so many years. And ICTV workshop actually is one such step in that direction. Another major step has been to really invite students and postdocs from other countries, and then to various components of ICGB, and then provide them facilities to carry out their research. Particularly, we actually get a number of PhD and postdocs students and train them in various areas of research. And ICTV workshop and meetings, another such avenue where we can impart knowledge to the various scientists around the world. Now, this is the beginning of ICTV workshop this year, and we have named it ICTV Infect 2021. This workshop will cover various areas of infectious diseases, and our main focus is actually to tell the students about the various developments in developing new drugs against these infectious diseases, as well as how the vaccine work against these various diseases being carried out around the world. And this workshop includes number of scientists around the world who have been doing pioneering work in various areas, particularly in malaria and TB. And also we have invited some of the clinicians in this workshop, as well as some diagnostics methods, people who have developed certain diagnostic tools to identify some of these diseases. So now I invite Dinkar to say a few words about ICGB, and then our Director General will also will present about the ICGB motto as well as aim of ICGB. Dinkar.
Good afternoon to all of you. Good morning to Professor Lawrence Banks and other colleagues in Trieste and Cape Town and many other parts of the world who are participating in this workshop. Also, specifically, I am pleased to see that we have quite a few of our colleagues sitting in the hall, including Professor Chauhan. I know the vaccines is very dear to Professor Chauhan as well. There is a continuity to this workshop in the sense that completed 30 years in 2017. Asif and Pawan both organized a similar workshop as part of the 30th year celebration of ICGB. And now they set out to organize as a series the next workshop now. Unfortunately, we had put a gap to ICGB workshops at New Delhi over last almost a year and a half, and finally Pawan Malhotra and Asif Mohammed developed courage and set it out initially to have it entirely physical activity, because the tutorials were to be done in the laboratory. But considering the situation that has developed during last couple of months, they evolved it into making it online plus in the laboratory. But they have developed fantastic online tutorials in video, which would all participants find very interesting to do even while sitting in their respective places, to go through it as if they are working in the laboratory. So that is the most important part of this particular workshop, and I am sure this would be very successful. In addition to these laboratory training, which is made in video form, we have a galaxy of speakers that are lined up to be giving talks in various aspects of infectious diseases, including development of vaccines and drugs. So we have really a retreat for next few days in terms of this field, and it is very refreshing that we have begun it in this hybrid mode now, but hopefully when we have our next workshop starting from January, we have 7 workshops to be conducted during 2022, and we hope that all of those will be in actually physical form right in New Delhi. So we will have this as a success that will set trend for us to organize series of workshops where a large number of participants from outside India and within India will join us and be part of this endeavor to learn more on various aspects of science that is carried out in ICGB's mandate. And with this, I don't want to take too much more time, but because we have a very important speaker lined up to give his talk, who is really hands-on person developing vaccines, which is Dr. Sanjay Singh. He happens to be alumnus of ICGB, so we are really very proud, and he has been engaged in developing a new biotech company, and then he is one of the pioneers in developing vaccines for COVID, and he is right in the middle of the handling of the Omicron new wave, and because of that he actually was forced to stay in Pune, so he will be joining us by video. Otherwise he was supposed to be here physically and interact with us. Nevertheless, we will have Sanjay Singh coming here, but before that I would like to invite Professor Lawrence Banks, Director General ICGB, to give his address. Lawrence.
Good morning. Thank you, Dinaka. Can you hear me okay? Okay. So, good morning, everyone. Good afternoon and good evening. It's really a great pleasure to be with you all today and to welcome you to this really what is an amazing workshop. And I think the timing for this is absolutely perfect. If we think about what's happened over the last 2 years, vaccines and drugs for infectious disease are really fundamental to what is really going to be driving us and getting us out of the pandemic. So the timing of this workshop is really impeccable. And I think a really massive thank you has to go to Asif and Pawan for doing a brilliant job in putting together such a great program, and also having the bravery and the perseverance to go ahead and put this meeting together despite all the trials and tribulations of the pandemic. Clearly, these types of meetings being done in a virtual format for the large part is one of the I would say one of the few advantages that we've gained from the pandemic is that we now learn how and know how to interact and exchange scientific information and ideas across multiple time zones throughout the world. Clearly, gathering in the same place and exchanging ideas over a coffee and over dinner and lunch is still the ideal way to go, but I think in the current climate, this is a wonderful compromise. The program is absolutely outstanding, and I must say I'm really looking forward to hearing many of the speakers over the course of the coming 2 to 3 days. And I think it's also very exciting that Pawan and Asif have managed to retain the practical elements of the course. These are absolutely incredibly important in these types of training workshops. And I think the way they've done this and embedded it in amongst all the lectures is really, truly tremendous. So I'll not take any more of your time. I wish you all a very enjoyable workshop and meeting, and I look forward to hearing many of the really tremendous presentations. So thank you very much. I'll hand the floor back to Asif and Pawan.
Thanks, Lawrence. And we are also very happy that you have been, since so early there in Italy, and you have been able to join us and give us your thoughts. Along with us, me and Asif, Dinesh has also been very instrumental, along with and Dinkar also. They have been really putting their lot of effort to organize this meeting. And thank you once again for joining us early morning from the Italy. So let's begin with this first talk from our speaker Sanjay Singh. Sanjay has been part of ICGB about 15 years back, and he joined Malaria Group, and he was earlier working in malaria field in CDRI, Lucknow, and worked on not on vaccine but on polysomes and various other areas, but also towards the infectious diseases. And then from ICGB, he started working along with Chetan and me on vaccine work. Particularly with Chetan, he worked on PVR2, a vaccine which is going towards the field trial. And then Sanjay always had a charisma to move forward and to achieve something big. I must say that Sanjay's story from going from basic research towards the translation part has been one of the real book, it is to be written in a book chapter, because this is a person whom I know from lab work, from the lab bench, who has who has risen to really a great heights. And he has been doing great work towards developing vaccine against number of diseases, including the malaria, and also he has been instrumental in developing a particularly also diagnostics or analytical tools, particularly in terms of developing diagnostics for COVID and sequencing of various COVID genomes from the clinical isolates. And one of the major theme of Sanjay's talk today, as well as his laboratory, is to develop mRNA vaccine. And I am sure this will be one of the first vaccine in India, which is mRNA based, which is to be released in next 1 or 2 months, 1 or 6 months at least. I think Sanjay will be more clear on this. He will be telling you. But I must tell you that Sanjay's growth from basic scientist to the translation lead is an example for many students as well as us, that how he has moved from one step to the top of this. In between, from ICGV to this company Geneva, Sanjay has also worked on malaria vaccine in an NIH, and there he worked on lots of antigen discoveries and taking them to the vaccine work. Now I invite Sanjay for the talk, which I think you all will be very happy to listen.
Thank you very much, Dr. Malhotra. I think I'm audible well to the group. So, very first, I would like to thank Dr. Malhotra and Dr. Salunke for inviting me for this very important workshop. And I am overwhelmed because I worked at ICGB in the lab, and where Dr. Malhotra was one of the important components. My group was led by Chetan and I think, head of department, and then the director was Professor Chauhan. ICGB culture, somehow, when I was doing PhD, I was very excited about it because it's one of the unique and pioneer institute in developing countries about the biotechnology, and which is not doing the research, but also giving a lot of training to the students who are coming from the developing countries in the area of biotechnology. I was also very well known to the current director, Dr. Salunke, because my wife has worked with him when I was at ICGP. But the most important factor I would like to say that for me at the ICGP was that I was fortunate enough to have a very good interaction with the founder director, Professor Tiwari. And somehow the science and business, it was a great influence from him on me and where I am today. So with these introductory facts, I would like to say one more very exciting, very funny thing about Dr. Pawan Malhotra. We used to say him Malik, means he is the boss. So he invited me that I have to give a talk, and I said, I very gracefully accepted. I said, I will come and give a talk. So today, I'm going to talk about more on the vaccine side. Yes, in my past, during my PhD, I worked on the drug side. It's still my company's major focus on the drug side, but we also are very keen in infectious diseases. So I'm basically going to present what is the work which we are doing today. And also, it is very pertinent related to low and middle income countries. So when we talk about the vaccine technology, I want to go for this, for the benefit of students, a very basic concept that there are different vaccine platforms. Once we know the antigens, and these platforms need to be tested, which is the very good platform or which is bad platform. So if I see this slide, very early, maybe a century ago, at Oxford, Professor Jenner developed this concept, whole organism, either live or weakened or killed form. They proved the concept of vaccination. And today also, polio, hepatitis A, rabies, influenza, yellow fever, many vaccines are in the market based on this generation 1. And then after we started moving into the recombinant DNA technology, and where I was fortunate to get introduced to this technology, platform vaccine 2, and where you have the recombinant protein, and you can express the recombinant protein in a most native form, and then you can see whether it can be a vaccine candidate or not. So here our examples are seasonal influenza vaccine, hepatitis B, HPV, and many other vaccines. So this is basically the vaccine 2 is the workhorse of the industry till some time ago. Then it will be a next generation vaccine came, which is basically adenovirus non-replicating DNA or plasmid DNA platform. And one of the vaccines which is emergency use approval is Ebola vaccine. This was approved a couple of years back when the Ebola epidemic came in Africa. And then this COVID-19 vaccine, it is AstraZeneca vaccine. So this is the platform which this vaccine came. And suddenly during COVID, the new platform emerged, and this is the mRNA vaccine. Yes, it was in the picture for last decade or so, and during my NIH days, one of the colleagues, Barney Graham, his lab was focusing a lot on it. And finally, the fastest vaccine which came from against COVID was from this mRNA, one from BioNTech Pfizer and another from Moderna. Now, what is different in these four platforms? What are the pros and cons? If we go from the vaccine 1.0, you have to grow mammalian cells in a big quantity, and then you have to infect the live virus, and you have to propagate the live virus, and finally you have to harvest the virus, inactivate it, do the potency, do the fill and finish, and finally go to the testing and market. But this has two limitations. The one limitation is that you need to have BSL-3 facilities or BSL-4 facilities because you are culturing a huge amount of live viruses. So their chances are it can spread. And then there's a limitation in its scale up, how much you can produce. That is also because the cost of building these facilities, keeping them intact in a condition of BSL-3, BSL-4 is very, very costly. And then you are exposing human being continuously to the infection, so you have to keep a pressure of vaccine and all that kind of thing. So this is, but still it is proven, and from India, our peers, Bharat Biotech, they have bought the COVID vaccine from this platform. Now the second generation vaccine is based on the recombinant protein. So once you have the gene sequence available, then you can make the synthetic gene from that. You can make master cell bank, working cell bank, so you don't have to go to virus every time. So here, first thing, you have brought back the safety point into control because you are expressing a particular antigen. And then you can have to have big fermenters, big bioreactors, and you can produce it, you can purify it, you can fill and finish it, and then you can go to the market. So this is the second generation. In the third generation, when this non-replicating adenoviral vectors or plasmid DNA came into the soul, yes, this is a much easier platform, and you have to produce it. But again, you need huge culture facility to produce it because expressing plasmid DNA is not easy. Either you have to use in bacteria or adenoviral vector. You have to produce in the big mammalian cell facility, and you have to come back. Now, with the emergence of this pandemic, mRNA vaccine came into serious play because before that, somehow this path was difficult, and especially because of many reasons. One of the reasons was the stability of the mRNA and delivery of the mRNA. But this field was, I think, ready for this time. So here you just need a DNA template. And then everything is synthetic. You can do in the test tube. You can do in a beaker. You can do in a big tank. So you can do the in vitro transcription. And once you have done the transcription, you just do the capping and tailing, and then purify. Very simple step of purification because you have not added many things into it. And then you might have to complex and put it for delivery. And then fill and finish and go for it. So what is unique into this particular platform? This is still very novel and new, but you don't have to create these big facilities for manufacturing. The first thing, The second thing is that anytime if a new variant has come, you can change this variant in no time. You have to only make a synthetic plasmid. And synthetic plasmid, you don't have to grow and make it big. You have just very small quantities of you can make, and you can switch it on. So within few weeks, you can turn it around. So now Omicron is there, and our government has asked us how quickly we can do. We are thinking that we can go within 60 days. This new version of the vaccine should be available. So this is what overall is if you see on the bottom of one is the term time of to manufacture, and second is economics of manufacturing. So economics of manufacturing is also going to change a lot. It will be much easier, much cheaper to produce these vaccines. And second is time to manufacture. So we have done some work to make the mRNA stabilize. Otherwise, we can start in the morning, and by evening, the vaccine is made clinical grade, which is ready to injectable. And this is the unique feature that has come into this particular platform technology. Now, I'm not going for further, but now I would like to jump on mRNA vaccine and what little bit we are doing and how we are doing and what are the advantages and how we are trying to solve this overall scenario. Because till today, mRNA vaccine has been made, but it has been only available to a limited countries, and there were reasons why it is so. So if you see global mRNA vaccine distribution, as I said to you, it is so limited to North America and few places to Europe, are few places here and there, but very little. Rest of the world, 90% of the world has not able to get the benefit of this great technology. Another part why mRNA is so important, and again, if this is the most efficacious vaccine, the expected date is somewhere we can start fulfilling this demand globally from 2023. So till that, it will not be available to low and middle income countries. And I'm so happy to present this work on ICGB platform, because ICGB kind of institution was created to make this technology accessible to low and middle income countries. So when this pandemic came, and we were more on the technology side, on the generation 2, which is recombinant protein. We have a couple of vaccine programs going on with a lot of collaborative work globally. But we decided to move into mRNA because we, as a Genova, we were working on some of the personalized cancer vaccine program already based on mRNA platform technology. So we switched back ourselves, and this is a huge program. So went to the board, talked to the board. Board was very not sure that whether we should invest this kind of money into this program, where it is such a no success at that time was known when I decided to move into it. So we put certain points into it that we will create this vaccine based on two basic platform technologies. One is the self-amplifying, other than what Moderna and Pfizer was making. And second, we will use the delivery system as cationic nanolipid emulsion as a delivery vehicle. Very thankful to Department of Biotechnology, because we talked to the secretary, we submitted a quick program, and I think one of the biggest quick turnaround time, though it gone through multi-layer of review, but DBT gave us the first check of 25 crores. And to start with, we said that we are very keen. It is risky, but we are taking this risk. So we used in our vaccine the most prominent mutant, which was D614G, and self-amplifying technology, which has advantage of low dose because it goes a couple of rounds into during its application, so you don't need to give heavy dose. And then this is unique, which is adjacent chemistry. We don't have to entrap our mRNA inside the nanolipid, but wrapped on the surface. And we've said that we will make this vaccine stable at 2 to 8 instead of ultra low temperatures in minus 70 degrees centigrade, and can be potentially used for mass vaccination and can also be used for a booster dose. And these things we invest very early into our program, because if these points are following, then we will keep on moving the vaccine. Going into the bit scientific detail, this is you can see the sequence. We use the spike protein, and these are the structural protein of self-amplifying format, and where you can, we put the gene of interest, made the mRNA, and then we made this cationic nanolipid emulsion here in-house, and then we wrapped on the surface of surface of this cationic lipid, because we used cationic lipids which is embedded in the surface, and this was the unique chemistry which moved ahead from us. So we just want to show you this is the mRNA, this is the purified mRNA. When we made such a big spike protein with the self-amplifying mRNA, our size of the transcript gone to around 12 kilobase. And the biggest size which was available in the world at that time was 9 kilobase. So you can see that we didn't have the ladder to run it. We didn't have the machine to run it. So we went to some of the vendors and talked to them and find out how to run such a big size of mRNAs. But this is, you can see the purity of the mRNA. And then when we emulsified with the nanolipid, it's a size gone around, you can see the middle of this around 100 nanometer. And a simple mRNA on the second picture, if you see the lane 2, this is mRNA, purified mRNA, but we mix with emulsified form, then it doesn't enter into the cell. And you can say that the lane 4, if you can see, untreated vaccine, so it catches on the gel itself. And even if we treat with RNAs, it is still protected. And then once we extract it, and then we can see it falls down. So this was a very, very unique and very basic for the students, how we can see that we have made a vaccine which is entrapped on the surface of the nanolipid carrier. But in case of, if you see the lane 3, if we treat with the RNAs, the whole vaccine goes away. We did, because with my past experience, we should not go with the lab grade materials. We produced the CGMP grade material, and just for a proof of principle, we immunized the mouse on day 0, and then gave a booster on day 29, and sacrificed the mice on day 57. So this is just an example I wanted to say. In the panel A, if you see that, yes, it is highly immunogenic. If we put on the plate the spike protein, and on day 57 also you can see it's just a significant boosting of activity is coming. The gold standard test for this is live viral test, which we do here in Pune itself. There are 2 facilities. One is NIV, but NIV was very busy, so there is another DBT funded institute, IRSA, where they have done this assay for us. And we found that our day 57 sera can give a neutralization titer of more than close to 1,000-fold 50 print 50 values, and which is the standard, you can say, if you feel the vaccine is going to work. And in column C, you can see there is a great correlation between the titer of the vaccine and the inhibition. But if you go upside, which is the right side of the panel, extreme right side of the panel, we wanted to test how convalescent sera is doing, but doing so many assays, it was difficult at that time. So there is a new kit was approved, and at that time, Dr. Khanna, who is also from the ICGEB, he was on the review committee, he advised to do this test, because this is a surrogate model of test is available, where you can test the efficacy of your vaccine for neutralization. But if you see the day 42 sera is around 90% giving neutralization titer. But when we have taken convalescent sera, which is the one is myself, the second is, at that time I was not infected. Second is the main person who's working on the mRNA. The third one is also one person who got infected, but he was in a very serious situation. He went up to ventilator, thanks God, came back. The fourth one is the moderate one, and fifth one is mild. And the last sixth one, this person worked in Genoa, he got infected twice. So basically the convalescent sera has inhibition ability in this particular assay is around 40 to 60%, but our mice sera has given inhibition close to 95%. So this gave us a big encouragement. and we moved forward. And we also did some cellular assays. And here we want to say the most important side, if you want to see on panel G, where we found that germ-specific, spike-specific germ cell B cells enhancement. And this shows that mRNA, why mRNA is so specific, why mRNA is so powerful, because it goes and create and excite the germ cell B cells, which is specific to your antigen. Another important feature in mRNA is that you have to give the signal the way the virus is giving signal to the body to make this particular protein. So you are using human body basically to make the most closest form of the native protein, because in the same format, virus also makes the protein. And when it in a big quantity is recognized by the human body, it creates the response which is as good as it has seen the virus. So this is the most advantageous point of the Generation 4 technologies. I have worked at ICGB, I think in the first 2 years, every day I have come, I refolded the protein, because first thought was at ICGB to make these proteins in mammalian cell. And when I realized that mammalian cell, if you produce a particular antigen, it gets glycosylated in mammalian cell, and the glycylation is mechanism by which the body defines differentiation between self and non-self. So I told Chetan that it's not possible to make it. So he said, would like to make an E. coli. I said, certainly, but the first protein which we tested, it was having 12 cysteines, so possibility is 1 into 10 power 12 combination, 1 into billion combination, you have to choose one functional combination. And that time we got interacted with NIH too, and NIH people said, oh, are you gone mad to select one combination? But with our hard work and thanks to peace and freedom which I got at ICGB, in 3.5 years, we succeeded making that particular protein in native conformation. So this part of the science was very engraved into me. So that was one of the reasons why going for mRNA vaccine. Now this vaccine has passed phase 1, phase 2. We are at the late stage of phase 3 trials. Hopefully, if everything goes well, by the end of this month, we will complete the phase 3 and we'll go to regulatory authorities for the approval process. But how good data comes based on that all will happen. But this is another part I wanted to focus, and when we work into the low and middle income countries, then we have to see the technologies which we develop, whether they are basically deployable in our environment or not. This is the most important factor, because if they are not deployable, then having a Ferrari and want to run into Purani Delhi space, so no use of the Ferrari is possible. So we decided, we looked into it. So what are the problems taking this technology in low and middle income countries? So first was ultra low temperature. Even in India, it would have not many times Moderna and Pfizer talked to the government, but government finally reached to the conclusion it is very difficult at this point to create the whole logistic related to ultra low temperature. Second is ease of manufacturing. Can it be produced into these environments in a big? And then the third, the most critical part is all the raw material required for this vaccine has to be imported from the West. The next question comes, if we solve these things, whether it's scalability or not. And the last is whether this technology can be moved from India to other places, which is country of our choice and needs, where it is required. So these were the 5 parameters we analyzed. Few weeks back, a WHO team led by Dr. Soumya Swaminathan was here. She saw all the things, and hopefully things will go very well. We are very working closely with WHO. So now the ultra low temperature logistics. So this here I wanted to say that we cannot afford it ultra low temperature. So can this vaccine be made 2 to 8 degrees centigrade? And this is the challenge we have. And how this challenge came into the picture, I would like to show a small video to you. And that was the inspiration for us. So I do not know whether you can hear or not, but if you cannot hear, then captions are written below.
Will be mRNA. And yet, we can't count on them alone, because They're very hard to scale that manufacturing, and they have a logistical problem because they require an ultra cold chain. Now, scaling these for the world would mean having hundreds of millions of doses in liquid nitrogen and keeping them at that negative 80 degrees Celsius as they're carried from plane out to get delivered in the world. I'm hopeful that as the mRNA platform matures in the years ahead, we'll solve both the scaling problems, which will bring the cost down, and will make this cold chain requirement unnecessary.
So I was also sitting on the Grand Challenge panel, and when first interaction I had with face-to-face with Honorable Prime Minister Modi, he's a very intelligent and person who takes the note. He said, can it be made at 2 to 8 degrees centigrade? Because you saw what Mr. Gates said. I said, we will try, sir, and if anywhere it can be made, we can also make. So that is the journey we started. And here I want to bring some part of the science into it. So this is basically this B middle picture, if you can see. mRNA is capped in these small pockets inside the nanolipid also. So there is the mRNA, there is small packets of those, and that has been entrapped into the nanolipid ball. And this is the basically a problem, and which you see that the papers came very late. And if we see into the real detail, these are the channels which have been formed and where mRNA is there. And at that point, we realized water is the reason behind it. If we remove the water from the system, we can have it. And that was one of the most critical factor, because other vaccine is very difficult to remove water because it is inside the nanolipid carrier. And why water is important? So if you see all the information related to genetic messaging is in the deoxyribose, and where if you see H here, but in case of mRNA, it is OH and it makes it liable. So we used to say in fun, the 1:bit atom that has created the problem, but nature has evolutionarily grown in such a fashion that it keeps things in a particular, so DNA is there, it sends a message, and which is there for a couple of hours, and after that it gets destroyed. So this is the way nature sends a message, but that message is not available for all the time. And when you go into detail what is there, so there is a phosphodiester bond, you can see, there's the OH group, and basically in presence of OH group is transfer the hydrogen, and finally it breaks the phosphodiester bond, and basically the basis goes away. So this was a basic simple chemistry, and which we learned. We gone into detail of this, and we find out that if we remove the water, we can make it stable. So we succeeded in doing it. We worked very hard, and finally we made a lyophilized vaccine. And this lyophilized vaccine is stable not only on the room on the 2 to 8, but if you see on the right side lower one, even it is stable at 25 degrees centigrade for 30 days. But we wanted to test whether it works fine or not. So we have taken the frozen form and immunized. You can see the titer day 14, day 28, day 43. And lyophilized vaccine doesn't make a difference in at all. But when you go and test it also, it's a functional activity of the antibodies which we have produced. In the same serum surrogate assay, you can find that it is even better on 28 days, and it is equally good after booster, a little bit better at day 42. So this was a huge advantage we created. It took us months and months and months of time, and at that time, Dr. Sarup used to call somewhere, Sanjay, how you are progressing on it, how you are, because every time when Prime Minister see me, he asked this question, that is the progress is made. So this was a big challenge we took, and today we can say that we are so happy that we have been able to solve it and things are moving forward. The second is ease of manufacturing. So not only solving first thing, it has also helped us in the second thing. So now in trap of mRNA in LNPs, it's a very, very typical activity, and here a lot of mRNA get degraded. And overall yield, if you start 100 mRNA, you will lose around 70, and you can only get 30 entrapped inside the ball. Rest of that you have to purify and remove it. But when we thought about it, we wrapped on the surface, and here our recovery is around 95%. So you just have to mix in a proper concentration, both the things together, and it get absorbed into it. Though it's not that trivial the way we are saying it, but it brought some physics into it, and with that we solved what should be the concentration, what should be the temperature, what should be the speed of mixing, what should be the rotating it is. It should not be stabilized at one place. So all this we work with few of the companies back in Europe, and they made this particular instrument for us, in which we can now from 50 liter to 2000 liter mixing comes with the same specification. And this also gives, it is not that particular developing infrastructure for this is much easier for us. And the third one is no water pocket has been left, because if mRNA would have been inside, water pocket would left. Here we have taken out all the water pockets. There is no water, and you just add simple water and it comes back to its life form. Now the third thing is, which is the supply of raw material. This is a huge, huge problem for us. So for that, first we thought about having the dose sparing effect, so less mRNA is required. So, and then we can do the backward integration of the key raw material. So if we see here what I'm talking about, in case of Moderna and ISN, BioNTech, Pfizer, they have made a simple mRNA. You can see the blue side, so 5 UTR, 3 UTR. But when we have gone, we have added this huge non-structural replicase and subgenomic promoter. And we have not used these pseudo-uridine or pseudo-bases into this. But this has given this particular advantage to us, and why we say that what is self-employing, and our dose in the trial is 10 microgram. Now coming back, what is required? There are basically some restriction enzymes required to mineralize the plasmid, then T7 polymerase, pyrophosphatase, RNase inhibitor, and then the capping enzyme involved. So these are the 5 enzymes involved, and all 5 enzymes we had to buy from US. Now we have gone back, so as a scientist CEO, this is advantage. We made T7 polymerase in-house. Looks like restriction enzymes we have also made. And feels like pyrophosphatase and RNase inhibitor is not required, because once we work in cGMP environment. And for capping also, we are working on something further where we can remove the need of the capping enzyme. DNAs we are struggling, but we will make that happen too. So basically what I wanted to say that we have to produce these things backward here in LMICs if we have to take technology forward. The only solution is backward integration, and it will reduce the cost of manufacturing. It will bring the self-reliance, it will ensure supply, and it will also be affordable vaccine. So now if you see raw material, so raw material, if you see from the GDP size, raw material cannot be made every place, so there are limited places where we have to make. So hopefully India is one of the ideal places where the biotechnology in such advanced form, or the Ministry of Department of Biotechnology is there. It is unique in the world, only few places in the world where you have a Department of Biotechnology. So at present we are talking to a couple of small manufacturers, and hopefully most of things will be made for us as a raw material from them. Now the third thing comes from scalability side, so whether we can scale it or not. So here, can we scale this process rapidly? So we have broken this whole manufacturing technology into 4 parts. One is plasmid DNA, you can make, store, and as a unit operations. Then you can take plasmid, linearized plasmid, and you can make the mRNA, you can make it, freeze it. You can make CLNE at separate place, 2 to 8, you can save it, and then any place where you want to go, the 4th place, you can mix with those both things, and you can make the lyophilized vaccine. So we have broken this into 4 parts. So if we have to go, we are thinking with WHO that we may have a couple of other manufacturing sites around the world, maybe one in Africa, one in the Indonesia, one in the Colombia or Brazil. So these kind of things we are talking. So how we can take these technologies in broken parts and finally make it one at one of the places. But this gives example for the initial move is we will have the drug substance as mRNA, CLNE, and we can go and fill and finish and make a lyophilized product in a joys of country. So now the transfer of the technology is comes. Yes, we are ready to transfer the technology because this is going to save millions and millions of lives because COVID is not going away and we can see this Omicron has just came and I do not know how it is going to behave. In a couple of weeks we come to know about it, that how bad infective it is and what kind of pathophysiology is coming. But what is happening because of need of the vaccine, till we all are not vaccinated, no one is safe. This is what we at Genova say, that if till we all are not vaccinated, no one is safe because virus will have a chance to go and mutate and come back in a different form, which may be possible that vaccine will not be effective. So we have around, for drug substance, we are creating a facility around 1.3 billion doses. At present, we can produce 350 billion doses a year. Once the government gives approval, this will be there. And certainly, we are very keen to take this technology to LMICs to many countries. And we will also share the IPs with the country of need. So this basically, I can say that one by one, we have tried to solve all these problems. And if God gracious, the vaccine will go. But this is another slide I wanted to present to this group. This is a very, very important factor. And see this. So what we can do is change in the plasmid variant only. So your CLNP is same, your enzymes are same, your pro-factor leukocyte and lipids are same. We can have this as a national stockpile. But in this funnel, the new sequence of plasmid we can add and finally the vaccine can be produced. So this is a unique advantage of this platform which has come, that this is a disease agnostic platform. Means you don't have to change the technology, you don't have to change the manufacturing facilities, and you can keep stockpile things ready, and wherever this kind of pandemic happen, you can turn it around. So on the right side, if you see, the roughly time turnaround injecting into human could be 50 days. At present, we are very seriously considering these 4 areas to enter, tuberculosis and dengue, and I may come back to Dr. Khanna, who is the expert into it. He has made the vaccine, which is protein-based, but we collaborate for the dengue on the mRNA-based. Chikungunya work we have already started, and malaria is again, we are, I feel, still defeated because we have not able to give the malaria vaccine, which can eradicate the disease. Yes, one of the vaccine came, it's a great knowledge, it's great step forward, but still it need to improve a lot. And so malaria program. So today I want to announce that we have started working on MA-based vaccine for mRNA-based vaccine on malaria in collaboration with Johns Hopkins and NIH. So this is a presentation, and I feel that vaccination, affordable vaccination, efficient vaccination can make a huge change in poverty versus life expectancy. One of the Gates Foundation program is based on rotavirus. Again, our past Secretary of DBT, Professor Khan developed, and that has changed such a big field, and it has saved hundreds of millions of lives of young children in the developing countries. So I feel that mRNA can offer a low technology cost, a very effective vaccine, and which will make a huge, huge difference. So with this, I thank you slide. So thank you, Dr. Malhotra. I'm open for the question if time permits.
Thanks again. It was really interesting and nice talk. And I think, I can tell you that I learned quite a bit on some of these new findings which you have, particularly stabilizing the mRNA. And I'm sure one thing which I don't know whether you mentioned or not, one thing which you can really achieve the goal in malaria vaccine is by putting a combination of mRNA, because we need vaccines for both the transmittable stages, mosquito stage and blood stage. So here with mRNA technology, you can put them together, 2 or 3 at various stage vaccine candidate and do a very good job here. So we have few questions which because of limitation of time, we will just ask you few questions some of the students have proposed. If you have time, can
yes,
you?
I'm ready, please.
Sure. This is Shreya from NII. I have a question for sir. What kind of
So here, it's not that straightforward. It's a bit complex, but at present, what we are targeting is basically MA1 from the blood stage, CSP from the erythrocytic stage, and PFS 48-45 plus PFS 230 from the gametocyte stage. So as Malik said, yes, we are targeting all three together. But we will be very open to look into the new, because we do not know whether they are going to give the best targets, but these are the known best targets available as of today. So we are making them independent also, and we are going to mix and match and see how the effect is going to come. Because I come from the drug development, initially my PhD is from the drug development field. Till you don't get sterile immunity, there is no immunity.
Yeah, I do agree with it. And you know that the GSK vaccine is not giving more than 40% protection. So I think you are on the right track in this direction. So we will take one or two more questions. Why protein-based vaccines are not successful in malaria?
So basically, the vaccine which has been there, if you ideally see if this hepatitis B particle-based vaccine is CSP vaccine. So I think there are 136 molecules of hepatitis B surface antigen and 1 molecule of CSP. So the whole power of the vaccine, your immune system is screwed towards hepatitis B. It is a very good hepatitis B vaccine. Now coming back to the malarial proteins, so yes, we have a lot of good targets, but they have to be combined with adjuvant because when they are presented to the cells, they need a lot of help. So we also have a program on adjuvant discovery, and when I came back here, adjuvant produced by us, it gone to the malaria vaccine program of ICGB led by Chetan. So at this point, we have to make it more effective, and we need more powerful adjuvant. That is one of the reasons. The vaccine targets, antigen targets in form of protein is not problematic, but it is we need more power into those antigens and run by either by antigen design somehow or by the adjuvant.
Sanjay.
So one last question from Professor Chauhan, who is also listening to you very minutely. So please.
Sanjay, excellent lecture as always, but I have a question about the early mRNA vaccine trials. And you have I'm sure you've already looked at it. There was a report about 4 months back that Moderna gave fourth of the
Professor Chauhan, can you be a bit loud? I'm having a problem in listening. Can you please?
Do you hear now?
Yes, I can hear now,
okay.
Very well.
All right. So this is about the early phase 1, phase 2 trials of the Pfizer and Moderna vaccines, because everybody was in a great rush, so they used 3 doses and settled on the middle dose. But later on, when they actually immunized with 1/4 of the dose of Moderna, they found that the immune responses were no less than when they had given large amount of Moderna mRNA. But FDA, having approved a certain regimen, will not allow anyone to change the regimen. So do you feel a lot of these early companies are stuck with higher dose because they can't change the doses? That's the FDA says, and you obviously have a chance to look at lower doses.
Yes, sir. This is a very important question. So Moderna started with 50, 100, 150, 250, and they have to scale back because a lot of toxicity was coming. But they wanted to raise it, so still they have gone with 100 microgram per dose. In case of Pfizer-BioNTech, they tested 10, 30, 60, 75, 100. But because of toxicity, they have come back and they said this is the negotiating point where 30 microgram can go. In our case, we cannot say at that time, but now it's quite obvious. What are the clinical correlates of protection? So based on the clinical correlate of protection, you have to give the titer up to that level. But we have found from self-amplifying mRNA we are getting the similar kind of titers only in 10 microgram. So at present, what the data has come from few thousand patient trial, our vaccine is super safe. And then we are not using PEG, we are not using cholesterol. So these things should also give a better safety profile for our vaccine. So now they are for booster dose, they are scaling back like Moderna has done 25 or 50, same way they are scaling up, scaling down in terms of going for the booster or going from the pediatric doses. So yes, this is a very important question. We had some opportunity to look into that, and that was obvious because we are only going with 10 microgram dose. Another part is the whole protection is not only based on the titer, this also comes from cellular immune response. So I hope cellular immune responses are very well, and that is at present giving in lowering the chances of moderate to serious infection. So I think this is one of the point also need to be seen when we are deciding the doses to go.
Thanks, Sanjay, for accepting our invitation. Though we missed you here, and we will be looking forward to your visit at ICGB. It has been long due, but thanks for really a great talk. And
Thank you, sir, and what honor and what a privilege and what to be on ICG platform. Thank you very much.
thank you, thanks a lot, and please visit us whenever you are in Delhi.
Sir, I will. Thank you, sir.
Okay. So do you hear me?
Yeah, just a minute, Christian.
Yes, yes, we can. We can. Christian.
Okay, so you tell me you tell me when you want me to go, okay? I'm ready.
Okay. So, good morning, good afternoon, good evening, depending upon in which continent you are. And welcome to this session on malaria parasite biology, where we have some eminent speakers this time. I would request if you need to ask a question, please put in the chat box and we can ask after the talk. So our first speaker is Professor Christian Dore. He is from Royal Melbourne Technical University from Australia. Christian did his PhD from Lausanne, Switzerland, and now his group is working on phosphosignaling and kinases in the plasmodium, and he has done some remarkable work to understand biology of these kinases with respect to survival in the human host as well as invasion. So today he is going to speak on host-directed therapy for infectious diseases, phosphosignaling as a target. Christian, over to you.
Thank you. So first of all, I just want to extend my thanks to the organizing people, especially Asif and Pawan for their kindness. And I have a long history with India, and I've been to ICGB a number of times. I've got a number of friends across New Delhi and elsewhere in India, so it's really delighted. I'm really delighted to be here. I'm just really sorry that it can't be in person, but hopefully it will be in person in the following years. So let me see if I can hide this. Let me see here. I'll put it down here. So what I'll do today is tell you about a part of what we're doing, which is mostly about host-directed therapy rather than parasite kinases. Okay. So what I'll do, I'll just very briefly give you the history and how the characterization of the kinome of Plasmodium falciparum led us to be interested in host cell signaling. And of course, I'll spend most of my time on Plasmodium, but I will do short incursions into Hep C and Wolbachia. And then I will finish with a few considerations of a very strange thing that we recently discovered, which deals with drug addiction in malaria parasite. And finally, I'll finish with one slide on the potential host cell kinases anti-infective drug discovery at large. So, our origin question was, in malaria parasite, a big question, how is the switch to gametocytogenesis regulated? So here you have a gametocyte. Here you have a red blood cell infected with an asexual parasite. They're very different. Nobody understands how that works. So what is interesting is that if you look at this single little parasite here, it is able to become either a male or a female gametocyte or an asexual. And though there's 3 different cell fates, there's no sexual chromosome. So how does it do it? And when I started in malaria, which is almost 25 years or more than 25 years ago now, none of this was known. So I decided to actually try to take some clues from systems that are better understood, such as yeast, because if you look at what makes a gametocyte and gametocyte, it's compared to an asexual, is first cell cycle arrest, then a new set of genes that are transcribed, and then morphological changes that you can see here. And so this is actually what happens in the differentiation of yeast from A to alpha. So in yeast, it's called A and alpha rather than male and female. But an alpha cell will mate with an A cell, and they will form a zygote from which will have spores. So it's in Plasmodium we have sporozoites instead of spores, but it's basically the same. And what is interesting is that at the time when I started this, what happens in yeast was already understood. So very briefly, in yeast, for yeast to become sexual,
I'm sorry to interrupt you.
Yeah.
But your slides are not visible, and several people have indicated that on the chat box. I'm really sorry, but I think organizers need to fix it up.
Okay.
So, sorry.
No worries.
Yeah. I was the brave one who indicated it to you, but there's several people have, have written in the chat box.
Yeah. No, I don't have my chat box on, of course. Oh, not visible. Oh, I see.
No, but don't worry, that they will fix. I'm sure Asif will fix it.
Okay. Christian, maybe you have to stop your presentation and do it again, then it will work.
Yeah.
Maybe just stop sharing or just stop the display and then.
Yeah. Do you want me to stop sharing for a while? Okay, I'm stopping. Oh, it says okay. You share. Stop share. Okay, I've stopped sharing.
Yeah, we can still see you. Yeah, maybe you can start the sharing again.
I'll start again. Okay.
Yes, please.
Screen. Can you see it? Can you see it now?
Yes.
Now I can see.
Can you try moving and you will know that if it is moving.
Can you just move a slide back and forth? I mean.
Okay. Yeah. Can you see the change?
Yeah, it did change. Yes. Yes, it is. It's
Okay, no, good. So let me know. Give me a shout if it keeps not working. Okay. But I can put this here. Okay, so I will start from here because basically I know this time is at a premium. I don't want to spend too much time. So it's just I was saying that we're using yeast as a model to actually try to understand sexual differentiation in plasmodium. And in yeast, what happens? A hungry yeast that wants to become sexual emits a pheromone. This pheromone is sensed by a receptor at the surface of another yeast cell. And then this triggers a signaling pathway that ends up in this MAP kinase pathway. And the MAP kinase substrates are transcription factors that actually mediate the shift in the transcriptional repertoire. And another substrate is a cell cycle component, FAR1, that actually inhibits the cycling-dependent kinases that drive the cell cycle, so you have cell cycle arrest. So at the time, I thought, it was 25 years ago, I thought this is going to be the same in plasmodium. So let's look for these MAP kinases. So we did. This was prior to the genomic age, so we didn't have the genome sequence. So we did it by PCR with degenerate oligonucleotides, and we actually found 2 MAPKs in plasmodium. Great. And then we say, okay, let's try to find the MEK, M-E-K, the MAP kinase, or MEK. And we tried that, and we never found it. And I won't go into the detail. We spent immense amount of time in different oligos. It never worked out. And then the genome sequence of plasmodium was published. So we very rapidly sorted out the kinome. So this is our original tree in 2004. This is a bit of a better, more visual way of saying the same thing. And so if you compare the human kinome and the plasmodium kinome, the first thing that that becomes obvious is that there are families that are lacking. There's no TK tyrosine kinases in plasmodium. There's no STE, we'll come back to that. But all the other groups are represented. There are, and some people in the audience will kind of be very, will kind of smile at that. But we also found out that there's a number of calcium-dependent kinases in plasmodium that are absent from the human kinome, and also FIKKs that are actually a brand new family of kinases. But more to the point of this talk, this analysis showed that there is 2 and only 2 MAP kinases. So we did a good job doing this before the sequence was available. But more importantly, there were no plasmodium MEK or MEKKs, which fall in the STE group. So we nevertheless thought, maybe this MAP kinases that we found will mediate sexual differentiation. So let's see what happens if we knock them out. So we did a large campaign knocking down actually all the plasmodium kinases, and we found that actually you can knock out PFMAP1, and it doesn't matter. The parasite can undergo the entire life cycle, including sexual differentiation. So clearly, MAP1 is not needed for sexual differentiation. And MAP2, I have to put this somewhere else. PBMAP2 in Berghii, at least, you can knock it out. It's not essential for asexual growth, but parasites that lack MAP2 are still able to make gametocytes. They don't make gametes, it's another story, but they're perfectly able to make gametocytes. And for those who are interested, We actually published this now 10 years ago, actually, goes time flies, in this Nature Communication paper. But more to the point now, this was our hypothesis that gametocytogenesis in malaria parasites is regulated by 3-component MAP kinase pathways. What we found is that there are actually no 3-component MAPK pathway in the falciparum genome, and you can actually knock out the MAP kinases and you don't impair gametocytogenesis. So clearly this hypothesis is wrong, you know, the saying of a beautiful hypothesis by an ugly fact. But nevertheless, what was weird is that before we knew that there were no MAP kinase or MEK in the parasite, we actually did a few experiments with inhibitors. And U0126 is an exquisitely selective inhibitor of MEK, and it actually does kill the parasite. So we were wondering what was happening. And then to make a long story short, what was happening is that this effect of the inhibitor is mediated by the host erythrocyte MEK. And we actually published later that host cell P21 activated kinase can actually phosphorylate host MEK, host cell MEK1. We don't know how this starts. In other systems, you know, the extracellular matrix interaction with the cell in fibroblasts, for example, triggers this pathway. We don't know how it's triggered in falciparum. But what we know is that once this is activated by phosphorylation of SER297, you have an auto-activation of MEK and the pathway is activated. And so again, this is published as well, so you can have a look. Now, the main point is that PAC, which is activated in infected cells, has a multitude of substrates and pathways that you can see here. The one I mentioned so far is, oops, what's happening here? Something went wrong, sorry. Okay. So I mentioned the effect of PAC on the MAP kinase pathway, but you can see it does lots of stuff in the cell. So clearly, if PAC is activated, there may be lots of things that are activated in an infected erythrocyte. So basically, we need a comprehensive tool to interrogate the entire host cell signaling. And so this tool exists, and it's an antibody microarray. And so it's a company called Kinexus makes this antibody microarray. Now the latest versions have more than 1,000 pan and phosphospecific antibodies against human signaling proteins. So here we deal only with human cells, or human proteins. And briefly, the way it works is that you have two identical chambers on the array. You make an extract of protein. In our case, it's infected and uninfected red blood cells. You fragment the protein, label it, and capture the peptides on the array here. And so these chambers are identical, and there's two dots for each of the antibodies. And then what you do, you just compare your two chambers. And so this is just an example when you treat a human cell line with epidermal growth factor, and you can see that here in these arrows, here this particular phosphospecific antibody does not give a signal here, but in the treated cell it does. So this means that this particular phosphorylation event is triggered by treatment. And so we did the same with uninfected and infected erythrocytes. But of course, we first wanted to validate the approach with a system where we could actually validate the hits by reverse genetics, by doing siRNA. And so we can't do this in erythrocytes, as you know. So with the first example we did was with Hep C virus. So it was done by Reza Hakshinas, a very talented postdoc in the lab. So I'll just give you a very short example. So this is the type of signal that you get from this antibody microarray. And so this is a heat map. So there's no change if it's black. It's decreased if it's red or if it's green, it means a decrease compared of infected cells in the infected cells compared to uninfected. And if it's red, it means you have an increase in this. So this is just a very simple example, the backstat pathway. So you can see that you can see that if you look at the protein itself, STAT1, the transcription factor STAT1, there's no change during infection, either at 6, 12, or 24 hours post-infection. It's all black. But if you look at phosphorylated STAT-A, it's vastly upregulated. So this tells us that infection with HCV triggers phosphorylation of STAT-A, STAT5A. And so we took about 100 hits that were modulated by infection, did an siRNA campaign, and found that some of them, when you siRNA and you measure the ability of the cell to support viral replication, some of them have a very drastic effect. And so here, the STAT-A, if you siRNA STAT-A, then the virus doesn't grow well. And interestingly, some of them are kinases, of course. So if you look at this example here, MAP4K2, it's a kinase that is vastly activated by infection. If you siRNA it, you actually stop the virus growing. Now, interestingly, I've got a friend in Harvard called Nathaniel Gray, so he's a kinase inhibitor guy, and he had just submitted, just published this paper called, Discovery of Type 2 inhibitors of MAP4K2. So I gave him a call and I said, I bet your new inhibitors has antiviral properties. Can you send us some? So he sent us some of this compound, and you can see that at low micromolar range, it starts to completely inhibit viral replication. So this shows that this microarray, where you actually first find human kinases that are modulated by infection, and then find inhibitors against these kinases, you can actually find anti-infective compound. In the interest of time, I will skip the Wolbachia series of experiments because it's been published. But now back to malaria. So most of what I will tell you now in the next 5 minutes has been published last year. So you can have a look at these papers. And the first author is Chaka Dele, a very talented postdoc in my lab. And part of the work is also done by Taylor, who is a PhD student in the lab. So we actually ran this microarray on rings, comparing red blood cells that are either uninfected and comparing them to red blood cells infected with rings, trophozoites, or schizoids. And this is an extract of the most interesting data in terms of fold change, positive or negative fold change. And you can see that there are a number of things that are interesting. So in this data set, MEC also was activated, so it confirmed our previous data. I will talk about BRAF, because BRAF is also in the MAP kinase pathway, and you can see that it's activated quite strongly at all 3 stages. And also about MET. So MET is this is also phosphorylated. The phosphorylation of c-MET, I'll tell you what this is in a minute, is actually increased on the number of phosphorylation sites. Okay. So this is the MET pathway. MET is another word for the hepatocyte growth factor. It turns out that it is actually expressed in the erythrocytes. It can be phosphorylated on the number of sites, and it controls a number of downstream pathways here. So this is just to show you the data, to show that all these are antibodies against specific phosphorylation sites, and you can see that most of them are activated at trophozoite stage, right? And so we checked that by these are activated sites, so they are in the activation loop of the kinase, and they're vastly activated by infection. And we verified this by Western blot. If you take extract from uninfected red blood cell or infected red blood cells and do a Western blot with an antibody that recognizes the protein itself, irrespective of the phosphorylation status, you can see that you have same amount in infected and uninfected red blood cells. But if you look at the phosphorylated site version, you find the signal only after infection. So clearly infection results in the activation of c-MET. And so if this is the case, you would expect downstream pathway to also turn up in our array, and actually they do. All these things are actually activated in infection. These are as well. Some of them are not, which makes a nice negative control, but this is an inhibitory site of PAC. So this actually may be one way whereby PAC is activated, as I mentioned before. And this feeds into the MAP kinase pathway. So just a few words now about how to deal with these large data sets. Because these arrays generate huge data sets, this is just a little fraction of the data set that we get from Kinexus when we do the experiment. And so, as I told you, we focused on MET because it was interesting, but there may be thousands more things that are interesting there. And so we need a system to actually explore this more comprehensively. So this is adapted from a paper in 2016 that described the human protein kinase network. So of course you can't read it, you see it's very bushy. If we extract from this data set the hits from our microarray, this is more simplified but still pretty hefty. And this is the same depiction, the lead signals from our Plasmodium study. So these are all human kinases that are, and their substrates that turn up as modulated by infection. And now, if I show you in red, though there's a bit of red here, this is CMET, and this is a bit of red here, this is BRAF. This is the only one that we actually looked at, and we are looking at. But there's a huge depository of interesting things to go, but which one how to prioritize that? So now we have actually teamed up with mathematicians to actually deconvolute this strong, very complex data sets using a random walk strategy. So it's a mathematical tool. We have submitted a paper now. So, but basically you go from this very bushy thing to an ordered pathway that are proposed. So we'll use this now for all our data sets. But anyway, now back to the biology. So of course, now we know from our array that in infected erythrocytes, we have an increased phosphorylation of MET and an increased phosphorylation of RAF. So does it mean that the parasite needs them to survive? So to test that, we take inhibitors of both MET or RAF, and you can see that we have strongly sub-micromolar efficacy of some of these inhibitors of c-MET on both falciparum and Nolazai, and the same is true for the ones in blue and black are RAF inhibitors. So it seems a little bit like in our HCV study, we found human kinases that are activated by infection, and if we block this, we kill the parasite. So that's really interesting. Now with MET, this is trametinib. This is an inhibitor of MEK. So as you remember, MEK is the first kinase that we found to be activated in infected cells by infection. And it turns out that a few years back, this molecule trametinib was approved for the treatment of melanoma because MEK is also involved in cancer. And so we bought some. We tried it on falciparum and on Nolazai, and you can see we have exquisitely low IC50. So this is a very good parasite killer. Now, with both trametinib and the c-MET and the BRAF inhibitors, Sarah Jackson, who was an honor student in my lab, and then this continued now with Taylor, I asked them to actually use the standard protocol to try to derive resistant parasites under sublethal treatment. So we used the protocols of David Fiddock. He gave us good counseling on that. And so basically, we did this in triplicate and tried to generate resistance to these inhibitors. So we were very happy not to be able to find resistant parasites against the BRAF and the c-MET inhibitor. So that was great because, if you target a host kinase, it's much more difficult for the parasite to become resistant because it can't just mutate the target and then selected under treatment. So that was great. Now, interestingly, with trametinib, we very rapidly got resistance. And so this is the first experiment, and you can see the IC50 curve is shifted to the right, so it means the parasites are more resistant, which suggests that there is enough target in the parasite. This is the only way you can get such fast resistance, I think. But the interesting point here was that if you look at the entire curve, we had this very weird phenotype here, where actually there is an optimum concentration of the drug. Here it's a bit lower than the 1 micromolar, where the parasite grows happily. And if you add more drug, it dies. But if you remove the drug, it also dies. So these parasites have become dependent on the drug. which is very weird. So I think what may happen is that if you have a red blood cell here, parasite comes in, and so there's a defense mechanism that involves the host immune. So the parasite has to have a defense against it, so it's got some way to actually counteract this mechanism so that it can survive. But now, if you come with a MEK inhibitor and you inactivate this attack, as you know, parasites get rid of what's not useful, so probably quite rapidly, the parasite loses the ability to actually respond to this threat by MEK. And now, when you actually remove the inhibitor, the parasite has lost the ability to counteract this defense and dies. So that's I think what happened. We have a few more data, but I don't have time to go into. Oops, sorry, into all the details. But I think watch this space because this is really interesting. I think. Now back to the big picture thing. This is such a powerful approach, this microarray approach, that we actually have now data on falciparum gametocytes. We've got data on Nolozi, on Toxo, on Babesia, and on worms even, Hemocontortus. As I said, we published a paper on Wolbachia already in terms of bacteria, and we have ongoing work on other type of intracellular bacteria. We published a study on HCV. We have ongoing work with HPV, influenza, and SARS-CoV-2, importantly. So we just got the data from SARS-CoV-2, so there's some very interesting target there. So basically, what we already know is that you can find similarities between how the cell responds to all this, especially with respect to cytoskeleton dynamics, which is not a big surprise because both viruses and bacteria and parasites have to enter the cell, so they need to mobilize the host, the cytoskeleton system. And so this could mean that we actually could get to broad spectrum anti-infectives. This is a paper that we published a year ago or so with Reza, where we actually review the literature. And so here you have a target kinase that is activated by virus pathogens. Here the pathogen class, the species, and if the kinase is reactive, the kinase activity of the kinase is actually required or not for the virus or the bacterium or the parasite to get in. And these are the references, right? So if you look, for example, at c-Met, which I've talked about already, you can see that c-Met is actually mobilized by a number of viruses, including influenza and Ebola virus, by Listeria, which, as you know, is a serious pathogen, and by malaria, not only in sporozoites, as published in the literature already, but also during the erythrocyte stage, as I mentioned before. Which means that, if we could have developed c-Met inhibitors, we could actually have broad spectrum anti-infective that could be useful for viral, bacterial, and parasitic. And so I'll stop here because I know that time is at a premium. So basically, this was done while I was before I came to Australia, and then I came to Australia, and people who involved in this were mostly Reza here, and Jack Adderley, who's now my postdoc, and he's here too. And all the drug addiction, the very weird and very, I think, fascinating phenotype of drug addiction was first discovered by Sarah and now being pursued by Taylor. So I'll be happy to take any questions. Maybe I'll stop sharing my screen and go back so that I can see if anybody has a question.
Thank you, Christian. Thank you very much. It was a very interesting piece of work. We actually enjoyed it. So I don't see a question in the chat, but if someone has a question, I suppose Pushkar has a question. Yes, Pushkar, please.
Christian,
yes,
very
Pushkar.
nice. Yeah, very nice. I could have asked you independently, but have you looked at the sporozoite or the hepatic stages? Because these pathways are likely to be, if not anything, even more rampant
Yes.
because they're dividing cells and so on. And then you may have pan inhibitors, pan host inhibitors, which may prevent.
Absolutely, no, absolutely. No, I would love to do that, but there are kind of technical hurdles because you need about 1 million cells to actually have good data. 1 million infected cells compared to 1 million non-infected cells, and so we could actually, and you also need a very good multiplicity of infection because you want most of your cells to be infected in your infected So it's challenging with sporozoites and hepatocytes, but it's probably not impossible. But
yeah, the other advantage is that you can use an siRNA like Maria Mota has done.
No, absolutely, yeah. Actually, that's very true. And Maria Mota, in her kinomes siRNA campaign for the liver stages, c-Met actually turns up as one of the potential targets. Okay. So that's really cool.
Yeah. Thank you, Chris.
Thank you, Pushkar. Yeah, thank you, Pawan.
Do you know the substrates for these, some of these host kinases? And
I didn't hear well. Did you ask for the substrates of these host kinases?
Plasmodium proteins.
Yeah. So, these kinases function in very well described pathways in other cell types. That's for sure. So. We don't know exactly which are the substrate, but we can infer from the fact that for example you start with c-Met and you go eventually to PAC, and all this is activated. So I think what happens is that we have quite the conventional pathway that are activated there. Now, why? Maybe your question is also why does this kill the parasite? What is the effector downstream? What is the substrate or the final effector of the pathways that is recovered by the parasites? And this is a very good question. We still don't know. We think there may be, part of it may have something to do with the activation of transporters, because these pathways, many of these pathways, the BAP kinase pathway, for example, can actually activate transporters. And we all know that parasites need to activate lots of transporters to the novel permeation pathways for it to gain nutrients. And so maybe this is why this would fit with the idea that MET is activated at the trophocyte stage and dies at the trophocyte stage if we block it. So this means that it maybe have something to do with the nutrition. We don't know really.
No, just a suggestion: if you put these drugs and then do the transcriptomic analysis of the Plasmodium protein.
So, can you repeat the question? I didn't.
If you add these drugs in the culture, and do the transcriptomic analysis or phosphoanalysis of the Plasmodium proteins.
Yeah. No, absolutely. We actually are doing this. So we are looking more at what's happening in the red blood cells. So what we are doing now is treat the cells with these inhibitors and then do a secondary microarray analysis to see which pathways are affected. Yeah, absolutely. Yeah.
That will be very interesting.
Yeah.
Yeah, Christian, just a quick concern. So in this host-directed therapy, it is good because you don't get mutant and you don't get resistance and all.
Yeah.
But also, the selectivity is a concern. So does it have a high selectivity? I mean, the kind of
Yes. So we try to do most of our stuff with inhibitors that are so-called type 2 or type 3 inhibitors, which are different to type 1. Type 1 are just the ATP competitors. So selectivity is not great. But if you take type 2, there are allosteric inhibitors. For example, these MEK inhibitors like trametinib, they actually block the MEK enzyme in its inactive state by binding to a pocket that is adjacent to the ATP binding cleft. And so this is very, very selective.
Okay. There's one question from Manoj Yadav. Did you notice any change in the expression profile of ROP complex gene?
Yeah. No, we have, because we actually not yet. That's a very good question, Manoj. But we have not yet looked at the transcriptional response of the parasite to all these things. Because, but it will be very interesting, yeah, absolutely, because maybe there's some signaling that actually tells the parasite to start expressing, these transport genes, something of the ROP gene. That's a good point, yeah. Thank you.
Okay, I think that's it from the audience side, and I will thank you again, Christian. Thank you very much for joining late in the evening.
I love to be here. Thank you. It's a real pleasure. I'll keep on, and I'll try to be there as much as I can for the. It's late at night now, but I'm definitely interested. Thank you. Thank you, everybody. Bye bye.
So our next speaker is Dr. Saman Habib. Saman is Chief Scientist at Central Drug Research Institute, Lucknow. And Saman's group is working on transcription and translation control, especially in the organelles of the parasite. And today she will speak on unique features in Plasmodium organelles, biogenesis and DNA repair of iron sulfur complex. So some of the students were interested in this two images of two different contrast building. So I can give them history of this Chhatar building later, but maybe you can just start with that.
All right. Can you hear me?
Yes, yes. And we can see your
All right. So thank you so much for getting me back to ICGB, although it's virtually, but it's very nice to see everybody again. The two buildings first then, as Asif said. So this is because I started my work in this beautiful building that you see to the left, which is called the Chhatar Manzil, which was the palace of the Nawabs, and CDRI was given this building when India became independent, and CDRI was set up in 1951. So this is where I worked when I joined the institute in 98, and I worked here for about 15 years before we moved to this new building. So I always acknowledge Chhatar Manzil's contribution to my work. Anyway, today I will talk about I work on organelle biology in Plasmodium, and I'm just going to discuss two aspects of that today. One is iron sulfur cluster biogenesis, and the second is DNA repair in organelles, and we'll try and see some unique features and proteins of these pathways. So if you see this slide, this is for students, obviously. If you look at the genomes of the malaria parasite, you have the nuclear genome, but in addition to that, you have the epicoplast genome, which is a very reduced plastic-like genome, which has lost photosynthetic genes. And now there is a 35 kb circular DNA in which genes largely coding for ribosomal RNAs and proteins, but also coding for the RNA polymerase and a few other open reading frames exists. And it performs very essential functions in the parasite. You know that it performs the Fas2 pathway, which is very critical in the liver stages, and also performs the isoprenoid biosynthesis pathway, which people believe by various experiments that you know of, which is because because of the isoprenoid synthesis pathway that the epicoplast is actually essential for the parasite. Apart from that, there are other pathways. I'll not talk about them. I'll deal with one later. There is this very small mitochondrial genome, which is a 6 kb repeat DNA element. It is among the smallest mitochondrial genomes known in nature, or maybe the smallest genome known in nature as far as mitochondrial genomes are concerned. Encodes for 3 open reading frames, which code for proteins cytochrome oxidase 1, COX3, and cytochrome B, and also for a series of fragmented ribosomal RNAs, right? Now, if you look at how functions are performed, so if the coding sequences are so limited and no proteins or very few proteins are made in these organelles, then all the essential functions performed by these organelles are because proteins are imported, are nuclear encoded and imported into these organelles through various systems, largely through the secretory pathway, but sometimes also through the non-secretory pathway. And for the epicoplast, they have to cross 4 membranes because the epicoplast is a secondary endosymbiont and therefore is surrounded by 4 membranes. And the mitochondrial membrane, as you know, is a 2-membrane system. Both organelles, this is a picture from our lab, from an infection in the RBC. And as you can see here, the mitochondria is labeled with red and the epicoplast in green. And they occur as elongated structures in RBCs in the strophozoid stages, but they become highly branched as schizogony takes place. And in the liver stage, they become really enmeshed, large web-like structures in hepatocytes. And they have been of interest because they also give unique vulnerabilities to the parasite in terms of drug treatment. And doxycycline, for instance, is an anti-epicoplast molecule and is used in clinical treatment for malaria. If you look at the genome, and I've circled here in red, in the epicoplast, apart from these things labeled here, RNA polymerase, large subunit ribosomal RNAs, ribosomal proteins, tRNA genes, is this protein called YCFB24 or ORF470, which is actually a gene coding for SUFB. Now, the SUF pathway is the pathway for iron sulfur cluster biogenesis. And if you look at iron sulfur clusters, what are these clusters? These clusters can be 2 iron 2 sulfur clusters, which are present as post-translational modifications on proteins in this rhombic state, which are coordinated to cysteine or histidine residues on proteins that harbor them. Or you can have a cuboid iron sulfur cluster of this shape, where you have 4 iron and 4 sulfur molecules, again coordinated onto cysteines or histidines largely on proteins. iron sulfur clusters are among the most ancient post-translational modifications known, because when the Earth's atmosphere was reducing, iron sulfur clusters were important post-translational modifications, and they take part in redox and non-redox catalysis and electron transfer reactions by proteins. Several proteins in the epicoplast and several proteins in the mitochondrion are substrates for iron sulfur assembly. So they require iron sulfur clusters for their functions. In the epicoplast, you have the ferredoxin reductase system. And two enzymes of the critical DOX-P isoprenoid biosynthesis pathway that I mentioned earlier, the last two enzymes of this critical pathway also require iron sulfur clusters for their activity. Lipoic acid synthase requires it, MAIA-B protein requires it, and DNA replication and repair proteins that I'll talk about today also require iron sulfur clusters. Similarly, in the mitochondrion, you have these enzymes, very important enzymes of the TCA cycle, require iron sulfur clusters for activity, and a DNA repair protein that I'll talk about also requires this cluster. Now, the question we asked was this. In nature, there are three major iron sulfur cluster assembly pathways. You have the ISC pathway, and the NIF pathway, and the SUF pathway. The ISC system, as is written here, is found in bacteria and eukaryotes, and in eukarya, it's largely in the mitochondria of the cell. And the ISC pathway is a major pathway in bacteria, whereas the SUF pathway in bacteria is a supportive pathway to the ISC system. The NIF pathway is largely found in nitrogen-fixing bacteria, and the SUF pathway, as you notice here, is found in archaea, bacteria, protozoa, including Plasmodium, and plants. And notice that it's absent in mammals and absent in humans, therefore, and then since the SUF pathway is absent in the human, can we then look at it as a putative drug target for malaria and to see if it's essential for malaria survival. So early on, our lab had started looking at this, and we showed that the ISC and SUF pathways partitioned to the mitochondrion and the epicoplast respectively in falciparum. And Peter Preiser's group then showed that the SUF pathway is essential for epicoplast maintenance and parasite survival in blood stages. Now, if you look at iron sulfur cluster assembly, you have the basic mechanism for the assembly is that you need sulfur mobilization, and the source of sulfur in the cell is cysteine, which releases the sulfur as a persulfide and gets it to a scaffold protein that then assembles an iron sulfur cluster using iron from an iron source. So both these come onto a scaffold. The scaffold assembles the protein, and then a number of transfer proteins then transfer this iron sulfur cluster onto the target apoprotein, which requires this cluster for activity. So we had worked out some years ago the iron sulfur SUF pathway, and we expressed each of the proteins of the SUF pathway recombinantly, did functional analysis, and ultimately delineated the SUF pathway. And we were able to show that you have a SUF-S-SUF-E interaction, and SUF-S is the main cysteine desulfurase that transfers the pulsarfide to SUF-E, and SUF-E enhances SUF-S activity. And we had also shown that if you use an inhibitor that actually forms an adduct with PLP, which is a pyridoxal phosphate cofactor for SUF, then you can inhibit SUF-S activity and actually cause parasite death. And then we'd also identified the components of the scaffold complex, which is like this, structurally, also shown that you can inhibit the sub-C-sub-D interaction, and also identified the transfer proteins, and then shown how this pathway works. Now, what was interesting in our work was that so it's been shown by knockout studies in Berghai, in the Plasmogem, if you look at the Plasmogem database, that if you make knockouts of sub-D, C, and E, then the parasite doesn't survive in the blood stages. And our work showed that if you make a conditional knockout of sub-S, which is a cysteine desulfurase, by using a flippase system, a flippase FRT system, then in the mosquito stages also the parasite does not survive. And the reason for this is that although you get oocyst formation in the midgut, you see the same number of oocysts are formed, but sporozoid formation does not occur. So the development of sporozoids in the oocyst is severely hampered. So this is the normal parasite, the trap one. This is the red one, is the knockout parasite. And as you can see here in the wild type, you see very nice clear sporozoid formation in the oocyst, but here you do not see sporogenesis, and you see just these dead parasites. And also you do not see therefore mature sporozoids in the knockout line. So it's very, very poorly infective, sporozoids are not formed. So clearly it's very important also in the mosquito stages. And this is important as if you want to look at SUF pathway as a target in malaria. And very recently, just maybe perhaps last month, the paper has come on toxoplasma in which they've made knockouts of SUF genes and also shown that it leads to the loss of the epicoplast and parasite death consequently, and disruption of the ISC pathway in the mitochondrion results in differentiation into a stress-resistant stage. This is also an interesting paper, people interested can look at it. So clearly both in the malaria parasite and in toxoplasma, these pathways have immense importance. Now, what I want to talk about next is the ISC pathway of iron sulfur cluster biogenesis in the mitochondria, right? Now, we have this ISC pathway in the mitochondria, and it's also important because mitochondrial ISC pathways are the source for iron and sulfur that helps of assembly of iron sulfur clusters in the cytosol. So they also have a role to play in cytosolic iron sulfur cluster assembly and are therefore essential for life in life forms. So a same system, ultimately you have a cysteine desulfurase that will mobilize, but sulfur, you have a scaffold complex, but in the ISC pathway, the scaffold and the cysteine desulfurase function together. So it becomes a larger complex, which has both the scaffold and the cysteine desulfurase forming a large complex together. And then you may or may not have transfer proteins, but in our system, we have transfer proteins. If you look at the way, so these are the putative ISC pathway proteins identified in the nuclear genome. All the mitochondrial proteins that function in the mitochondria are nuclear encoded and would be, should be imported into the mitochondrion. And if you look at Zhang's work, on John Adams' work on identifying essential indispensable p-falciparum by piggyback mutagenesis, and the clues we get from there are that ISCS and ISD11 and ISCU1 and ISCA1 and these proteins would be essential, but there are two which might be dispensable, and so therefore it's important for us to understand their functions and what they do in the ISC pathway. And as I said earlier, that the ISC mediated iron sulfur cluster assembly contributes to the essentiality of mitochondria for life, and even in very reduced mitochondria, or what you call mitosome or hydrogenosomes, it's seen that the ISC pathway is conserved, that this protein still come to that organelle and ISC pathway operates, telling us that it's very essential for survival and for important life functions. So we express all these proteins in a recombinant way, and we started to build the entire ISC complex in vitro to understand both functionalities of assembly as well as to see if we can solve structures of the complex. So we could say that from our work that ISCS is a cysteine desulfurase whose activity is enhanced manyfold by interaction with ISD11, which is not a cysteine desulfurase but is a protein that interacts with ISCS and enhances its activity multifold. We could see the interactions both in vitro and also in pull-down experiments from the parasite lysate, so they do interact in vivo. So ISCS and ISD11 had both been localized earlier, but the other proteins had not been localized with the mitochondria. So we made antibodies to express them, made antibodies against them, and did localization studies. And this showed us very, very clearly that all these proteins are targeted to the parasite mitochondria. As you can see here, absolute overlaps were seen with mitotracker red and the antibodies generated against these proteins. Now, when we express and purified each of these, and we wanted to build this entire complex. Now, we understand that we know what the cysteine desulfurase complex is, it's this ISCS and ISD11. We also had a putative scaffold, which is ISCU, but we also wanted to test whether these other proteins could function as a scaffold or do not function as a scaffold. So we made different combinations of these and started testing for iron sulfur cluster formation. To cut a long story short, the only combination that formed an iron sulfur cluster in vitro was the ISCS ISD11 ISCU combination with the iron sulfur cluster assembling on ISCU, and all other combinations were non-functional, no iron sulfur clusters were formed. What is interesting here is that the cluster we saw forming was not a 2 iron 2 sulfur cluster. Now in other systems, the other ISC systems, both in bacteria and in the ISC of eukaryotic mitochondria, it's been shown that the first cluster to form in the scaffold is actually 2 Fe2S, and then two such clusters, 2 Fe2S, two clusters are further reduced to give rise to a single 4 Fe4S cluster. But in our case, we were not seeing that, because if that had been happening, we would have seen an additional peak coming over here. But we could not see that. We just kept getting 4 Fe4S clusters. So what it seemed to us that it's not that we are getting a formation of 2 Fe2S clusters. First, we have a direct formation of 4 Fe4S onto the ISCU. So we wanted to test this and make sure that that was happening. So we exposed this to oxygen. And if you oxidize this cluster, it should actually split into 2 Fe2S again, and we should be able to see this additional peak. But that did not happen. When we exposed it to oxygen, we either saw a 4 Fe4S peak or we saw an absence of peak. We didn't see an intermediate 2 Fe2S peak. And similarly, if we took ISCU alone, which is a scaffold protein on which this cluster assembled, again exposed to oxygen, we either see a 4 Fe4S peak, the 4 Fe4S peaks keep reducing in its intensity, but you don't see an additional peak for 2 Fe2S, telling us that there was a direct assembly of 4 iron 4 sulfur clusters, which is different from all other examples known in nature so far. So in Plasmodium, that was a unique feature. So we wanted to see what the complex was like now. and we could use our antibodies to both do pull down experiments as well as do in vivo cross-linking experiments. And when we did in vivo, so we could pull down all these complexes with each other. So we knew that this was forming a big complex and they were all interacting. And when we did DSP in vivo cross-linking using DSP, we found that the expected size, the size we were getting for the complex was double nearly the size we expected to get if we would follow the conventional understanding of the ISC complex, which is that two ISCS molecules interact, and then an individual ISD11 interacts, and then two ISC molecule interacts as a heterohexamer complex. But what we were getting seemed to be double the size of the heterohexamer. So we started looking at structures of these using SAXS, scattering, excess scattering studies. And using SAXS, we found that the ISCU alone, if you just take the scaffold protein alone, it forms a trimer and not the conventional dimer or tetramer. And from small angle excess scattering, we could understand that this was a primary complex of this nature with the putative 4-iron 4-sulfur cluster binding site, as I show over here, which can take a 4-iron 4-sulfur cuboidal structure here. All the 4Fe4S binding sites were coming towards the trimer vertex. So this is the trimer vertex, and all the 4Fe4S sites were oriented in this manner. But the question to ask was, is this ISU assembling a single 4Fe4S cluster, or is it assembling 2 or more clusters? So we actually did measurements of the number of iron atoms and sulfur atoms in this complex, and we found that there were only 4 of each, which tells us that this trimer has only one 4Fe4S cluster. Either it would be formed between these two, or either of these alone could carry the 4Fe4S cluster, and we don't understand yet what happens. But essentially, the trimer has only one cluster. Now, what is the structure like of the entire complex? So when we did, again, small angle X-ray scattering of the entire complex together, we found this. So this is just the ISCU and the ISCS proteins without the ISD elements. You're just taking these together here. And you see, this is one half of the complex. And as we suspected, this was actually dimerizing to another level. So you have the whole complex and it further dimerizes again. This is not known earlier in any of the structures solved for ISC complexes in mitochondria. And this is the kind of complex we were getting. So if you look at the interaction interface of the dimer of this large complex, you find that the interaction interface is actually a section of ISCS, which is not conserved in other ISCS proteins in other organisms. It's a large N-terminal extension. These amino acids are not conserved in other ISCS proteins, and they form either hydrophobic or electrostatic interactions. And if you make a deletion of this region, the protein becomes extremely unstable and the complex does not form, telling us that this large extension, which is a low complexity region, actually might have a role to play in higher order assembly of these proteins and in formation of this kind of unique complex. When you put ISD11 in this, you see a similar complex. Now you have this heterohexamer and a dimer of the heterohexamer, and this V-shaped structure now becomes more tightly packed. So these two arms of the V come closer together, and this is the final complex shape that's taking place. So we did this in collaboration with Afsar, who is a student of Dr. Ravi Shankar in my department, who's a structural biologist. So now we know what the scaffold is like. We also understand that the structure is different from known ISCS structure, from known scaffold structures of the ISC pathway. But then what are our transfer proteins? ISCU assembles the cluster. Can ISCU directly transfer the cluster onto a target apoprotein, or does it require intermediary proteins to transfer the cluster, as is known in other systems? In our case, we tried all these, right? So we had the scaffold assembled, and we tried to transfer this thing onto a target apoprotein. In our case, it's aconitis B, which is a mitochondrial protein. So we use aconitis B as a recipient of the iron sulfur cluster and tested whether each of these proteins could serve as carrier proteins. Interestingly, ISCU not only assembled the cluster, but also could directly transfer the cluster in the absence of a transfer protein, to transfer the cluster directly onto aconitis B. And NIFU-like protein could not transfer the cluster, ISCA1 could not transfer the cluster, ISCA2 could transfer the cluster, but it was less efficient than NIFU itself. But it was a carrier protein. So both these are, so ISCU is not only a scaffold, but also can directly transfer clusters onto a recipient. PFISCA2 is not a scaffold protein, but is a carrier protein and can interact with the scaffold and transfer the cluster onto a target apoprotein. In our case, it's aconitis B. And we did not only see the transfers and saw assembly of iron sulfur clusters, but we also measured activation of aconitis B as a result of the formation of the cluster complex, as confirming our results further. So basically, we worked out this, and it's been published this year in Mol Micro, telling us that these are the differences that exist. You have a heterohexamer normally in other mitochondria or bacterial systems, but in Plasmodium, you have a further higher order assembly where you have a dimer of the heterohexamer, you have a much larger complex. You have this essential ISU as a scaffold, which can not only assemble a 4-iron 4-sulfur cluster directly, which is also a unique thing without 2Fe2S intermediates, but also transfer this cluster onto recipient proteins. There might be other mechanisms of transfer. So it's possible that ISU transfers directly, but it's possible also that you have an involvement of chaperones and a glutatidoxin-like protein, which is present in the Plasmodium falciparum system. You can also have ISCA2 here, perhaps, transferring this onto a transporter, which can take this down to the CIA machinery into the cytosol. And currently my lab is looking at this transporter protein to understand whether it serves as an iron sulfur cluster transporter for the mitochondria into the cytosol. You can also have NICFU-like protein that we showed was a possible transfer protein, because there might be specificity in the proteins that transfer these FES clusters. So it's possible that for some proteins, ISCU directly transfers the cluster. It's also possible that many proteins such as fumarate hydratase or succinate dehydrogenase, because we haven't tested them, do not take the cluster directly from ISCU, and that other carrier proteins would have substrate specificity, and this mechanism might be operated. We still don't know that. But we know that these can actually serve as transfer proteins. So this is what we have. Now, so I mentioned many proteins like, fumarate hydratase, succinate dehydrogenase, the re-escape protein, ferredoxin, reductase, etc. But what is interesting is that in the mitochondria, our work has recently shown that you have we have another protein called a mitochondrial endo-3, which is a DNA glycosylase, which also has an iron sulfur cluster. And in the epicoplast, you have an exonuclease with very novel properties, which also has a 4-iron 4-sulfur cluster, which would be incorporated onto the protein by the subpathway of the epicoplast. And I will talk a little bit about DNA repair to give you a glimpse of what we have. So I'll quickly move on. So DNA repair in organelles has not been very well studied in the malaria parasite. As far as we understand, we have the base accession repair mechanism would be the major mechanism, but recent work from Mrinal Bhattacharya's group has shown that double-stranded break repair also operates in the mitochondria, although BER can be the major mechanism. And because the epicoplast has lost photosynthesis, it has resulted in a reducing environment rather than an oxidizing environment. And we need to understand whether that has caused a difference in or alter the DNA repair machinery present in the organelle. And if you look at accelerated resistance to multiple drug strains, what are called ARMD strains, it's when people look, so this is a report that came out in 2016. And when these were sequenced, it was found that several of the DNA repair proteins, both in the nucleus and putative proteins of the organelles, were mutated in these. So these also have a role to play in resistance to frontline antimalarials. In this particular paper, it was artemisinin. And we know that epicoplast is a very unique DNA polymerase with multiple domains and multiple functions. So how does that play out in the repair pathway is also a question that needs to be asked. So if you look at the base accession repair pathway very quickly, we basically have an oxidative lesion over here, which will be recognized by a monofunctional or a bifunctional glycosylase, a DNA glycosylase, which would remove the base and give you this kind of substrate, which would then be cleaved by what are called apurinic or apyrimidinic endonucleases, AP endonucleases, which cleave the DNA. And if they cleave the DNA over here, they give rise to what is called a 3' hydroxyl group and a DRP group, or a ribosyl phosphate group. And if this is recognized by a polymerase, by polymerase beta lyase activity, the DRP group would be removed, and a polymerase can extend this, and then a ligase can cleave it, can join it. But if this does not happen, that is, a polymerase is not able to cleave the DRP, then the polymerase can only extend this further, and this kind of flap will be generated. And this flap will then have to be cleaved by what is called a flap endonuclease. And once this cleavage happens, they would be joining by a DNA ligase. So this is the short patch BER pathway, and this is the long patch BER pathway. The long patch BER pathway requires the additional function of a flap endonuclease. So we worked on several enzymes. I'm going to show you some work on AP endonucleases, which is interesting. A flash of the monofunctional glycosylase, because my student is presenting in the evening today, and so you'll see her work on that. And a little bit on the flap endo, which is also a presentation by a student. So I'll just sort of go through this to give you the gist of what we understand from there. Now if you look at the parasite genome, there are only 2 AP endonucleases encoded in the genome. Only 2 AP endonucleases, and both these have organelle targeting signals. From our early prediction, we thought that APN1, which is the first AP endonucleases, which is a homologue of the E. coli NFO and does not have a homologue in the humans, seemed to us to be an epicoplast protein based on targeting because it seemed to have a signal and a transit peptide. But when we expressed the protein and made antibodies and tried localization experiments, both by looking at nuclear cytoplasmic sections in terms of cellular fractionation as well as by confocal microscopy, it was very clear that this is not an epicoplast protein, it is actually a mitochondrial protein. And when you do chromatin immunoprecipitation using these antibodies that we made, again, it was only a mitochondrial protein. It was not a plastid or a nuclear protein. So this was exclusively a mitochondrial protein, and this was different from what we had thought it would turn out to be. So APN1 turns out to be a mitochondrial APN2 nuclease. Interestingly, also, from other studies done on other systems, primarily, in bacterial APN2 nucleases, it's been seen that the structure also has been solved of bacterial APN2 nucleases, and it seems to bind zinc. But when we did activity assays with zinc, we found that there was an initial increase in activity, but then an inhibition of activity. And when we use manganese, on the other hand, you get this nice enhancement of activity. When you use manganese and zinc together in different proportions, I'm just cutting short. Basically, when you use both of them together, you see maximal activity. So this protein has 3 metal binding sites, and it seemed to us by from this series of experiments that it requires the binding not only of zinc but also has to have manganese binding. Otherwise, zinc would inhibit function of this protein. So we did a lot of molecular dynamic simulation studies with my colleague in Iran, and also CD spectro to understand how the protein was folding. And from this, we could understand that there is a possibility that this histidine 542 can actually be a manganese binding site, while you have 2 other zinc binding sites over here. And if you look at the way the protein folds, because if you have only zinc being provided, the protein is non-functional, and the way protein folds. So if you completely deionize the protein, you remove the metal ions. This has a very, this is completely deino metal ions. This is the kind of structure you get. You start putting in metal ions. You put only manganese, it kindly partially folds. If you put only zinc, it is really strangely behaved. But if you put both of them together, it folds into this very nice structure, and this is what is the active protein. So then we have wanted to confirm whether this was actually the case, that this protein requires manganese in addition to these zinc ions. And yes, you make a mutant. And when you make this mutant protein where we predicted zinc would bind, the protein loses activity. It loses activity, and we could show that this was because there was no zinc binding. So if you actually measure the levels of manganese and zinc binding on this protein, on this mutant, so this is a wild type has 2 zinc and 1 manganese, but in the mutant, you have only manganese and no zinc binding. And this mutant also loses the capacity to bind DNA. So our work showed that you had these 3 metal binding sites, of which 2 were zinc binding sites and 1 was manganese. We knew exactly where manganese bound, and this was essential. We don't know why this happens, and the protein folds completely differently when you remove the manganese and have only zinc. So and it also alters the way DNA binding takes place. Now, APN2 nucleases not only cleave DNA, in our case, a tetrahydrofuran thing of a modified base, but they also shown some of them to have additional activities, like they can function as 3' phosphatases. They can also function as exonucleases. Some of them can also function in nucleotide incision repair. I'll come in a minute to that. So this protein could function as a 3' phosphatase. It could cleave this phosphate at these positions, and therefore had these additional activities. The other protein, but it could not function as an NIR or as an exonuclease, so it had very limited function in these aspects. The other protein, which my student will talk about, had all these activities. So the other protein, APE1, had all these activities. And if you actually make a mutant of APE, you find that the parasite now grow very slowly. I'll just take a minute to finish, if that's okay. They actually grow very slowly, and you see very slow growth, and the prepatent period is elongated. So the prepatent period is actually elongated, and the conversion from the liver stage to the blood stage happens very slowly, which tells us that the APE protein is probably a very important protein, and the APN can probably play a backup. I will skip all this, and I'll skip also because my student Triveni has a presentation, but only to tell you that we have this other protein which has a very interesting insertion, totally unconserved, very different in Falciparum and Burghi. Burghi lacks the insertion, and because of the lack of insertion has very different activities. There is complete change in activity. And so therefore, what we are proposing by this analysis of DNA repair proteins is that 2 things. One is that from this ISC thing, the LCR seem to have an important function in protein assembly because you have this large complex being formed. From our work on the exonuclease, that the LCRs also seem to have a role in protein functionality. And Triveni's presentation will describe that. If you remove the LCR, you use a series of functionalities. So they seem to have a role in enzymic activity and DNA binding. So this is very interesting in terms of Plasmodium falciparum, because still we are yet to understand what these LCRs are doing apart from immune evasions. So I'd like to end here. We're glad to take questions. This work was done by Sadik, who did all the ISC work, Anupama, who did initial work on the replication pathways, Neetu, who worked on APE, and Triveni, who's done on the exonuclease. Both these students at the bottom are presenting today, so I'm not elaborating on their work, and my collaborators from CDRI and Stuart, who worked on the OGG localizations. Thank you. I'll be happy to take questions if there are any questions.
Yeah. Thank you, Saman. Very nice talk, especially the work on in vitro activity assays developed for iron sulfur biogenesis complexes and all. Let me just check if there is a question in the chat. If not, just I will start. Among these proteins, which of these you think are good targets in terms of their similarity or differences with the human homologs?
So if you look, so in terms of the iron sulfur cluster assembly, I think the SUF pathway stands out because the SUF pathway is in its entirety absent in the human list. Yes. And so clearly anything, and we by knockout studies both from our group and other groups in the world, now and also now in toxoplasma, as I showed you, they're essential. So you do a knockout of any of the SUF genes, and not only in the blood stages, but as our work has shown, also in the mosquito stages, there is no sporozoid formation. So it's very clear that this is a very good, but unfortunately, because the SUF pathway has not been worked out in great detail in bacteria, because in bacteria it is the backup pathway. It's activated in ISC is the main pathway. It's activated only in iron starvation oxidative stress. So really people have not looked at inhibitors for bacteria, and we really don't have any clues. We don't have starting points. Okay, so now we have to actually start from scratch and start identifying inhibitors. In principle, by doing the PLP adduct experiment, we established that you can actually inhibit SUF-S and cause parasite death by using D-cycloserine, which is a PLP adduct forming compound. And it forms an adduct with PLP, which is a cofactor for SUF-S. And by doing that, you inhibit enzyme activity and also inhibit cause parasite death. So we have a proof of principle, but we don't have very specific, high activity inhibitors.
Okay, Dr. Malhotra has a question.
Saman, very nice talk.
Yes. Yes, ma'am.
You know what are the DNA repair enzyme which are activated or down-regulated when we add artemisinin or chloroquine?
Down-regulated at?
Artemisinin and chloroquine. When
Okay. So in the ARMD strains, in the associated resistance to multiple drug strains, there's an APE mutant. So a mutant of the AP endonuclease that I talked about, which is the main AP endonuclease. There's also an APN one. This, and there's another protein called OGG1, which I didn't talk about, which from our work and with Stuart we have done localization. We show that it goes to the epicoplast. It's an epicoplast DNA glycosylase probably. That also. So, and that's also upregulated in oxidative stress. So this seems to be an important protein. These three mutants have been identified in ARMD strains.
Okay. Any other question? Okay, I don't see anyone in the chat. So in that case, thank you very much, Saman.
Thank you.
It was very nice having you at the conference. Okay, so next speaker is Professor Suman Bhatt. Suman is a senior professor in Jawaharlal Nehru University, and his group works on DNA replication and cell division in malaria parasite, and all the machinery involved in DNA replication, including those in the organelle. And in the same line, today he will talk about a unique acetyltransferase, which is important for parasites. Over to you, Suman.
Yeah, can you see the slides?
Yes, we can see your slide.
Okay. Thank you, Asif, Pavan, for the invitation, and all other organizers. It's really nice to be here and to see you. Hello? I did.
Well, is it more important to see a slide or him? Don't bother him.
Okay.
Okay, please.
Okay, maybe next 25 minutes you may not be able to see me. So thank you again. And today I will be talking on GCN5 acetyl transferase, a HAT protein in Plasmodium falciparum, and its unique processing pathway. Now, our lab interest is we are working on DNA replication and cell cycle regulation in the parasite. And this slide, I don't need to elaborate, except that there are at least 4 different stages in the parasite life cycle where multiple rounds of DNA replication takes place. And our interest is to find out the key regulators of replication process so that we can have a better understanding of parasite proliferation, which may lead to intervention processes. Now, as you know that the parasite is growing inside the RBC, which is a definite volume. Now, the key difference of Plasmodium parasite with respect to other eukaryotes is that replication always followed by cell division. However, in the parasites, the nucleus keep on dividing, and also the organelles, they also grow and divide until a certain stage, because you cannot keep on doing this because everything is happening within this definite volume of the RBC. So at that stage, so therefore, there has to be a signal which turns it on, and there has to be a signal which turns it off. So this is the parasite with respect to replication. We also have an interest in apicoplast, but right now we are focusing more on the nuclear division and the key factors which are regulating this process. Now, if you look at yeast, what happens? Hello? Can you hear me? Yeah. So in yeast, 6-protein complex that binds to ARS consensus sequence, which leads to loading of the helicases along with CDC6, and the DNA is unwound, and then from the initiation it goes to the elongation where you have pCNA, proliferating cell nuclear antigen, as well as polymerase, which takes it over for the elongation. So therefore, even this process, we think that the chromatin modification, which is very important for transcription process, is also important for replication, because in both cases, the chromatin, which was in the tight bound form, has to be unwound so that replication as well as transcription can take place. Sometimes transcription helps, this unwinding leads to replication, but sometimes it could be only for replication, and I will explain it that to you. Now, over the years, this is for the replication part. What we have shown is that we believe that ORC1 is the major protein in Plasmodium, which sets the platform for replication, and this replication foci are formed in the parasite. The parasite keeps on replicating until a stage where enough DNA replication has taken place. Then a kinase, PFPK5, it's a CDK-like kinase, it phosphorylates ORC1, and once it phosphorylates ORC1, ORC1 comes to the cytoplasm and it gets degraded, and this cycle go on. So we have characterized all these factors, and this is kind of eukaryotic system. The ring stage is kind of G1, and then the trough stage is the S phase, and then followed by the Sison stage for the division. Now, while we were working on this, we also were amazed that many of the proteins, they have a very long N terminal, like ORC1, ORC2, ORC5, as well as GCN5, which is the main thrust of today's talk. So if you look at in other human or yeast, they have a shorter version. However, the plasmodium proteins are really huge, and many a time the conserved domains are at the C terminus, whereas the N terminus has lots of repeats. So those can be used either for some other functions, or there are signal sequences present which take it to the organelle of their interest or function. One such protein, ORC2, also has a long N-terminus extension, and we found if we have an antibody of N2 and do a Western blot, you see the full-length protein. But if you take the antibody against C-terminus, there are two bands. One is the full-length, and the other one is the short form, and that is the C-terminus of ORC2, which is actually conserved among all species. So what we showed later, that ORC2 actually gets processed in the vicinity of the ER, because if you used the signal peptide peptidase and then you can block this processing, and ORC2 then doesn't go to the nucleus. So therefore, all these N-terminus, the various proteins which has a long N-terminus, and I have found this for many replication proteins, they have this unique processing pathway that it is using different organelles for getting processed, because as you know, normally in other eukaryotes, what happens, they are translated in the cytoplasm, or the co-translation and migration through the ER takes place. But normally, the proteins which are destined to the nucleus, they have the nuclear localization signal, and then it takes to the nucleus. Here is one example where a protein has to be associated with ER and then get processed, and then it goes to the nucleus. Now, the protein of today's interest is this HAT protein, GCN5. And as you know that parasite life cycle progression occurs through coordinated waves of gene expression. Our interest started with this protein because we wanted to see which HATs are involved in chromatin modification that will lead to replication, unwinding of chromatin, and will lead to the replication process. And unlike the majority of higher eukaryotes, falciparum chromatin is predominantly in a euchromatic state, and with only few heterochromatic island, they are marked by trimethylation of lysine 9 of histone H3. You see H3K9 acetylation as well as H3K14 acetylation, and that modulate the gene expression, and this is mostly done by GCN5. Histone H4 lysine acetylation also have been reported, and that is done by PF-MIST, another acetyl transferase. And as I have mentioned, that chromatin modification is also very much important for DNA replication. So what happens? Chromatin, this is in tight bound stage, and then you have the interplay of the HAT and HDAC. So HAT will acetylate them in the lysine residue, which will unwound, and HDACs in a very simple way, it will remove that, so it can, from the wound state, it goes to the unwound state. And that actually lead to various cell cycle functions, as has been mentioned here. Now, GCN5. GCN5 is a very long protein in Plasmodium, almost 1500 residues. However, the HAT domain is only at the C terminus. It's not known what it is doing, the long N terminus sequence, which is mostly around 1000 bases, 1000 amino acid. So what is known for GCN5, it interacts with Plasmodium ADA2 protein as well as, and it forms the chromatin remodeling factor, And also, it has the conserved hat activity with the substrate preference for H3, K9, and K40. Very brief note on MIST, because I'll come back to that. MIST is also a hat protein, and this is also around 600 amino acid long. And again, you can see that the head domain is at the C terminus with a long N-terminal extension. And it acetylates histone H4 at K5, K8, K12, and K16. An overexpression of MIST is deleterious for cell cycle progression, and this also affects nuclear division and cytokinesis. Now, first, to raise the antibody against GCN5, we expressed and purified the C-terminal domain, which has the head domain, and purified and raised the antibody. Now, when we did Western blot against the parasite lysate, we got this so many bands, which you doubt that if you are getting these many bands, then that along with the full length, which is around 170K. So either these are the breakdown products or these are some non-specific bands. Well, if you do immunoprecipitation, you can see here, again you see these bands, specifically they are with the immune sera, but not with the pre-immune sera, suggesting that they are specific. We did LCMS of the immune sample, and the major protein was the GCN5 as well as ADA2, which has been earlier shown, suggesting that the immunoprecipitation which we have done is authentic because the antibodies against which they have been raised, that's the maximum number of peptides are coming, along with the other proteins which have been reported as an interacting partner. There are other interacting partners, but I won't get time today to get into that. So, if that is true, are these bands, all these bands which we are seeing here, they are present in all stages? Yes, they are present. However, the protein is mostly a punctate status they give in the nucleus. And the protein by subcellular fractionation is only in the nucleus, all these bands, cytoplasmic, we don't see anything, suggesting that it's a nuclear protein. Now, if there are so many bands, so either there could be translated products from different splice variants, because there are 3 introns in the GCN5, or it could be the outcome of proteolytically processed protein products. These are either non-specific bands, that is also possible because these are polyclonal antibody, so you can get these non-specific bands, and or they are degraded from the full-length protein. So what we did, then we used different protease inhibitors. So this is DMSO, this is peptidine A, aspartyl protease, and this is cysteine protease. And we also used NG132, which has the proteasomal degradation pathway inhibitor, but it has been also shown that it also has cysteine protease activity, emitter activity. We used epoxomycin also because that's more specific for proteasome. Now, if you look at peptidine A, all these bands are present, but as soon as we put cysteine protease, now all the bands which are there, now they're almost gone, and you are getting the full-length protein. Same is with MG132, which is also cysteine protease, but not with aspartyl protease or epoxomycin, which is the drug for proteasome inhibitor. So therefore, GCN5 is proteolytically processed by a cysteine protease-like enzyme. If that is true, if we use the drug and try to co-localize with other proteins, we found that as soon as you use NG132, now the protein is localized in the food backbone. As you can see here, this is E64D, this is NG132. Now the protein is in the, and we can also co-localize that with ATG18, which is a vacuole, food vacuole marker. So therefore, the protein possibly has to go to the food backbone to get processed, and from there it goes to the nucleus. This was also shown by EM, as you can see here. Now, mostly in the nucleus, in the untreated, but when you use the drug, you see a lot of proteins, these dots in the food backbone. Now, the full-length protein, it traffics to the food vacuole via ER-Golgi secretory pathway. So if you use Befeldin A, you can also see this full-length protein. So therefore, the protein, and we can also co-localize that with BIP, which is a ER protein. We also did the chromatin immunoprecipitation to see where are all these GCN5 are binding. And most of the, more than 50% of sites were at the transcriptional site, which makes sense because that will allow the chromatin to be annulled and the transcriptional machinery to come. If that is true, then if we inhibit now, there should be two consequences. One, the H3K9 AC should go down, and if H3K9 AC goes down, then all those sites where, particularly the transcriptional start site where GCN5 was binding, now those transcript also, at those sites also they should come down, and that's what happened. As you can see here, the acetylation level went down when we used the drug, and which was concomitant with going down or reducing the GCN5 at those promoter sites. So what is happening is that the GCN5 goes through the ER, and then it goes to the vicinity of the food vacuole, and this here it gets processed, and after getting processed it goes to the nucleus. So this is kind of a bizarre that a protein to go to the nucleus, it has to take this pathway. But more and more we are getting evidence that many of these proteins, plasmodium proteins, indeed take these pathways or different organelle to get it to its destination. We have also got inhibitors for GCN5. These are the known inhibitors, but mostly they are at the micromolar levels. So using the comparative sequence analysis and homology modeling and looking at the residues where they are different from the host, we have, after docking, got several inhibitors, and one of them actually gave us IC50 value at below micromolar level, and that has no effect up to 20 micromolar for mammalian cells. And also, if you look at this inhibitor led to the H3K9 AC level to go down, as you can see here, which is actually better than embelin. To get that, embelin is a known inhibitor of GCN5. You have to use 25 micromolar of embelin. So now, just next 2 minutes, what I will show you that what is the role of acetyl transferases in DNA replication. Apart from chromatin modification, these proteins, these hats, also they modulate replication proteins. Like MCM2 and ORC1, they interact with the histone acetyl transferase HPO1, as well as acetylation of GCN5 regulates CDC6 phosphorylation, which loads the MCM2. So therefore, this regulation through hats that happens at different levels and the now, if that is true, then where are the replication origins in parasites? We have shown that by using nascent strand abundance assay and also bioinformatically, there are ARS-like sequences. And nascent strand abundance, the nascent strands which are predominant at the replication for, they are more at the ARS sites and not at the non-ARS sites. And ORC1 is a protein which binds to the replication origin, they are also enriched at the ARS as well as the nascent strand abundance assay shows that the enrichment of ORC1 and the replication are more at the ARS sites but not at the non-ARS sites. So if that is true, then which of these hats, either we have talked about GCN5, now I've also mentioned about another hat that is MIST. Now if you look at MIST, again, as you see here, that there are two bands, full length, and then there is a shorter version. And this is the shorter version which has the hat domain. And if you do a subcellular fractionation, the shorter version is in the nucleus, however, the full length protein is in the cytoplasm. Now this is also very, very interesting, but this is not due to the proteolytic processing which we have shown. There is an internal ribosomal entry site which leads to this, and that today I won't be able to discuss about it. However, after this, we looked at the occupancy of GCN5 and MIST, those sites where replication initiation has taken place because of abundance of the nascent strand as well as the ARS-like sequences. And as you can see here, we see that mostly the binding of MIST is much more at those sites than the GCN5, and this is the controlled non-ARS. So therefore, although we are seeing GCN5 as those ARS-like sequences, we believe at this point that probably for replication, MIST will be the guiding factor for chromatin unwinding, and GCN5 could be more responsible for overall transcriptional regulation, although we need to have more data on it. Now, in conclusion, we have shown that GCN5 follows a novel proteolytic processing pathway, which is very critical for its activity in vivo. GCN5 interacts with several proteins that are associated with diverse biological functions. We have also identified the genome-wide binding sites of GCN5, and we have shown that the protein is majorly enriched at the promoter regions, though through comparative modeling, we have identified different residues which are different from the host at the active site, and that has led us to designing potential inhibitor GCN5. And finally, ARS-like sequences are active in Plasmodium falciparum, and although we see both enrichment of GCN5 and MIST, MIST is much more relevant, looks like, for replication. And I must acknowledge all the students. This is an old slide, lab has changed, and all my collaborators and funding agencies. The GCN5 related work is mostly done by Krishanu. He is now doing postdoc, and also Ankita. Ankita has submitted her thesis. And the MIST work is done by Shashank, and the new students Poonam and Reena, now they are taking over the GCN5 work. And thank you very much for your attention. If you have any question, I'll be happy to answer.
Thank you, Suman. It was a very nice talk.
Thank you.
So before anybody asks us, so I understand that the processing of this GCN5 is critical for its transport and then for its function. So which protease you have predicted, if you have identified or trying to identify, involved in this processing?
Yeah, it is a cysteine protease, and as you know, food backwell has at least 1, 2, and 3. So we don't know at this point because it's difficult to express those proteins and do one by one. But as of now, we know it's the cysteine protease, and if we get the knockout, that would be interesting to see that which one is involved specifically for processing of GCN5.
One student, Shilpi Jain from ICGT, she's asking, did you check if it is also get alternatively spliced at the blood stages, or is it only the processing which is involved?
Yeah, so the introns are very, very small, and then very clearly it shows when you use the, the cysteine protease inhibitor, the full length protein we are getting. So it is, and this happens like, as I have told you, that for MIST, there is internal ribosomal entry site that leads to a shorter version of the protein. But for GCN5, we believe it is proteolytic cleavage.
So next question is from Christian. He has raised the hand. Christian?
Yeah, thank you, Asif. Very nice, Suman, very nice, very interesting. I have a question about. Yeah, that I enjoyed. So I have a question about, you mentioned that the second protein that you studied doesn't have a processing with a protease, but has an internal entry site for ribosomes. Yeah. Yeah. Okay. Yeah. So I can understand why, in the regulatory scheme of things, for the GCN5, you need to cleave it to then release the active component. But I have more. Can you speculate why? What would be the function of the long version and the short version of this other protein? Because clearly, if you have internal entry sites, you probably need both versions for some kind of activity. What do you think?
Yes, very interesting and good question. So, as I have told you, the histone acetyl transferases, they not only acetylate histone, there are a lot of non-histone substrates also. So what we believe, and our fractionation data is very clear, that the shorter version is very specifically, it is going to the nucleus. Yeah. However, the long version, it is in the cytoplasm. So we believe that the cytoplasmic, those will be acetyl transferase function for some cytoplasmic protein. And we are trying to get, the cytoplasmic fraction and the nuclear fraction pull down, and then trying to do we have specific antibodies also to look at the substrates. And that will tell us that compartmentalization of these proteins is very unique, like the way one part is going to the nucleus, the other part stays in the cytoplasm.
So I think there are... So do you think in this case that the internal extension that is present in the cytoplasmic version but not in the nucleus, do you think, could you test if it acts as a docking site for substrate in the cytoplasm or something like that, or?
Yeah, possible. Possible as a docking site for the cytoplasmic protein that may lead to the acetylation of the protein or some moonlighting function because there could be some other functions.
Yeah, absolutely.
I don't know why these parasites, they won't waste so much of energy to express a region which is not required.
And no, of course. Yeah. No, it will have a function, I'm pretty sure.
Yeah. Right.
Very interesting. Thank you, Subhan. Yeah.
Thank you.
Enjoy it. It's lovely to see you too.
Yes, I'm here. Thank you. Thank you.
Okay, our next speaker is Dr. Pushkar Sharma. Pushkar, are you here? Yes. Pushkar is a senior scientist and director at National Institute of Immunology. And he's a close friend. And his lab is working on kinases in Plasmodium for last many years, and today he will present a novel signaling pathway for regulation of phospholipid biosynthesis. Pushkar, over to you.
Thank you very much, Asif. Can you see my first slide?
I can see you also.
And although you see the content every day to work, right, on the slide. So.
Can you put on display, please?
Hello, hi Asif.
Yeah, can you put on full screen your slides?
I think I have put it on full screen. Let me just do it again. Is it? Does it show as full screen?
Not yet.
No.
Can you try?
Pushkar.
Yes.
I also had the same problem.
have to come out and come in then, right?
Yeah. Or you can actually, go to the left. Yeah, please try.
Is it better?
No.
So how, from my side, it is so full screen.
The slide and we see that if they are moving or not.
Are they moving?
No.
Okay.
No.
Moving. So, let me just try again. Suman, you have stopped sharing?
Yeah, I think stop.
Yes. Shared, only thing they are not moving or not showing the full screen.
Okay. Let me try yet another slide. Is it full screen now?
No.
Okay, and then?
Do I log in and log out and log in again?
No, can you go to the top of your screen? You have view options or display options, something where
I
you
have.
see full screen or duplicate maybe.
It says new share.
Pushkar, we can see your content, so perhaps you can just increase the size of your, a little bit, maybe up from 68 to 72 to something, a little less. And then you can just move in this way because this will take a long time to sort out.
ICGB wants a remote control of his screen. That is scary, man. I mean, yeah. There you go. With Pavan sitting there, I'm very skeptical. Yeah.
You can't give him remote control of.
Because then I will receive reagent requests for everything which is there on my slide, and I will not be able to match his. Yeah. So Asif, you will control me by remote now, right?
You haven't given the control.
Yeah, we still don't have the control of your slides.
So should I stop sharing my this thing? It says ICGB is controlling your screen. So I've stopped my share. I have stopped my share. Nice to see you, Suvira.
Hi.
Unusual circumstances, yeah.
So, Pushkar, when you actually share your slideshow, when you go to
I have come out of this thing because I have passed on the remote to you.
Yeah, so when you do the share screen, you select the individual screen that you want to share with us. And
Actually, I have a full screen which I cannot toggle. So apparently, I think it is with you.
Okay, let me again see if we can make you a co-host. Does it work for you now?
Yeah, try now again.
Let me just come out of the PowerPoint and then log in. Then that may work better. Let me just go again. Yeah, I will just say the share screen just once again.
You can do a shift F5 to make it to the
display
or a control L, whichever works in your system.
Yeah, one second. Too many instructions. 1 second. It'll get sorted out. Don't worry. Sorry about this, people, but I should have just come there. I mean, like I was planning to. I think it will be done. Now, let me just see.
No, you are the host, so I'm sure you can do it.
Ah, God. I just can't see the presentation. There you go. Is it visible?
It's coming.
Is it full screen now?
No, the internet is taking its time.
I have got it full screen. I'm sharing the screen.
It's showing a blank space right now to us.
Oh, I don't know what's going on. Should I log out and log in again? What do you suggest?
Yeah, I think please log out and log in. Maybe it will work.
Oh,
Pushkar, can you send your slides to ICGB by email and they can project from there? It'll be faster.
Yeah, I'll just do that.
Yeah, and they can open it there and just use next slide.
I will do both the things. I will log out and log in as well as I will share it with them.
With Asif.
Yeah. It's better, it'll be faster if I get there.
Yes. We can have tea together.
Asif, which email ID? Gmail?
Yeah. आपका दिनेश. Dinesh at ICGB.
Dinesh, I have Dinesh at icgb.res.in.
Yes. How big is the
I've sent it to both of you. Okay. And I'll come out and I'll stop share, I'll come out of the meeting and then join again. Joined again, Asif.
Welcome back. Welcome back.
Yeah. You're still ready to welcome me after all this, huh?
We have made you the co-host.
Let me just see. 1 second.
You have been allowed to make a share screen as well.
Do you see anything on the screens?
No, not yet.
Are you using two screens by any chance?
Sorry?
Are you using two displays?
No, just one display.
No, you can't see still.
But I've sent you the email with this thing. And your screen sharing is paused, just keep saying me. And it's not allowing me to resume.
Okay, let me stop your sharing and then restart it.
Yeah.
Just try again to share your screen.
Okay, 1 second. Is it?
No, not yet.
Oh God.
But yeah, now we can see.
Yeah, can you?
We can see your documents.
Can you see my screen?
Yeah, it has all your
Yes.
Is it moving?
Yes, now we see all your.
No. But not moving.
Now you can see your screen.
Is it moving?
No. It is also not.
No.
I think there's some problem, internet connectivity problem, because if I'm moving it, it's no problem here. So what do I do now? Hasn't the email reached you? Email?
Dinesh send your Gmail address. Can you send on that?
Yeah, first, Asif, I sent you on your Gmail address and Dinesh's this thing. ICGB, you with great pomp you said, oh, send it to ICGB. I knew that that doesn't work fast, but you still insisted. Let me just say stop streaming. आसिफ आ गया जीमेल अकाउंट में? आ गया?
Pushkar, can you open your presentation and press F5 to see if it goes on the full screen?
One second, I will do exactly what he said.
F5.
F5, right?
Yeah.
No, no, it has to be F7, right?
You're on a Windows or a Mac?
I'm on a Windows.
On a Windows?
Yeah.
F5 should work.
No, so it is the full. The point is the full screen is working here.
So I'm making Dr. Dinesh co-host. So should I remove Dr. Pushkar from the co-host?
अभी भी नहीं दिखाई दिया?
Pushkar, your slides are on the full screen mode. You can start your presentation.
Is it? Is it now me projecting or you projecting?
No, Dinesh is doing.
Okay, so now I'll stop sharing, right?
Yes.
Yeah, okay, fine. I can see them move.
Okay, good. You can start now.
Thank you very much, Asif. I think the first option would have been better, where I could have just come in. And my apologies to everyone. So I will try to save everyone's time by cutting my talk shorter. Once again, ICGB has been great in hosting these meetings, and I was just craving for one when Asif's invitation came, and of course I said yes. And my apologies to him because I delayed my abstract and title, but so on. But can you move to the next slide? I cannot move it, right? You will have to move, right, Dinesh? So Dinesh, it will be you who will be moving the slide, right?
Yes, Pushkar, Dinesh will move it. You can't move it.
Yeah, and I can't see the full slide. They're getting cut, but that's okay. So basically, I cannot see something is getting cut at the bottom, but that's okay. That has to be resolution of this thing. Let's not waste time on that. Yeah. So my lab works on Plasmodium blood stages typically, although we are getting into some gametocyte biology. But and then what we have been doing in recent times is trying to look at some of the pathways which may have similarity in Toxoplasma gondii, which is I would say reasonably closely related epicomplexin, but in a limited way. And an example of that we will see in a minute. It's an extremely good model for studying obligate intracellular parasitism, and is also very amenable to genetic manipulation. Can you go forward? Next slide, please. Yeah. So what we have been looking for almost 20 years now. 15 years now. We wanted to characterize, delineate, elucidate signaling pathways organized in the parasite biology, because that has been my interest, to dissect signaling pathways at the molecular and cellular level, and in Plasmodium, and also in some other organisms, but predominantly in Plasmodium, and now also to some extent in Toxoplasma. And these are just a brief summary of what we have done over the last few years, where we have actually looked at calcium-mediated signaling, which is also related to cyclic AMP-mediated signaling, and also a phospholipid phosphoinositide-mediated signaling, which basically is resulted in efforts from us and others have resulted in identification of some of these pathways, which are basically mediated by these second messengers by virtue of their ability to regulate their effector proteins such as protein kinases and so on and so forth, and they, as indicated here, regulate critical parasitic processes which are important for parasite development. Can you go forward? In the context of today's talk, I would talk about phosphoinositide mediated signaling in Plasmodium, which we have been interested for quite some, almost more than 10-12 years. And they started off by our interest in a phosphoinositide 3 kinase, which is the only phosphoinositide 3 kinase in Plasmodium. Just briefly, phosphoinositides are these phospholipids which have this inositol head group and which have 5 hydroxyls at position 2 to 6, and 3 of these at position 3, 4, and 5 can be phosphorylated by specific lipid kinases. And a combination of these events of phosphorylation can lead up to 7-second messenger phosphoinositides in yeast and mammals, and also in epicomplexans, although there's with subtle differences and so on. And the way these guys and the enzymes which are involved in this process are phospholipid kinases, and as I just mentioned, we worked on a PI3 kinase, and we are doing some stuff on others enzymes relevant in the regulation. Now, if you could go back, or even if you don't, what was indicated was the typical mechanism of action of these second messenger phospholipids or phosphoinositides. They look for proteins which have affinity towards them. Basically, these are small protein binding domains present in wide variety of proteins and which have affinity to specific phosphoinositide. And some of the very popular ones and the common ones are indicated here, such as the pH domain containing proteins or FIB proteins and so on and so forth. As you can see, they have affinity towards the specific phosphoinositides. So one of the ways they operate is they actually translocate the proteins which have this ability to bind to these phospholipids to different subcellular compartments where they're supposed to act functionally and so on and so forth. Can you go forward? So in our quest to identify the phospholipid signaling and phosphoinositide signaling, one of the obvious questions about for any protein or any enzyme or any second messenger is what are its targets in the parasite. So what we looked for was protein domains such as the pH, pX, and the FIV domain, and amongst others, which are present in proteins encoded in the Plasmodium genome. And 2 of the ones which are indicated here is what we call as a pH domain containing kinase, but now we call it CDPK7 because that's how the nomenclature goes, and a couple of others which have been indicated, which we have been working the last few years, indicated below also in red. But as shown here in circled is CDPK7, which is basically the central figure of today's story. And if you can go forward, and the reason for that is, so CDPKs, very briefly, as Christian gave a very elegant presentation on kinome of Plasmodium, just to highlight, the CDPKs are absent from the host, but they carry out critical parasitic functions. So for obvious reasons, traditionally they have been considered to be good drug targets and so on and so forth. And this thing, and they're conserved in epicomplexans like Plasmodium and Toxoplasma. Next slide will be an animation. Next slide, yeah. So shown on the right is the difference. Shown on the top is the canonical classical CDPKs with the calcium binding domain at the C terminus and the kinase domain at the N terminus. But the CDPK7 is kind of an outlier or a non-canonical or atypical kinase, because as you can see immediately, it has a very different domain architecture. It has a pH domain adjacent to the kinase domain, and then EFN motifs are at far away at the N terminus, and there has been no indicator that it can be regulated by calcium. The reason we got interested is because in this enzyme is because it has the pH domain, and the pH domain traditionally have been, as I said, have been the targets of phosphoinositides. Can you go forward? Yeah. So several years back, Praveen Kumar in my lab demonstrated that 4' phosphorylated phosphoinositides have the affinity towards the pH domain of CDPK7. Can you go next? And what it does is, not shown here, but this has been published for several years. I can't see it. I'm sure you can see the citation of the paper. Is that phosphoinositides bind to this and translocate this enzyme to cellular compartments, which are these typical punctate vesicular structures. And what we did was we actually generated a knockout line in collaboration with Christian Doric, which was, the transfected parasite we got in our lab, and then we did limit dilution cloning, and we got a population parasite which was pure and which has CDPK7 knockout. It was not essential, but these are very slow growing parasites as shown here. And it's important to state in Plasmodium burgii, it is essential. So it is clearly CDPK7 was critical for parasites growth. And one of the features, two features that I would like to highlight, there was a abrogation of ring to trophozoite transition, where, as you can see in the upper panel, the CDPK7, the 3D7, which is the normal parasite, and knockout, they're abnormal looking parasites which had these membranes which are all over the place, and it will become more evident in subsequent slides. So that was one place they were showing defect in parasite growth. The other one was the parasite division, as indicated by Merozoites per schizon, was significantly reduced in the case of knockout parasite. While we were working on it, we also noticed a ortholog at that point in time in Toxoplasma, and that was our venture into Toxoplasma, and we collaborated with Dominic Soldati to get the know-how of Toxoplasma knockouts and so on and so forth, and then brought it home from Geneva, and we knocked down because it's an essential kinase in Toxoplasma as well. And what Priyanka did was she was not able to knock it down, and using a tetracycline-based system where you add ATC is tetracycline. When you add ATC, it will deplete CDPK7. This is important for the reference in subsequent slides. And the number of parasites per vacuole was significantly reduced, suggesting that there was a defect in division. Basim had published this paper on Toxoplasma, and he had similar results, but we are not too bothered about it because we actually were interested in seeing how this enzyme in Plasmodium and Toxoplasma controls the growth of the parasite. And to answer this question, if we can go to the next slide. For any enzyme, as you figured out in the previous two plots, the answer to the question what it is doing lies where what is this substrate, which make it so relevant, and so on and so forth. So for a kinase, the relevant substrates are obviously the proteins it phosphorates. So we took a phosphoproteomics approach and collaborated with Keshav Prasad and Akhilesh Pandey at IOB, and then now in Apoya, and do quantitative phosphoproteomics. Just shown here is a protocol for that. Can you go forward? Can you first click? Yeah. So what we found was, this is the results for Toxoplasma, knockdown parasite, the differential phosphorylation or changes in the global phosphoproteome when Toxoplasma CDPK7 was knocked down. So we found several peptides which were differentially phosphorylated, and I cannot go into the details, but what we found was there were two striking features of these studies when we did gene ontology and other bioinformatic analysis. They belong to two major pathways, one in phospholipid biogenesis, another one in trafficking. Shown here is one of the important enzymes which forms this node in this protein interaction network, and will become relevant in subsequent slides. And lo and behold, in the case of next slide, in Plasmodium, what we found was when the TF-CDPK7 knockout also showed significant differences in proteins and critical proteins which were differentially phosphorylated or hypophosphorylated, and shown here, they turned out to be enzymes indicated in red, which have implication in phospholipid biogenesis, and so on and so forth. Of course, in this case also we found some trafficking proteins as altered as well. So what was clear was that this was the first indicator that maybe something going wrong with phospholipid biogenesis, coupled with the fact that in the case of at least Plasmodium found the membrane structures of some of these newly developed developing parasites were altered, suggested that there may be some issues related to membrane of phospholipid biogenesis. But what is something important to be noted by this slide is still on your screen, is that nature of the enzymes or the enzymes in the two parasites are very different. So even though it's phospholipid biogenesis, but there is hardly any commonality between targets between Plasmodium and Toxoplasma, which was interesting and becomes relevant as you go subsequently. Can you go forward? This is just a selected list of the enzymes which I just mentioned in phospholipid biometabolism and also vesicular trafficking. We did a fair bit of work on RAB11A, and I can tell you it's published in a paper earlier this year in PLOS Pathogens, where RAB11A is a critical mediator of CDPK7 signaling, as is important for the vesicular trafficking. But in the context of today's talk, I'm not going to talk about that work. If you're interested, you can go and read this published paper. Can you go forward? Yeah, first and foremost, we wanted to know what is going on with this and so on. We did some metabolic labeling assays for phospholipids, and which I'm going to show you in the next slide. And we had some results then lo and behold, ICGB comes into picture because I meet this guy called Cyril Bote, indicated, I hope it is visible on your slides, on the left, and at a meeting at ICGB 3 years back, and when he showed him the data, Cyril said, oh, let's do lipidomics. And this is exactly what we did, and the results are in front of you. And what we found was, when you look at global lipidomics, when you knock out, knock down CDPK7, the only phospholipid which showed the cursor is at the wrong, this thing should be on the top green circle yeah, there you go, because that will land me in trouble. So this is where we found the difference. This was phosphoryethanolamine. Most of the other phospholipids remained the same. And as shown in the bottom panel, we also found that the phospholipid content was also altered. The C16, C18 containing fatty acids were significantly lower, which was also partly true in the case of the phosphoryethanolamine specific fatty acid. So these results suggested that toxoplasma may regulate in toxoplasma, CDPK7 regulate phosphoryethanolamine biogenesis. Can you go forward? Then we would, as I said, we're doing these parallel assays which are very informative in terms of understanding phospholipid synthesis. So we provided two precursors, ethanolamine and choline, and looked at formation of the phospholipids which they contribute to. And when ethanolamine was 14C-ethanolamine was put into the parasite and shown in the right-hand TLC, you had we had the formation of phosphoryethanolamine, and you put choline, and then you get phospholycholine. But as you can notice on these TLCs, in sync with the phospholipid data, phosphoryethanolamine was significantly reduced and not phospholycholine. For a reason I put PFPMT, this is an enzyme basically which facilitates the formation of phospholycholine from ethanolamine, and this is very important, interesting, because it's not coded in toxoplasma. It is coded only in Plasmodium, and this becomes relevant in when I talk about the Plasmodium data. And what we also what we found was that's why you only see ethanolamine making PE and choline making PC. And what we also managed to do was when we supplemented or provided ethanolamine, excess ethanolamine in the cultures shown on the left quadrant, is that the loss in the parasite application or defect in parasite application was somehow reverted when we provided ethanolamine, as shown on the right bar, on the right of the third most bar there, and so on and so forth. On the right hand side, this is a quantification of the TLC data. So collectively, these data, these results were pointing at phosphoryethanolamine mediated regulation of PE, but by toxoplasma CDPK7 is critical for parasite application. Can we go forward, please? So how is it all happening? And this is what we are addressing as we speak. As I said, glycerol 3-phosphoside transferase GP80, which is indicated here, was differentially phosphorylated in our phosphoproteomics results. We went on to show that it can be phosphorylated in in vitro kinase assay by CDPK7. It interacts with it, as shown also in the IP results on the right bottom corner. But this enzyme, what does it do? It contributes to loading of fatty acid chain to glycerol 3-phosphate, and which makes a precursor lysophosphoric acid, which is been converted to phosphoric acid, and which upon dephosphorylation by phosphoric acid phosphatase converts into DAG, which becomes a precursor for PE, PC biogenesis, and sometimes it is selectivity, and I don't have time into getting into it. If the fatty acid content of this DAG formed as this thing can determine which phospholipid it has no preference for. So we think both GP80 and PAP, phosphoric acid phosphatase, since they are differential regulators, so CDPK7 may regulate these enzymes and contribute to this process. It is important to note that as you heard, epicomplexin have another organelle called epicoplast. They have their own prokaryotic Fas2 pathway, and then there is a GP80 analog for which is separate, which is there in the epicoplast, which is different. But this is the ER-like GP80, which is a typical transmembrane domain 3, the C terminus and a single peptide, and is shown here in the center are the two phosphorylation sites. So what does CDPK7 depletion do to GP80? Can you go forward? This thing. Yeah. So as I mentioned, as shown on the left-hand side in the IFA, the GP80, this is ER-like. As you can see, it nicely circumscribes the nucleus, and we can actually have not shown here, but colocalized with the ER marker. But lo and behold, when you knock down CDPK7, it's mislocalized, it's all over the place. We can do biochemical assays wherein you can see, since it has transmembrane domains typically embedded in PR membrane, but when you knock down CDPK7 on the right hand panel, most of it becomes soluble upon depletion when you provide ATC. So now what we did was, it was a challenging task we faced, wherein we actually looked at we made the phosphorylation site mutants, wherein we mutated these residues to alanine and looked at what happens to the mutant. The wild type was expressed as at the ER, but lo and behold, the double phosphomutant was cytoplasmic, which basically corroborated the data shown above, that CDPK7 knockdown is critical for phosphorylation of these sites, and which in turn is critical for its localization. And if it is not present at the ER, it's most likely not doing its job, which is to provide these fatty acids for phospholipid biogenesis and so on and so forth. We have made a GPAT knockdown in the lab, and we are trying to complement it with the wild type and the mutant, and although the data, I mean, we need to repeat more, but it is hinting in support of this hypothesis, but only time will tell. Can you go forward, please? Yeah. So what is happening in so these studies, which I in the case of Plasmodium falciparum, it was very striking, and it was that when you knock down CDPK7 in case of Plasmodium falciparum, there was significant drop. In this case, phosphatidylcholine, not phosphatidylethanolamin, was not that much altered, but phosphatidylcholine was the major phospholipid which was altered. And as you can see on the right, and now if you notice, if you remember what I said earlier, there was one spot on TLC when you provided ethanolamine. If you and shown on the left is the pathway, it was making only one phospholipid, PE. But in the case of toxoplasma, when you provide ethanolamine, it has two spots suggesting that there are two phospholipids, and this is precisely what I was trying to say. Because of this presence of this enzyme PMT, the ethanolamine precursor can lead to formation of both PE as well as PC, and this is not my discovery; it's been known. But what was interesting to note was when you knock down CDPK7, knockout CDPK7, there was decrease in PC formation via both these pathways, as you can see on the right hand top right corner in the TLC and the quantification. And the PE levels were not that significantly altered. We provided choline. We provided choline excess choline in the parasite. There was modest but statistically significant recovery of the growth, as shown on the bottom right corner, and so on and so forth. This was very interesting. The PMT knockout, published by Chakriba and Mauman many years back, also showed that it is not essential, but it is actually slow growth phenotype, and this is exactly, we actually follow the same protocol, and choline can partially rescue the defect in parasite, replication. Can you go forward? Yeah. So how does what does PFCDPK7, how does it regulate phosphatidylcholine? If you remember on the previous slide, and don't have to go there, and I have shown that PMT as well as shown here is phosphorylation levels are significantly reduced in the case of Plasmodium falciparum. So we wanted to know what's happening to PMT. The answer is right in front of you, where you see the CDPK7 knockout, the levels of PMT were significantly reduced. We checked the transcript, they were just fine, but we added the same proteasome inhibitor, which I think Suman used in his studies. It recovered the PMT levels, suggesting that CDPK7 knockout somehow is leading to degradation of PMT in these parasites, and possibly, given its importance in PC synthesis, that could be one of the major contributors for decreased phosphatidylcholine formation. Can you go forward? These studies are going to be going online today. We'll publish today. But after that, in recent times, what we managed to do was which is not going to be published, but it's, I think, interesting data, which is I decided to show. We made these phosphomutant lines for PMT. The reason it's interesting is there was severe growth defect in the phosphomutant. The parasite was slow growing, and shown here is a defect in parasitemia. And you can also see on the right hand side, while the wild type parasite is expressed, this is GFP, the mutant parasite expression was significantly very low. I mean, of course, you can, if you blow the blot up, you can see some levels, but when you provide MG132, which is a phosphoproteasome inhibitor, you can recover it back. So which basically came together and suggested that CDPK7 is important for the stability of PMT, which obviously is critical for phospholipid biogenesis, and that's how it may be controlling PC biogenesis. Go forward. The story doesn't end here, because ethanolamine kinase was another target which was shown in the previous pathway in the first step of phosphorylation of formation of ethanolamine to phosphoethanolamine, which is extremely critical, which can be used both for PE and PC biogenesis. And we found was EK phosphorylation was significantly reduced in our phosphoproteome, and that's why we followed it. And we generated parasite line, and we had the phosphomutant, which also showed defect in parasite replication. But what interesting to note was it was also showing those similar abrogation of ring to trapezoid maturation, as shown in the GIMSA slide on the bottom left panel, which had some similarity with the CDPK7 knockout I talked to you about. Can you go forward? You can quantitate these kind of parasites, and also we also found defects in parasite replication, as shown in the extreme right. And when we did IFA to see what is happening with these parasites, what we found was, as you will see in the ring or late ring or early troughs, you will typically see MSP will be circumscribing the parasite membrane, even though it is there in the merozoite stages, but just after the parasite has been formed. The RAP1 is in the parasitovacuolar space in the first two slides, which is what you typically see. But in the case of the mutant, you saw very abnormal parasites, as I just mentioned, and often you will just see RAP1 floating around in the parasite in the erythrocyte cytoplasm, which also suggested that some leaky parasite membrane, so on and so forth, some defects. And collectively, these findings suggested that contribution of CDPK7 phosphorylation to the function of these enzymes is important, and so on and so forth. As if I won't have time, or I will have time for 2 minutes, this thing, because since it is a drug symposium, I thought I will show this. So, and I'll quickly I'll skip my slide, but I'll drive home the point of this thing. So this is all with the CDPK7. This thing, and remember what I said at the outset. We have found the CDPK7 pathway, which is regulating phospholipid biogenesis by regulating the enzyme. But remember, what is at the top? It is the PI4 kinase, or the lipids it makes, as shown here. So what has happened in the last few years, PI4 kinase from Plasmodium has emerged as a major target by courtesy work done by Elizabeth Winslow first, and now Kelly Chibale and some others. To cut a long story short, and these inhibitors which they have developed, one of them actually has gone into clinical phase 1 clinical trial, and so on and so forth. So we asked Kelly for the inhibitors and MMV for the second inhibitor, and we see what does this inhibitor do to CDPK7 and downstream processes. Can you go forward? So as I said, CDPK7, as we published earlier, resides in these small puncti, as shown on the left. When we treat with this inhibitor, it becomes cytoplasmic. It's all over the parasite, which is basically again telling you that the binding of this phospholipid because this inhibitor will block the formation of PI4Ps. And if they do not dock to CDPK7, they will not take it to the localization of which is these vesicles, and that's exactly what we are seeing. Can you go forward? Can you go forward? I'll skip this slide. And we found similar defects. But we also found was that similar defects in PC, and not only PC, also in PE formation. And given that CDPK7 is regulated regulates PC biogenesis, and now this upstream PI4 kinase inhibition suggests similar results, suggesting that at least, if not all, most of this, some of this is, may be a result of PI4K CDPK7 signaling. And lo and behold, at the bottom two slides, when you add the inhibitor, you see similar degradation of PMT as well as EK, which we found with CDPK7 knockout. Can you go forward? Which basically, can you go forward, Asif? And so on. The last data slide, which is, then we wanted to see what consequences, where it could be the problem. One of the things with the Plasmodium is that it has these parasitovacuolar vacuolar membrane, and then it has these tentacles or these tubulovascular networks, which are extension of this PVM, which go in, as shown on the DMSO or the 3D7 slide in the case of two parasites, and so on and so forth. And these are important for nutrient uptake and so on. Both in the inhibitor-treated parasite as well as in the knockout, these tubulovascular networks were greatly stunted. So their formation was abrogated, suggesting that one of the ways, and since they are important for nutrient uptake, and we still need to demonstrate this thing, which nutrients and so on and so forth, that may be via this, at least one of the ways by which it, because these are abrogated, and that may be causing the resultant effect, which is effect in parasite. Can you go forward? Yeah, so this is the summary slide, and shown on the left and the right. I mean, I think I've already explained to you the phosphoinositide regulate the two CDPK7 in Plasmodium and Toxoplasma. In the case of Toxoplasma, it is PE biogenesis, and of course Rab11A vesicular trafficking, which I didn't talk about today, which contributes to parasite replication, and so on and so forth. And these studies were published earlier in PLOS Pathogens. In the case of Plasmodium, it is very interesting that this is a different set of targets CDPK7 has, and it is PI4 kinase signaling which regulates CDPK7, and in turn regulates these enzyme in phospholipid biogenesis, which in turn regulates phosphatidylcholine formation. Thus far, while this drug I talked about, which has been used in clinical trials, therefore there are no targets of PI4 kinase which have been identified. So one of the given in the light of our results, I think not only our studies delineate this pathway, it also suggests at least one of the putative targets by which it may be acting on the parasite. And these studies, which also has Christian as one of our this thing with Cyril, and this thing are going to be published in more reports soon, and so on. Go forward. And this, so Priyanka, Ranjana, the senior student and a senior postdoc. They were and along with Anuj Tripathi, they contributed majorly from our lab in these efforts. And Praveen and Rahul in recent times, Praveen earlier and Rahul now have also made their share of contributions. Very importantly, our collaborators without whom it's not be possible, Keshav and his group and, on proteomics, Cyril Boti on lipidomics, whom you will be hearing tomorrow. And of course, Christian Dorig, longtime friend and collaborator. Most of the funding came for this work from the Department of Biotechnology. And my apologies for the goof up, if I can call it, but also my applause for the person who was moving the cursor around. He was absolutely put to the point. Thank you very much. Yeah. He's just showing that he's still awake.
I think, I'm awake. That was great. I know the story because I contributed a tiny bit, but I have a more general question.
Sure.
So, because as your nice figure at the beginning showing that when you knock down these proteins, you make fewer nuclei per schizont.
Right.
So, my question, I've got two questions. First,
sure.
This is very similar to the phenotype that we've got for a couple of knockouts. So, for example, right. PFPK7 knockout has almost exactly identical histograms, and the and the PFCDK5 as well. So, my question is, at least for the PK7, there is a published phosphoproteome of the knockout versus wild type. Yeah, it would be interesting to actually see if there is some overlap in the differences in phosphorylation between the two, because they might work in a pathway, maybe. Who knows?
Yeah. So Christian, if I can answer your question and then also add to what you just said. So, especially in case of Plasmodium, I know you have limited appreciation for Toxoplasma, but that's why we do in a limited way. But we'll stick to Plasmodium. But Toxoplasma has been a great system, and what it shows is basically, even when we say they are similar parasites, how beautifully the substrates are different. Because both are leading to phospholipid biogenesis, but some of the sites are actually not even conserved on Toxoplasma protein or Plasmodium protein. So basically, this shows the plasticity in the system, and that may be because to do with the one of the things may be the host in which these parasites wish to survive. Now, coming to your question, so often we got asked this question before we had this data. You are seeing a defect in the ring root trophozoite, and how can you see a defect also in the division of the parasite? I mean, I said, well, I thought it was a very stupid question, but anyway, because the person who asked is not here, so let me go ahead with that. My point is just that. My point is exactly this. It is, if let us say, for argument's sake, there was a nutrient uptake effect, if what you're saying, what we showed with TV, we believe it's correct, but if it's activating, it is contributing in a major way, then it may have, because we know nutrients are important for cell division. So that is then it may have a downstream effect.
Yeah, I agree. Yeah.
And so it is quite possible, because remember one thing, our phosphoproteomic studies were done on the ring to trophozoite stage, which is the first defect we see. Not at this thing. So I do not know if it has direct targets during division in the trophozoite this thing, but if what you're saying is correct, it won't surprise me because there is a stallation of cell cycle. Subsequently, if you look in Schizons, PFPK7 and the other one, PFCDK4, whatever, CDK 5, they are altered and that can be checked.
Yeah, it'll be interesting. Yeah, no, that was great. Thanks, Pushkar.
And if I may one more, because since you are author on the paper, I mean you would know. Matthias Marty had this diisophosphatidylcholine paper in Cell, and they did the transcriptomic studies where, of course, they say that phosphatidylcholine deprivation leads to more differentiation. On the flip side, it can lead to more sexual division if you provide more phosphatidylcholine. That also they say, but in a limited way. There, they did a transcriptomic analysis, and they found, lo and behold, PMT, I think also EK, lo and behold, CDPK7.
Okay. That's good.
But which probably is a way of parasite, to fight back, and that's why he probably trying to turn down the genes. We put it as a speculation in a paper, but I think it was very interesting to know that.
No, very, very good, very consistent. Yeah, thank you.
Yeah.
Thanks, Pushkar.
Yes. So all the lipids, which I showed, the only lipids which showed difference by lipidomics was PE and PC. Yeah, so I think Cyril may have done it. I may not have it in the data, but I don't think there was significant change. But I can get back to you with a more accurate answer. So I'm not that concerned about it, Asif. Please, I'll let you complete. Sorry, So here is my question. Here is my answer. If you have a mutant of the parasite which is showing defect via this pathway, which is having defects in choline formation, there you can see when you add choline, this thing. So over and above that. And at the outset I said it's a modest but statistically significant change, and they also do it in the PMT knockout. That's exactly where we got the idea from, where they show but it is a modest, but it is just a proof of the principle. To answer your question, it is indeed possible then if the PMT arm of the pathways is not working properly, parasite may induce this, because there are choline transporters which are available and they are drug targets as shown by Henri Vial and others, and there it may just activate they may, because the requirement is such, it may just try to take up choline and use it, but of course not in a major way, like I rightly pointed out. So if you add choline to already running culture, you will not find that much significant difference. But of course, it is 200 micromolar of chlorine. It is just as a proof of the principle which we followed because PMT was one of the targets, and I thought, let me see what is happening there. So that could be the reason why you didn't find and so on and so forth. It won't bother me much because I'm not anyway trying to make a big deal out of it. Yeah. My apologies. I'm sorry. Yeah. So, Asif, I have to continue logging on till the end. I don't know. We can leave it on. When you have the next break, to be fair to the student, do it after the next talk so that she doesn't get interrupted. All right? Yeah.
Excuse me, everybody. May I request everybody who is joined us online to switch on their video cameras so that we can take a snapshot to make a memory out of this hybrid workshop that we are enjoying today. Thank you. We are done? Okay, thank you everybody. Now we'll start with the next session, which is the short presentations by our students and postdocs. The first presenter is Neetu Verma. Neetu, are you ready?
Yes, ma'am.
You can unmute yourself. We made you a presenter. You can start your presentation now.
Should I share my slide?
Yes, please.
Should I start?
Yes, please.
Good evening, everyone. I will be just giving a brief description of my work that is based on the functional characterization and localization of organelle base excision repair protein of Plasmodium falciparum. As we know that mitochondria is constantly exposed to oxidative stress due to the high level of ROS that are generated, and which leads to the DNA lesions, and these lesions are then further repaired by the organelle base excision repair. Now, DNA glycosylase catalyzes the first step of the birth pathway that recognizes and excises the damaged DNA base and creates the AP site. Now, PF-endo3 is a homolog to human NTH1, that is a DNA glycosylase, and it shows 35% sequence identity with its human homolog and consists of a mitochondrial target sequence at its N terminal. Further, the purified PF-endo3 also showed a sharp peak at 420 nanometer when under UV spectroscopy and indicating the presence of iron sulfur cluster. The mitochondrial localization of PF-endo3 was confirmed by immunofluorescence microscopy. And for the functional characterization of protein, we used 5' FAM labeled synthetic DNA analogs, and we found that PF-endo3 was able to remove all the modified bases, that is thymine glycol, 8-oxo-G and DHU, although with lesser activity compared to 8-oxo-G, suggesting that PF-endo3 act as a DNA glycosylase that can process structurally dissimilar lesions. Further, the AP site that has been generated by DNA glycosylase is processed by AP endonuclease. Plasmodium consists of two nuclear encoded mitochondrial AP endonucleases, that is APN1 and APE1. APE1 is the human homolog of APE1 and
Neetu, are you logged in from another device also?
Along with the endonuclease activity, also gave the additional exonuclease activity. These exonuclease activity were much more prominent in case of mismatched template compared to matched template, suggesting that PFAPE1 also function as a proofreading enzyme. Further, we analyzed the 3' to 5' exonuclease activity on recessed, blunt and gap template, and we found that it showed almost similar activities for all the three templates. And the kinetic study of PFAP-E1 further revealed that the PFAP-E1 has quite less enzyme activity compared to that of human AP-E1. Further, PFAP-E1 also exhibited the NIR activity in TG and DHU template, although this activity was not observed in case of 8-oxo-G. Then APE1 also gave the endonuclease activity at wide range of pH, where the maximum exonuclease activity was seen at 8.8 to 8.5 pH. And in case of thymine glycol, it was 8 to 8.5 pH, the NIR activity. Further, since the PFAP-E1 is a metal-dependent enzyme, it requires metal for its functionality, and although it is functional in presence of MnCl2 and MgCl2, but it was inactive in presence of zinc or calcium. Based on the pH requirement and the metal content, it was seen that the human APE1 human APE1 shows the functionality mutual exclusion of both endonuclease and NIR activity, whereas such differentiation was not seen in case of PFAP-E1. Hence, we can say that both endonuclease and NIR activity are exhibited on the same physiological condition of metal content as well as pH. For the PFAP-E1 also exhibited RNase H activity, where it indicating that it also plays a role in RNA quality control, and 3' phosphatase activity was also exhibited by PFAP-E1 or blunt and recessed and nick template, where the phosphatase activity was clearly seen by the upper shift of the band. So here I can conclude my work that endo-3, that is a DNA glycosylase, exhibited N-glycosylase activity, beta-lyase activity, whereas PFAPE1 is a multifunctional protein exhibiting endonuclease activity, NIR activity, 3', 5' exonuclease, phosphatase, and RNase H activity, whereas PFAP1 exhibited only 3' phosphatase activity and endonuclease activity. Hence, I can say that PFAP1 is the major endonuclease in the plasmodium, whereas PFAPN1 likely to have a backup role for the base excision repair. Thank you.
Thank you, Neetu, for your talk. Any questions? So now, since
Asif, can you remove me as co-host? I need to. I will log in.
okay, sir.
If it's easy, otherwise I can leave my computer.
Just give me 1 second.
Yeah, I mean, if it's too much of a distraction, no worry, because because I'll be shutting down my computer, taking it down. So I hope I don't create a mess.
Yes,
so
you
that,
can log out if you like.
thank you so much, and my apologies again. Sorry. Yeah.
Thank you, Neetu Verma, for your presentation.
Thank you, ma'am.
Next is Mithali.
Hello.
Mithali?
Yeah, hi. Should I start presenting?
Yes, you can start sharing your screen and start presenting your slides and your talk.
Okay.
Mithali, your screen name is showing as Meeta, right?
Yeah, I'll just switch. Yes, I think it's showing as Nita.
Yeah.
But, yeah. So is my screen visible?
Yes, you can make it into a full screen display and start presenting.
Yeah. So is it fine now? So good evening, everyone. Today I'll be talking about the brain pathology and cytokine expression in Plasmodium burgei infected C57BL6J mice. First of all, talking about the cerebral malaria pathogenesis, there are two theories which have been discussed in literature over the years. One is the cytokine theory and other is the sequestration theory. We'll be focusing more on the cytokine theory. According to the study that we have conducted, we have studied about the infected RBCs. First, the infected RBCs that bind to the overexpressed ICM1 in EPCRs. This binding leads to release of toxins, which further leads to the activation of a cytokine storm. This cytokine activation, it leads to the bind. Suppose, for example, we have two cytokines, TNF gamma and TNF alpha and IFN gamma. They bind to the TNF receptor. After that, what happens is they lead to the upregulation of they lead to the activation of NF kappa pathway. NF kappa pathway further leads to the upregulation of CXCL10 and CCL2 and other chemokines. These chemokines, and along with the cytokines that have been released during the sequestration, lead to recruitment of CD8 plus T cells in the brain. This recruitment of CD8 plus T cells further lead to exaggerating the cytokine response and leads to the pathology that we observe and that we'll be studying in the slides to follow. Now, why do we need to focus on the cerebral malaria pathogenesis? Firstly, there is enhanced multi-drug resistance, and the first line defense that we use, that is artemisinin resistance, has been developed even against those. And then therefore targeting the parasite is not sufficient, and we also need to focus on the host pathogenesis if we want a drug of choice. Therefore, we need a drug that can target a parasite as well as the pathogenesis that is going on in the host. So therefore, we are focused on the cerebral malaria pathogenesis in the host. First, what we did is we infected the C57BL6J mice with Plasmodium burgei ANCA strain, and then After the 15 days of infection, or when the mice were in morbid stage, which was around 8 days, then what we did is we dissected the mice and we made sections of the brain. After doing HE staining on the brain sections, we compared the histopathology of brain of mice, of control mice, and then of PBA infected mice. What we observed is, when the mice is not infected with PBA, we will observe vessels which are vacant. That is, vessels will not have RBCs that will have the parasite. Whereas in Plasmodium burgeri infected mice, we will observe vessels, as you can see in figure 2E, we will observe the vessels which have blood clogged in them, and this blood will be infected with this you can see is a ring stage parasite. So the ring stage parasite, even if there is one parasite that is infecting the blood, it can block the whole vessel. Now this blockage, what it does is, it can lead to the hypoxia and further lead to coma that is observed in most of the patients suffering from cerebral malaria. Another finding that we made from histopathology is the monocytes also are recruited when there is an increase in inflammatory cytokines. It also leads to an increase in monocyte infiltration within the brain and within the vessels. Here that you see is there is a monocyte that has hemozoin pigment. These monocytes try to digest the RBCs and the hemozoin pigment is produced. This hemozoin pigment can be observed here and it is brown in color inside these monocytes. Whereas if you compare with the control mice, the vessels do not have any sequestered parasites and the cortex is also looking fine without any parasite or hemozoin pigment. And the monocyte infiltration is also not there. Further, when we observe the cortex of brain of infected versus the control mice, we will see this kind of inflammatory cytokine storm. This is reactive gliosis that is observed in the brain of infected mice. Whereas if you see the cortex of brain of a non-infected mice, you can see the pathology is totally clear and we observe normal neurons in normal cells. Also, if you see closely, there is endothelial swelling in these cells. All these features together, this sequestration leads to the release of a cytokine storm. Now what the cytokines does is they will lead to an increase in the REMO scoring. REMO scoring is a quantitative estimation of the cerebral malaria symptoms that we observe in mice. This has been modified a little for our usage, and if the mice are totally fine, they'll be given a score of 0, whereas as the disease progresses and there are increased symptoms such as ruffled fur, hunchback, and we'll observe paralysis also, this will lead to an increase in the RAMO scoring. As this RAMO scoring increases, we observe that the histopathology got worsened and there were increased infiltration of monocytes, endothelial swelling, and perivascular edema, as you can see here. Further, what we did is we wanted to see if during this infection, the brain of mice is also, the hippocampus of mice is also getting affected. For this, we did CV staining. During CV staining, we observed that the control mice displayed a very well intact neurons and the hippocampus was intact. Whereas the PBA infected mice showed the neuronal dropout, which could be a factor responsible for the increase in the RAMO scoring. So further, we moved to check if the cytokines were affected. Then we see that we studied 4 inflammatory cytokines, IL-4, IL-10, IFN-gamma, and TNF-alpha. The role of IFN-gamma and TNF-alpha is well established in cerebral malaria pathogenesis, but still there are huge number of controversies associated with all these cytokines. In our study, we found that the levels of IL-4, IL-10, IFN-gamma, and TNF-alpha, all these cytokines increased considerably. And as we all know that to maintain the inflammation in the host, the regulation between anti-inflammatory cytokines and pro-inflammatory cytokines is very important. If this balance of pro-inflammatory and anti-inflammatory is dysregulated, it leads to the pathology that we observed earlier. So here we can see that the balance was dysregulated since an increase in the anti-inflammatory cytokines IL-4 and IL-10 should have led to decrease in TNF-alpha and IFN-gamma concentration, but this was not the case in our study. This is a highly controversial study and some say that IL-4 and IL-10 are protective, but not in our study. Here, the IL-4 and IL-10 concentrations did not lead to a decrease in TNF-alpha and IFN-gamma concentration. Here we observed that IFN-gamma increased, and along with that, all other cytokines also increased. And the increase in IFN-gamma and TNF-alpha can be responsible for the sequestration that is observed here, because this increase leads to the increase in ICAM and other adhesion molecules, which further increases the sequestration. So this is a totally interdependent pathway, both sequestration as well as the inflammatory cytokine hypothesis. First, the innate and adaptive immune responses are activated, which leads to increased production of adhesion molecules, which can further...
5 minutes is over.
What?
Your 5 minutes are over.
Okay. So in conclusion, we can say that both the theories are valid, but still the cytokine and are interdependent. Thank you.
Thank you, Mithali. So, any questions? Since we don't have any questions, we are moving on to next presentation.
Thank
Triveni?
Yes, sir.
So now you share your screen and start your presentation.
Okay. Thank you. Good evening. Today I'll be talking about exploring a novel exonuclease of the malaria parasite with an unconserved functional LCR. Our lab majorly, I'm currently undergoing my PhD under the supervision of Dr. Saman Habib. Our lab has already given us a detailed in silico analysis of the organellar base excision repair pathway. And as we all know that the organelles, mitochondria and epicoplast are very essential for the parasite's survival. And hence, it is imperative to maintain the genome integrity. While delineating role of several such enzymes, we have come across a very particular unique exonuclease, 5' to 3' exonuclease with a 173 amino acid insertion region that I've marked in purple in the model. Immunofluorescence microscopy have confirmed that this protein targets to the epicoplast. As far as the biochemical activity of the protein is concerned, it has got 5' to 3' exonuclease activity on double-stranded DNA, which include recessed, blunted, and one nucleotide gapped template. And it also has 5' to 3' activity on single-stranded DNA. Interestingly, the protein has an intrinsic 5' endonuclease activity. Now, while finding out putative candidates for the long patch Burr mechanism in the epicoplast, we found a dearth of a 5' flap endonuclease, and our protein can fill in that position. While carrying out the flap activity, we found a prominent 25 nucleotide flap alongside with lower oligonucleotide products. So we thought that probably this protein has got additional 3' to 5' exonuclease activity, and by in vitro DNA cleavage assays, we proved it to have 3' to 5' exonuclease activity on single-stranded DNA, which made our protein bipolar on single-stranded DNA and unipolar on double-stranded DNA, and as a result, it makes our protein quite different from other known classes of exonucleases. We went on to compare the activities of the falciparum protein with its Burghi ortholog that lacks the insertion and also the 4F4S cluster. As far as biochemical activities is concerned, the Burghi protein showed a difference in oligonucleotide product that is, gave a higher oligonucleotide product in comparison to the falciparum, which gives a lower nucleotide product. And for the one nucleotide gapped template, single-stranded DNA and blunted template, the protein did not show any activity. As far as the blunted template was considered, the protein not only did not show activity, but also it did not bind to the DNA, which means that the DNA binding to blunted was compromised for the Burghi protein. We then set on to plot the plausible causes for the differential activities in the two orthologs. The first we thought was the lack of 4F4S in Burghi, but however, it was proved to be not. Next, we mutated a certain aspartate residue, which is present on only few species, including falciparum, and largely absent in the other orthologs. And this aspartate residue lies very close to the DEDD motif, which is in general responsible for the 3' to 5' exonuclease activity of proteins. However, the mutation caused no difference in biochemical activities. Next, we went on to delete the insertion sequence by overlapping deletion mutagenesis and generated a shorter protein. Carrying out activities on different templates, we found out that the deletion mutant acted like the wild type protein as far as the 5' resist and blunted templates were concerned, but it acted more like the Burgheim protein in losing activity on the 1 nucleotide gapped template and the single-stranded DNA in a 5' to 3' direction. Additionally, the protein completely lost its 3' to 5' exonuclease activity on single-stranded DNA. And this was very interesting because this property was what made our protein unique in the first place. Next, we went on to carry out site-directed mutagenesis and identified residues important for the formation of the FPS cluster. This included H393, C532, and C436, where we can see abolition of the peak at 420 nanometers. Thus, I can conclude by saying that our protein is one of the first that is going to give a role of a functional LCR that attributes to its diversification of activities and makes it a putative class of a new exonuclease. Thank you.
Hello, Triveni, are you done?
Yes, sir.
Okay, thank you for your presentation. So we don't have any questions here. Thank you for your presentation. We are moving on to next presentation. Ankit Gupta.
Yeah.
Yeah.
Hello everyone. Good evening everyone. Today I am going to present my work on a topic CLAG3 plays a key role in nutrient channel formation. So first of all, I would like to introduce with the Ropich complex. Ropich complex proteins are being mainly synthesized during Schijont stage. Once they have been synthesized, they have been stored in the rotary. Once the Schijont rupture, these merozoites invade fresh RBC where these proteins are get transported at the surface of the infected erythrocyte, serving a number of functions such as cytoadherence with the endothelium, vascular endothelium, and nutrient transport. Okay, so Ropich complex have basically three proteins: Ropich 1 complex, Ropich 2, and Ropich 3. Ropich 1 complex is a CLAG multigene family which consists of CLAG9, CLAG2, CLAG3, and 8. CLAG3 further has two paralogs, 3.1 and 3.2. Those are epigenetically regulated and expressed at a time.
Ankit, your slides are not moving.
Sorry? Yeah, they are moving.
No, not here actually.
They are moving at my side.
So you have to actually reshare your slides.
Okay.
Now share your slide, share your screen.
Yeah, now you got.
So yeah, now it's moving.
Okay, so so in order to so the previous report support that the CLAG3 is a key component for the nutrient channel transport. So that's why we try to make a knockout by introducing a stop codon in the ORF in the parasites having the single CLAG3, which is known as a hybrid CLAG3. In addition, as a control, we also make a knockout in the CLAG 3.2 in the parasite that has two paralogs. So in the 7G8, the CLAG 3.2 was knocked out, whereas 3.1 is remained expressed. Okay, so we further tried to see that the expression is lost or not in the CLAG 3H knockout parasite, and by using a IFA using CLAG3 antibody, and we found that the CLAG3 expression was disappeared, which was also reproduced in the Western blotting, where you can see that in CLAG3H knockout parasite, the band get disappeared while it was present in the wild type parasite. In the control parasite, 7G8 knockout, the band was slightly fainter than the wild type because CLAG 3.1 remained expressed. So the same result was getting verified by the transport assays, where we found that the CLAC3 knockout parasite have a reduced solute uptake in the various categories of the solutes, such as sorbitol, PHTMA, and proline, whereas the control parasite 7G8 knockout, where the 3.1 remained expressed, although the 3.2 was knocked out, there was no change in the knockout.
Sorry,
There
Ankit,
was
to
no
interrupt
change
you
in the
again.
solute
So your
uptake for
slides are
the
again not
knockout and
moving.
the wild type parasite.
Sorry, Ankit.
Okay.
Your slides are again not moving actually. So
No, they are moving.
so we aren't able to go to next slide. What you were saying.
Okay, I should again. Okay, now you got?
Yeah. Every time you have to do this.
So now we check the growth profile of the knockout parasite in the nutrient media, which is RPMI, which has an abundant nutrient. While in the nutrient deficient media, we found that the 17-fold reduction in the growth of the knockout parasite. Okay, so that suggests that the knockout parasites have they have a vital role in the nutrient deficient media, which is quite similar to the human plasma. So in also I found solute uptake assays that the 25 to 30% slower uptake for the nutrients in the knockout parasites. And once we checked for the pharmacology of using various inhibitors, we found in defloridine there is a 100-fold decrease in the there is 100-fold increase in the Ki values. In the same way, ISG21 inhibitor and IPSA1 inhibitor, we found a 10-fold increase in the Ki values. Means the parasite becomes 10-fold less sensitive to the inhibitor. Whereas in the ISPA28, there is no change in the sensitivity level of the mutant and wild type parasites, knockout and the wild type parasites, supporting that ISPA28 is interacting with the CLAG members. Those were encoded in the different chromosomes other than CLAG3. Okay, so now we checked for the transcript profile at last. So we found that in the knockout parasite CLAG3H knockout, the CLAG3 expression get lost, whereas there is a significant upregulation in the expression profile of other CLAGs, especially in the CLAG9, which was significantly expressed and the expression was increased. Whereas in 7G8 knockout parasite, which was taken as a control, there was no change in the transcript expression levels. So our hypothesis is that the nutrient channel is basically composed of 3 proteins. ROPES2 and ROPES3 are constant and they are indispensable for the parasite. However, CLAG3 is dispensable. It means if you knock out CLAG3, the other CLAGs such as CLAG2, they will take their position and forms a channel. So, and if you knock both, then the CLAG8 or CLAG9 will take a position. So this is basically our hypothesis that CLAG on the other chromosomes, they participates in the channel formation. Thank you.
Thank you, Ankit.
Thank you.
Any questions? So Ankit, we have no questions. So thank you very much for your fast presentations. So we will move to next presenter. So Seema, are you ready?
Okay.
Yeah. Yeah, Ankit, thank you.
I'm done.
Okay, thank you. Achha. Oh. Thank you, Ankit, for your presentation. Thank you. So we don't have any questions right now. So we are moving to next presenter. Seema, are you ready?
Yeah, sir, I ready. I am. Please allow me to share the screen.
You share your screen. You are allowed
to. Is my slide is visible?
Yes. Yes, your slides are
Hello?
visible. Yes.
Oh.
So kindly check if they are moving.
Good evening to. Okay, I start my presentation. Good evening to everyone. I joined CDRI in Jan 2020 and my doctoral work revolved around to explore the structural and functional diversity of HSP40 in malaria parasites. This HSP7040 forms a central hub in protein folding machinery, in which it helps in the folding of proper folding of native protein co-translationally and post-translationally. This HSP40 is a co-chaperone of HSP70, where it binds to unfold protein and deliver to HSP70, and it also stimulates the ATPase activity of HSP70 through interaction by its conserved J domain. And this cycle will lead to the release of correctly folded protein. So the striking feature of plasmodial HSP40 is the specific expansion and diversification of these HSP40. So there are 4 HSP70 and 49 proteins are suggested to fall under this HSP40 family. This will hint towards the moonlighting, or we can say HSP70 independent rule. This HSP40 is characterized by J domain with signature tripeptide HPD motif at the N terminal and a less conserved substrate binding domain at a C terminal. So on the basis of domain organization, these are classified into 4 types. There is a difference in type 1 and type 2 only because of cysteine rich domain, which is present in type 1 and absent in type 2. Whereas type 3 and type 4 are unusual larger proteins which have only J domain. Another interesting fact is that this type 4 HSP40 is unique to Plasmodium falciparum. So these different types of HSP40s are localized in different cellular compartments, and some of our HSP40s are also exported to human host RBCs. These exported HSP40s has Paxil motif at N terminal. This will help it from export from parasite to host RBC. So these are the representative HSP40s. On my research, studies are ongoing. These HSP40s are localized to cytosols, organelles, and these are exported. They vary in the electrostatic potential that hints towards their different binding affinity, or recognize how do they recognize their specific client or substrate. So for the exploring the structural and functional characterization, I performed some biochemical in vitro aggregation inhibition and limited proteolysis activities to check their conformational dynamics of these cytosolic organelle exported HSP40. In my some preliminary data, it seems difference in the aggregation inhibition assay by cytosolic and exported HSP40. This structural investigation is will studied by the help of SARS and X-ray crystallography. Through the help of peptide spot assay, we will identify the specific client recognition motif of these representative HSP40s. So these proteins are purified and the polyclonal antibodies are raised to check their differential expression and localization under different conditions. And amongst these representative HSP40s, the HSP40 with a better aggregation inhibition assay and activity would further studied for the identification of their interacting partner. So these are some questions and approaches for my for my research studies. That's all. Thank you.
Thank you, Seema.
Yeah. Yes, sir.
So, since we don't have any questions in chat.
Okay, thank you, sir.
So, thank you very much for your presentation. So now we move on to next presenter. It's Nirupa. Nirupa, are you ready?
Yes, sir. Yes, sir. Just give me 1 minute.
Can you share your screen?
Yes, sir.
So now, can we start now?
Yes, sir. Just, I'm sharing. Share. Yeah, I saw it. Share. Okay. Okay. Good evening, everyone. My name is Nirupa, and I have started my PhD at CDRI last year in November 2020. My research work revolves around Plasmodium falciparum telomerase, where I will be exploring the canonical and non-canonical functions. As you all must be aware, telomerase is an enzyme which acts as a savior during the DNA and replication problem by replicating and maintaining the telomeric length by using a RNA as a template. This field is highly unexplored in Plasmodium falciparum, and also the human orthologs of telomerase interacting enzymes are not present in the Plasmodium. This makes this field a highly difficult and also interesting one. There are various studies that have been done in human yeast and tetrahymena telomerase. Human telomerase have been majorly studied in the field of cancer and cell homeostasis. Even now, cryo-EM structures are available for tetrahymena and human telomerase. There are evidence that telomerase has non-telomeric extranuclear function as it is known to transport to the mitochondria under stress condition. But if we look in the studies of Plasmodium telomerase, it is still at a very nascent stage. The Plasmodium telomerase is just known to maintain the telomeric length as its common function. One important finding that has been found is that the Plasmodium telomerase is expressed in all the stages of the parasite, while human telomerase is expressed only in cell, germ cell, stem cell or cancerous cell. So, telomerase is also determined as an essential gene because its mutation in P. burgei showed delayed death of the parasite. This is the domain organization of the telomerase of human and Plasmodium falciparum. There are 4 major domains of telomerase. The first one being this N-terminus domain, which helps in the stabilization of the RNA-DNA duplex. The second one being a telomerase RNA binding domain that facilitates the duplex formation. Telomerase reverse transcriptase domain, that is the catalytic domain and synthesizes the DNA strand. And the fourth one is the C-terminus domain that maintains the telomerase processability and also the localization of the telomerase. If we look at the telomere sequence of Homo sapiens, it is a 6 nucleotide repeat that is around 10 to 15 kb, while in Plasmodium it is a 7 nucleotide repeat with 1 to 2 kb. Also, the telomerase in Plasmodium is about double the size of that of human telomerase. Also, the RNA component, which acts as the template for telomere synthesis, is greater in Plasmodium falciparum as compared to human. So these are the questions that I'll be addressing during my research. The first one being telomerase expression and localization under stress condition, for which I am currently purifying different domains of telomerase for generation of polyclonal antibodies. The polyclonal antibodies that will be generated will also be used for CHIP for understanding the regulatory roles of telomerase in telomeric and non-telomeric regions. I also focus to study the small molecule based perturbations of telomere dynamics, for which I am currently standardizing the TRAP assay, that is telomerase repeat amplification assay, to determine the telomerase inhibitors. As very less is known about the telomerase interacting partners, I wish to identify them through immunoprecipitation experiments and further validate them through transfection experiments in Plasmodium falciparum. That's all. Thank you.
Thank you, Nirupa. So, do we have any questions? So I think chat box is still empty. So now we move on to our next presenter. This is Iqbal from ICGB.
that could be linked to drug resistance. So first, let's have a look about how plasmodium stays in the RBC in an RBC cell. And this plasmodium has its own plasma membrane, and there is another membrane that is called parasitic bacterial membrane that separates the cytosol of the two. And then there is this important vital organ that is called food vacuole that has a number of functions. Now let's zoom into this food vacuole. So one of the major event of the food vacuole is hemoglobin degradation. So hemoglobin is degraded to toxic heme, which is then converted to non-toxic hemozoin through different protein complexes. The majority of the drug that is present today and in the past, they have the food vacuole as the drug target. As you can see, for example, as you can see, the frontline drug, that is chloroquine, acts at this particular site, inhibiting the conversion of heme to hemozoin, thus killing the organism. So the main problem that we are facing today in context of the eradication is the increasing drug resistance. A number of proteins in the food vacuole, are known to show mutations that are contributing to the drug resistance. Two of those are this MDR, multi-drug resistance, and CRT, chloroquine resistance transfer. Point mutation in these amino acids makes them drug resistant markers. So for this study, we first isolated food vacuole. Food vacuole isolation can be isolated through two different methods. One is chemical and other is magnetic method. For both the method, we need droposide lysate. In the chemical method, the droposide lysate was mixed with 42% alcohol containing 0.25 molar sucrose, which is then centrifuged to give a upper debris layer and a food vacuole pellet. Estimate of the food vacuole pellet is in confocal is as shown. You can see the food vacuole here. The dark colored region is the hemozoin. And in the magnetic method, there are two components. One is the supermagnet and the other is the magnetic column. The droposide lysate is passed to this column to bind, and then it was washed and it was elected to get the intact food vacuole. So proteomic analysis of the food vacuole fraction from both chemical and magnetic method, we were able to detect this much amount of protein, 680 proteins. These two different methods. Among those 246 were detected from both. We got 246 common proteins. So out of these 680 proteins, we selected 16 proteins on the basis of transmembrane motif, and we wanted to further study their role in drug resistance mechanism. So we wanted to further confirm the detection of these proteins through confocal. So we performed confocal for these 5 proteins. So this is this confocal data. So we performed confocal using the food vacuole lysate against peptide antibodies for these 3 proteins, MDR, CRT, and SNAP. So as you can see clearly, these three proteins is clearly localized to the food vacuole. To get a more clear view of the localization, we also established 2 transitioning lines for these 2 proteins, aquaporin and DDAC. And here also you can clearly see the proteins localized to the food vacuole here. We were also able to detect the presence of these proteins in lysate, as you can see. So here is a table showing known or already reported mutations among 3 different strains, K1, DD2, and 7G8 in different food vacuole proteins. As you can see, one or two mutations have already been reported in many food vacuole proteins. In our lab, we have also detected mutations in 2 different Indian strains that are yet to be reported. So the main motive of this study is to detect detect new mutations in these food bacterial transporters, and to discover new drug resistance markers, and we want to study the drug resistance mechanism of these food bacterial transporters. That's it. Yeah, thank you. So, any questions? Kind questions?
Yeah. Hi, Iqbal. I have a question.
कौन पूछ रहा है? हाँ, तो मैं आई बोल. Yeah. यहाँ पे ऑन कर दो, सबको भी सुना चाहिए. यहाँ पे ऑन कर दो. हाँ. Yeah. No, those 5 proteins were randomly picked, okay, to just confirm the detection of these proteins in food bacterial fraction. Yeah, 16 proteins were selected on the basis of transmembrane motif. And yeah, random 5 to detect the confirmation if they are present in the food bacterial fraction or not. Yeah. Yeah.
Thank you, Iqbal. So we have our next speaker, Nikunj Bhai. Are you ready, Nikunj Bhai?
Yes, sir. I'm ready, sir.
So can you share your screen?
Yes, sir. Thank you. Is it visible, sir? Hello. Hello. Hello.
Hello. So
Is it visible, sir?
Hello.
Lights?
hello. So sorry, Nikunj, we have actually some change in order. So
Okay.
we have Hina next to present.
Okay, sir. No problem.
Okay, Hina.
Yeah.
So should I present?
Yeah, sure. Share your screen.
बोलो टॉप चल रहा है फोन ना करो कुछ शिफ्ट बैक पीछे क्या हुआ वो बोलेंगे
So Hina, are you ready? Hina? Hina, you are mute. Hina.
Audible?
Hina, you are
Hello.
Yeah. So are you ready?
Yeah, I'm ready. Should I start?
Yeah, start. Please start.
Okay. Is the slides visible?
Yes, slides are visible.
Okay. Good evening, everyone. I would like to thank all the organizers for providing me this opportunity to talk about structure-based screening of the pathogen box compounds for identification of potent antimalarials against blood stages of Plasmodium falciparum with PFU-CHL3 inhibitory activity. As you all know, malaria is a devastating disease and its eradication poses immense challenge due to emergence of resistance to the existing treatment modalities. Such daunting concern warrant identification of novel therapeutics. Since Plasmodium falciparum is believed to be the most important causative agent of the malaria, our study was focused in identifying parasitic components that can serve as potential targets.
Deubiquitinases plays critical role in several biological functions and has been associated with various diseases including malaria. Among a large plethora of Plasmodium dubs, PFUCHL3 is cardinal to the parasite survival and emerges as a potential therapeutic target. However, despite evolutionary conserved biochemical trait, it displays considerable architectural variation in the residues defining the interaction in the active site pocket. So taking lead from this, the study begins with a structure-based virtual screening of pathogen box compounds against the crystal structures of PFUCHL3 and its human counterpart. So on the basis of better docking score, top 100 molecules from this primitive screening were evaluated for the ADME profiling, and from this, top 50 compounds were evaluated for the inhibitory action on PFUCHL3 activity using in vitro assay. So among 50 compounds, 2 compounds displayed an inhibition rate of more than 50%. Subsequently, dose-dependent inhibition curve of these compounds indicated that compound MMB67603 and 6888704 displayed an IC50 of 25.1 and 87.14 micromolar respectively against PFUCHL3. Notably, the compounds at the respective IC50 value displayed no discernible effect on the human counterpart. So indicating that both the hits potentially inhibit PFUCHL3 activity over its mammalian ortholog. Additionally, these compounds were found to exhibit significant toxicity against the asexual stages of the parasite at concentration 0.4 and 0.2 micromolar respectively. However, no cytotoxic effects were observed against the mammalian cell lines even at the higher doses, indicating the selective nature of these compounds. Next, treatment of our compounds arrested the parasite growth at the late stages of the parasite life cycle and impeded the erythrocyte reinvasion. Importantly, our compounds reveals a continuous reduction in the parasitemia after 24 hours with a 10-fold reduction at 56 hours, implying that both the compounds are slow inhibitors of the parasite growth. Our Western blot analysis indicated that in comparison to the control set, both the compounds showed enhanced accumulation of polyubiquitinated conjugates, suggesting that the compounds inhibited PFUCHL3 activity for effective parasite killing. So, overall, an amalgamation of computational and experimental approaches led to the identification of MMV67603 and 6888704 as potent inhibitors of PFUCHL3 activity with selective inhibition of parasite growth, which hold promises for further translation studies for development of effective and malaria therapy. So with this, I would like to acknowledge my supervisor, Professor Alonov, for making this work possible. I would like to thank my lab mates for their help and support. I am thankful to Rotary Blood Bank for the continuous supply of blood for parasite culturing, CIF for instrumentation facility, and MMB for pathogen box compounds. Fundings from DU, DST, UGC, CSIR are highly acknowledged. Thank you so much. Happy to have questions, please.
Thank you, Ina. We have a question actually from Shilpi.
Hello.
Okay. Shilpi, you are not audible. Can you be a bit louder?
Hello. Can you hear me now? Hello.
No, your voice is very bleak.
Okay. Just wait. Can you hear me now?
Yes.
Okay. So since you have shown very good data on inhibitor study using UCHL3, so I just want to know what about the essentiality of this gene at blood stages? Have you tried to check or any other data related to it?
Basically, major functional studies on this gene have not been yet done, but a paper in which it is characterized, they have tried to make a knockout of it. So in that case, when they are trying to make, the parasite was not able to survive. So in that case, they have validated it as a essential drug target. Otherwise, the status of its role and its importance or its levels in different stages, in different pathways is not yet been elucidated. So we are working on it currently.
Okay, so you have not tried to check this out? Not in any
What?
stage?
We have checked, it is present in every stage with more prominent expression in the Saigon stage. So that is why what we are getting that the inhibition in the later stages. So that is why we are correlating that the slow in the action of this is means they are inhibiting the slow, showing the slow inhibiting potential is might be due to the higher expression in that stage, so that these compounds are effectively killing at the Saigon or the later stages, which is implicated in study as well.
Okay, thank you.
Welcome.
Okay, thank you, Hina. So we will be moving
Okay,
on to
welcome.
our next presenter. Our next presenter is Prakar.
Good evening, all of you. I will be presenting my work on the glidosomal gas safety, looking in the inhibitors for this protein. So first, I will describe about the glidosomal complex, which is present in the epicomplexes. This complex is very well conserved in all the epicomplexes throughout, from plasmodium to other epicomplexes such as cryptosporia and toxoplasma. So why this protein? So this protein has a function. It is a kind of a phosphatase protein, and it has a very high kind of a structural similarity with human phosphatases, which you can look at here basically. But the point being, this it has a sequence similarity below 20% with this human phosphatase. So it's a very good drug target because this protein is very well essential and act as an anchor protein in glidosomal complex. So if we have inhibitor against this protein, so we can inhibit the glidosomal association and further the invasion of the malaria parasite. So initially, my screening methodology, I screened around more than 1,000 compounds against this protein. First by in silico method using Schrodinger, and then by in various in vitro methods. And lastly, I selected around 100 compounds, around 37 compounds with selectivity more than 100. These compounds were belonging to different class of small molecules. Basically, you can say quinolines, thiophenes, carboxyamides, and sulfonamides. Based on, then I selected top 10 molecules based on the selectivity index and IC50 against the malaria parasite. From our this top 10 included our lab compounds and some compounds from MMV also. We discovered two basically compounds, hiatinine and 124-8 from a library, which showed a very good IC50 against the artemisinin resistant strain, that is MRA1240. These two molecules showed in a single digit nanomolar against artemisinin resistant, but however in a kind of a nanomolar range against 37, that is chloroquine sensitive stage. These two molecules were showing selectivity index more than 100, and we took all these 10 molecules further elucidate their mechanism of action. These 10 molecules did show the good activity against the different stages of mature stages of the Plasmodium falciparum, particularly schizons and late schizons. Then, in order to understand how they are acting, we looked upon the ROS production in the presence of these molecules. We did see that these molecules were showing elevated amount of ROS similar to the artemisinin DHA and the H2O2. But the point being, these molecules don't have any peroxide linkages just like in artemisinin or DHA. Therefore, we can rule out the ROS production by any kind of a peroxide linkage breakage. Therefore, we thought that these molecules might be acting on the hemoglobin pathway, that is the major source of ROS production, or any ROS detoxification. They might be inhibiting some ROS detoxification enzymes such as catalase or superoxide dismutase. We are still looking into that part. Further, we looked whether these molecules can inhibit the Schizon rupture and subsequent merozoite invasion. If that is really the case, we can actually hit at the Achilles heel of the malaria parasite and decrease the, which we can say, the exponential increase in the percent parasitemia. Upon elucidation and looking, we did saw that creatinine molecule did show a good activity against the rupture and subsequent invasion. Further, 132.4b and 2076 did prevent rupture. However, bedaquiline prevented rupture, also the invasion. Same with the 1634402, that is the MMV compound, vildacidine, was showing good activity. Further, then we did all the screening against the GAP50 protein. So we further thought that whether these molecules are preventing the association of GAP50 to its partner proteins. So there was a paper by Dr. Pavan Malhotra in Scientific Reports and further in PLOS Pathogens telling about the FIL1 complex in association with the GAP50. So we took the FIL1 antibody and checked whether the FIL1 is correctly trafficked on the malaria parasite membrane in presence of this compound or not. So here you can see, in case of control, we have a correctly trafficked FIL1 on the surface of the Schizons. But in case of bedaquiline, you see the presence of the basically FIL1 fluorescence in the cytoplasm and not on the surface. Same in case of hiatinine, we are seeing a kind of irregular pattern of the FIL1 association, and 1634402 is also showing a very depleted kind of a FIL1 fluorescence. 2076 was not showing any kind of a difference with that of the control, suggesting that 2076 is not preventing the association of FIL1 with the GAP50, therefore the protein is correctly trafficked. Further, we looked upon whether these molecules are preventing invasion or not. The same, we did the staining with the FIL1 antibody. In this part, we did see, in case of hiatinine, there is invasion blocked, and same in case of bedaquiline, there is no invasion. We saw the merozoite is sticking to the surface of the RBC but not invading it. In case of MMV 1634402, that is vildacidine, we saw a digressed Schizon, and in purine and 688271 there was no rupture, signifying that these molecules are did prevent associating with the GAP50 and preventing the proper formation of IMC complex. Further, based on the selectivity, we selected 4 molecules for the P. burgeri infected mouse model malaria to check whether they are preventing the exponential rise in parasitemia in mice infected with P. burgeri. Based on our data, we did saw the bedaquiline did prevented the increase in percent parasitemia along with 2076. 2085 and 124A was no different with the control and was parasitemia increased significantly. Based on the survival curve, we did saw that 2076 out of 7 mice, 2 survived and 5 died with the median survival of 28 days. That is was more than with that of control. In control, we saw median survival of 20 days. In bedaquiline, 3 mice survived out of 7 with a median survival of 19 days. Based on this data, we did see that bedaquiline and 2076 are very good molecules for against malaria. Further conclusion, out of like 1200 molecules, we did identify bedaquiline, 2076, hiatinine and vildacidine as the potent molecules. Bedaquiline is currently a drug against MDR-TB, and we were the first ones to report it as a anti-malarial activity, similar with the vildacidine molecule. Vildacidine is currently used as an anti-COVID molecule and is in clinical trials with the Innovation Pharmaceuticals. Hopefully, they will take it as an anti-malarial trials also. Future perspective, that these 4 molecules can be further taken as anti-malarials, and they have never been before reported for their malarial activity, and they provide a novel scaffold which don't have any resistance in the, you can say, a chloroquine resistant TF-window or artemisinin resistant MRA1240. So therefore, these molecules are very promising as anti-malarials. Thank you. Any questions?
So we don't have any questions right now. So we are moving on to our next presenter. So we have a question actually.
Okay, we have a question. Bedaquiline has a very high cytotoxicity. I don't think so. It has a high cytotoxicity. Basically, in human trials, people have reported up to 800 mg per kg of body weight for TB clinical trials, and in our study also, bedaquiline did not show toxicity up to 200 micromolar. So, with the selectivity index of more than 154, so we can't say that bedaquiline has a high cytotoxicity. Basically, bedaquiline accumulates and it has a very half life. Therefore, if on that basis you can say it's a toxic, but up to 800 mg per kg of body weight in humans, it has not shown any toxicity.
We have this question as well. So can you please?
Generally, out of 10 molecules, we can say bedaquiline, bilirubin, hiatinine, and MND688-271 are showing inhibition of IMC formation in relation to the GAP50 molecule.
So I think we don't have any question. Any more question? So now we are moving to our next presenter. Nikun Bhai, are you ready?
Yes, sir, I'm ready, sir.
Okay.
Can I share my screen, sir?
Yeah, you start sharing your screen.
Is it visible, sir?
Yes, it's visible.
Yeah. Good evening, one and all, and greetings of the day for all the international participants. My name is Nikunj, and I'm working under the guidance of Professor Dalai, and my work is on mainly depends on understanding the role of B cell in cross-presentation of antigen to CD8 T cells. So, as we all know, that mainly the drawbacks of the subunit vaccine is related with the lower amount of the antibody production which compared to the CD8 T cells, and the main thing which we also observed during the RTSS vaccine. So, with the aim of that along with the CD8 T cells can enhance the antibody response, which will further helpful to the CD8 T cells in the activation. Our main hypothesis whether the B cell can do the activate CD8 T cells via cross-presentation mechanism. For that, we have used the CSP protein, which is of malaria, which we have procured from the Geneva Pharmaceuticals, from Dr. Sanjay Singh. As you can see in the upper left panel, we can see that every booster dose with the treatment of the 14 days interval, we are getting the higher antibody response, as we have checked the different kinds of memory B cells, which has CD73 plus and CD80 on the upper left side panel. So every booster dose, we are getting the higher frequency of this different kinds of memory B cells population. Similarly, we have also checked for the main CD8 T cells, specifically central memory T cells and effector memory T cells, which we have been getting using the flow cytometry machine of Atenueps. T effector memory cells are basically CD44 plus, CD612 minus, and central memory T cells, which are CD44 low and CD612 mainly plus. So, in both of the right side, lower panel, left side and right side, we can see that with the gradual booster doses, both of CD4 plus, specifically CD8 plus, central memory and effector memory, both cells are increasing. So once the point has been clear that along with this antibody response, CD8 T cell response are also increasing. Later on, what we did is, as we know that repeated immunization can also create the immune complex, and immune complex can be presented mainly by the dendritic cells with the cross-presentation mechanism, which we have been cross-checked through flow cytometry analysis by using the dendritic cells or as well as BC2.4 cell lines. So is it not shared actually?
So your screen is not being shared. You are mute also.
So now can you hear me, sir?
Yeah, now I can hear you, but your screen is still not shared.
Now is it fine, sir?
So yeah, now it's fine. So please continue.
Okay. So next, what we did is we make an immune complex by using the hyperimmune sera from the repeated immunization to the mice and then feed to the dendritic cells. And here we use OVA as a model antigen, and by using the OVA specific antibody for the flow cytometry, that is synthetic specific H2K of B. So as we can see that when we pulse with the synthetic specific as a positive control, we are showing the higher percentage of the H2K B cells in terms of CD11C, which means the dendritic cells are doing the cross presentations along with the immune complex formation. And main target of our study is to check whether the B cell can do the cross presentation or not. So here we have done a preliminary study with the model antigen OVA, in which we have used the B cells from the naive mice as well as from the OVA immunized mice, and later on we have isolated the B cells as well as then afterward we use the CD8 T cells from the same mice, and we can also use the synthetic peptide CD8 T cells from the mice, and we have further pulse T naive cells with the B cell and OVA. Simultaneously we also pulse with the peptide. So as we can see that in the pulse cell specifically, we have seen that CD8 T cells, which are specifically cytotoxic in nature, 3D107A, we can see a higher response with compared to the neo T cells. So here through this preliminary study, we can say that yes, T cell do the cross presentation. However, there are it has to be repeatedly done, and there are certain things which has to be keep in mind, such as whether it is in vivo, is it possible to confirm that, and there are certain other criteria which needs to be looked after. So in summary, I can say that when during the CSP immunization, we have seen the antibody as well as CD8 T cell generation, and through both blood as well as spleen and lymph node have shown that yes, it is increasing gradually. Simultaneously, CD19 plus CD73 plus cells and CD19 plus CD80 cells, memory B cells, are augmented alongside the double positive cells, which can say that our hypothesis made that yes, along with the gradual CD8 T cells, it will also help the B cells to increase, and further we shall have the CD8 T cells. And finally, I would like to conclude that through OVA cross presentation mechanism as a model antigen, we have shown that yes, we shall do the cross presentation. However, in case of malaria, it is yet to be done. And specifically, me and my guides, Professor Sharadale, we're thankful to Department of Biotechnology, ICMR, as well as the FIST grant for this flow cytometry experiment. Thank you.
Thank you, Nikum. So, do we have any questions? So, I think we don't have any question. Thank you, Nikum.
Yes, sir.
So we move on to our next presenter. It's Ruma Bakshi. Are you here, Ruma?
Yes, yes. Good afternoon, sir. Good evening, sir. Good evening. So should I share my screen?
Just a second. So, Ruma, so now
Yes.
you can share your screen. Thank you.
Okay, okay, sir. Thank you. Is my screen is visible?
Yes, yes, your screen is visible. So please start.
Okay. Good evening to all. Myself, Ruma Bakshi, postdoctoral research fellow under Professor Sarath Dalai from Institute of Science, Nirma University, Ahmedabad. I am going to present my research entitled Reverse Vaccinology using prediction of B and T cell epitopes from the liver stage antigen of Plasmodium vargai ANCA strain. So as we all know that conventional vaccinology deals with the cultivation of the microorganisms in the laboratory, and it takes decades to develop any vaccine candidate. In current era, the reverse vaccinology has come forward, where the vaccine candidates can be synthesized using immunoinformatics tools, and it is in a very short period of time, that is a span of 1 to 2 years.
Sorry to interrupt you, but your slides are not moving. So reshare your slides.
Okay, I am resharing.
your slides are not visible.
Okay, so now it is visible, sir?
Yeah, it's visible.
Okay, so now it is moving?
No.
Still it is not moving?
No. I think when you play the slides, it's not moving. So you just don't play the slides.
So actually from my side the slides are moving, sir.
Yeah, I think it's from your side only.
Okay, so
so you just reshare your slides, but don't play it. You just make it, without those.
okay, then every time I am resharing my slide, okay?
Let it be. So you run on Windows mode only, means don't play the slides.
Okay, now this slide is visible?
Yeah, this is visible.
Okay, so in reverse vexillology, it deals with the use of proteomic and genomic sequences which to develop a new vaccine candidate against a particular pathogen. It is a very efficient and rapid method to design a vaccine candidate. In 1990s, Rainer Rapoli first used this reverse vexillology method to develop a vaccine candidate against meningococcus bacteria. In future, structural vexillology has play an important role in vaccine development, where antigenic structure of any proteins are determined by X-ray crystallography or NMR methods, and this helps in rational design of target epitopes. So again, I am sharing my next slides. Is this slide is visible, sir?
Yeah.
Hello?
Yes, your slides are visible.
Okay. So the objective of the present project is to predict B and T cell epitopes of Plasmodium vargai ANCA strain of liver stage proteins and validate these epitopes at preclinical level. The study design is, first of all, we retrieved the protein sequences of Plasmodium vargai ANCA strain of liver stage proteins from UniProt. It is a universal database for proteomes, and next these retrieved proteins are uploaded in the Vaxigen tool in FASTA format to check the highest antigenicity, and after that the highest antigenic proteins are against predicted for subcellular localization. As we all know that any antigen or any protein which are present in the surface of the cell can be easily determined by antigen presenting cells or B cells or dendritic cells. Therefore, the proteins which are present in the surface of the cells are identified and further used for further analysis. Next, B and T cell epitopes are predicted by IEDB. It is a immunogenicity prediction tool and freely available online. And next, these predicted epitopes are molecular dog for receptor epitope interaction analysis. Finally, the conformation peptides are synthesized and they are validated at preclinical level for immunogenicity of predicted B and T cell epitopes. So this slide is visible, sir? Hello?
Yes, your slides are visible. Now it's
Okay, thank you. So the basic concept of this present project is that the C57BL6 mice is repeatedly immunized with sporozoites of Plasmodium vargai liver stage, which is a liver stage protein of Plasmodium vargai, under chloroquine prophylaxis to raise hyperimmune sera, and the predicted B and T cell epitopes from the sporozoites of the Plasmodium vargai ANCA strains are used for peptide array. The basic principle of this peptide array technology is to find out the interaction between peptide and the antibodies and validation of the B cell epitopes. Finally, the antigens having both B and T cell epitopes are identified from the predicted epitope list. Further, the antigen or novel antigen having both B and T cell epitopes are incubated with the B cells isolated from spleens of the immunized mice, and they are the T cell activation is assessed by flow cytometric method. So, any exogenous antigen whenever enter into our body and help in activation of the CD8 T cells through MHC1 pathway, the B cell the cross presentation concept is established. So, the our project is aiming to establish a novel antigen or novel vaccine candidate having both B and T cell epitopes, and they can immunize or they can help in immune response in a preclinical level by raising antibodies as well as activating the CD8 T cells response or adaptive immunity through B cell mediated cross presentation concept. So, thank you, sir. Thank you all.
Thank you, Ruma. So, do we have any questions? So, thank you, Ruma. We don't have any questions. So, we are moving on to our next presenter.
Thank you, sir.
Nazneen, are you ready?
Yes, sir.
So wait just a second. Okay.
Sir, is my slide visible?
Yes. Wait a minute. Okay. So, Nazneen, you are ready? So please.
Yes, sir. Good evening, everyone. I am Nazneen Rejwana. I have recently completed my Master's in Genetics from University of Mysore. Today, my presentation title is Design of de novo inhibitor for peptide dehydrolase, from Plasmodium falciparum by computer-aided drug designing method. We know that malaria is a life-threatening disease. According to the World Health Organization, there is a 239 million cases are there, and among them 2 million deaths are annually presented. So malaria is normally spread through the mosquito bite. At first, the infected Anopheles mosquito bite, when the bite to the healthy human, the spores goes inside to the body and it targets to the liver cell. And after that, from the liver cell, it goes to the red blood cell, and from the red blood cell, the spore turns into the merozoite and it is spreaded, and the malaria symptoms are expressed. So there are several drugs actually, but the problem is the drug resistance is there. So we need to time to time we need to develop the new drug. So from the previous studies, we found that peptide deformylase is a good attractive target that is identified to inhibit the malaria parasite Plasmodium falciparum. So why the peptide deformylase as an attractive target? In the peptide deformylase, there is a sequence signal that contains there is a sequence signal, and it suggesting that it would be targeted to the apicoplast. And apicoplast is the small plastic that present in the parasite. And this apicoplast is necessary for the parasite survival. So if we can inhibit the parasite survival by blocking the apicoplast protein synthesis, the Plasmodium parasite survival can be removed. So in the de novo drug designing is very important to reduce the time and the cost. So for the in silico drug designing, at first we have to select the target. So here the target can be protein or gene or immune. Here I selected the protein selection, and then from the literature the ligands also being selected, and the ligand pocket, that is the active site of the protein, is also being selected. So the druggability is very much important to identify the interaction between the ligand and protein. So higher druggability or higher affinity means there is a good binding, there is a good interaction between the ligand and the protein. So after having the good binding affinities ligand, we generate the pharmacophore. Pharmacophore, that is the arrangement of the atoms, and then from the pharmacophore, the virtual screening was done to screen the best inhibitor from the data. Then again the binding affinity was checked, and then which molecules have shown the good higher affinity, the toxicity checking was done. So and here, so from the protein data bank, the protein was selected, that is protein ID is 1RL4, and from the protein this protein I removed all of the unnecessary element, that is water and heteroatom, and then the energy minimization is also be done, and the protein structure was also being checked by the Ramachandran part for checking the structure of the protein. Then from the literature, there is these three substances were found. They are showing the higher affinity within the binding, within the protein binding site. And before doing this, checking the these things, I selected the active site of the protein through the software CAT-SPIN. And these three molecules are getting from the literature, and the similar related compounds are chosen, so that these 10 compounds are selected to generate the pharmacophore, that is the ligand-based pharmacophore, and through the pharmacophore we screen the de novo inhibitor. So after from the inhibitor de novo, after the screening of the molecule, we find that this CID, these molecules that showing the minus 8.5 kilocalorie per mole binding affinity with the protein, that is very high from the previous binding affinity from the previous ligands. And then the toxicity is also being checked. Here there is finding that molecular weights of this protein is less than 500, and the hydrogen donor receptor is less than 10, and the number of hydrogen donors is also less than 5, and the lipophility is also less than 5. Lipophility means there is a ability of dissolving the molecules to the oil. Then, and this is the protein and ligand interaction. There it shows that amino acid that interacts between the ligand and protein. So here, here is the ligand and protein interaction. It shows that it has a good interaction between the in the protein, and there is no problems of this. So here it is the amino acid, that is the hydrophobic amino acid interaction. We know that hydrophobic interaction is very important for the protein structure development, and the hydrogen bond is also important for the protein structure. We know that when the hydrogen bond is smaller, the interaction is the stability is also less, but when the hydrogen bond is more, the stability of the protein or the bond is very high. And here it is the this yellow part is the previously selected molecule that is already presented in the literature, and the red one is the that is screened molecule, and here it shows that there is a superimposed structure there is a superimposed structure between among these ligands. So that's all. It is the in silico study. So in future, the in vitro analysis can be done. Thank you.
Thank you, Najni. So I think we have a question. So Asha is asking how the T cell activation is tested.
I'm sorry, sir. Just 1 minute.
Previous. So sorry, that question was not meant for you. So I think we have no question for you.
Okay, sir.
Okay, thank you.
Thank you.
So we move to our next presentation. Shreya Bhattacharya, are you ready?
Hello.
Hello, Shreya.
Yeah, am I audible?
Yeah, you are audible. So can you share your screen?
Yeah, has the previous speaker stopped sharing?
Okay. Nazni?
Yeah, okay. I'll just share now. Is my screen visible now? Are the slides visible?
Yeah, Shreya, your slides are visible.
And are they moving?
Yeah, they are moving also.
Okay. So a very good evening to everyone. So what I'll be discussing today is very briefly the work plan of the project that I'm currently involved in. Now, as we have all heard before during the presentation today also, that malaria has remained one of the most devastating diseases for decades now, which has been affecting a huge population across the globe. Now, several malaria control strategies are being implemented, but clearly given the current global scenario, these strategies don't seem to be working very well. Now, in the year 1991, when immunologist Patrick Duffy was joining the US National Institute of Allergy and Infectious Diseases, the research community at that time was quite skeptical that it would even be possible to vaccinate against parasites, especially the Plasmodium parasites, because there's no doubt that we are dealing with an extremely complex system now. That is a part of the, and hence a part of the impediment to a successful vaccine is obviously attributed to its complexity, but also due to our gaps in knowledge regarding its interaction with the host and the host immune responses against it. But in 2019, when children in Ghana, Kenya, and Malawi began receiving the first malaria vaccine, which was shown to offer protection in the phase 3 trial, which is the RTSS, Patrick Duffy mentioned that the biggest news is that we have malaria vaccines that can actually work. So, therefore, multi-antigen vaccine formulations are now rapidly becoming the area of study when it comes to malaria vaccination, where potential antigens from different critical stages of the Plasmodium life cycle, like the pre-erythrocytic stage, the blood stage, as well as the gametocyte stage, are being identified and they are being characterized. And once they are being characterized, different combinations are being used in order to prepare a vaccine formulation. But for such formulations to be prepared, it is essential to not only identify the antigens that can play a role in protection, but also try to characterize the molecule in terms of the immune response that it can elicit in the host. So one such molecule that has been identified in our lab, which is capable of providing partial protection against Plasmodium burgii, is a putative DNA J domain containing heat shock protein. Now, heat shock proteins, through previous research studies, have shown to have a protective role against pathogens. That is, they can either immunomodulate in an antigen-independent manner, where they, in an antigen-independent manner, where they themselves function as strong immunogens, or they may help in carrying important antigenic peptides of pathogens for presentations, or they may also help in the assembly of critical immune complexes. Now, this particular molecule that I'm currently working on is, as I mentioned, is a putative DNA J domain containing heat shock protein belongs to the HSP family, which is found to be expressed in all stages of the parasite life cycle, but predominantly in the blood stage. And this molecule also possesses a single functional pexel motif, which basically aids in the export of the protein from the parasite into the host cell cytosol. Now, through our study in the lab, what we had observed is when we use this protein to immunize mice, we observed a delay, a 4-day delay in the pre-patent period. That is, the emergence of the parasite in the blood stage was delayed by 4 days as compared to the naive or the non-immunized mice. So, this observation further intrigued us to explore the immune potential, or rather the vaccine potential of this antigen. Now, at this point in time, I'm not equipped with too much data because I've currently started working on this project. But what my overall aim at this stage is that I would want to understand the nature of the immune protection conferred by this molecule, that is, whether it is humoral or cell mediated, as well as understand the individual contributions of both the arms of the immune system. Further, if it's a cell mediated immune response, and I would want to identify as to which T cell subtype or types are involved in the immune protection, as well as to see whether they are polyfunctional in nature or not. Third, my aim is to identify the protective and immunodominant regions or epitopes of this particular antigen. That is, in case this antigen shows some promising potential as a vaccine candidate, and if we would like to take it further as part of a combination vaccine, I would want to just stick to that part of the antigen which is providing the maximum immune protection. And finally, I would want to see if this antigen is capable of conferring any long-term memory response, which is one of the critical features associated with any vaccine candidate. So yeah, this is all that I wanted to talk about. Thank you.
Thank you, Shreya. Thank you for your short and crisp presentation. So, do we have any questions? So, Shreya, I think we don't have any questions from the
Okay. Thank you so much.
Thank you for your presentation.
Thank you.
So we now move on to our next presenter, Ayushi Singh. Ayushi, are you ready?
Yes, sir.
So can you start sharing your screen?
Yeah, sure. Just a second, sir. I think my screen is not visible.
Yes, your screen is not visible. Now it's visible.
I don't know why.
Now it's visible.
Can anyone see my screen?
Yeah, we can see your screen.
Okay. So, good evening, everyone. I'm giving a short lecture on the topic of BCL2 negatively regulates protective immune responses during mycobacterial infection. And as we know, tuberculosis is caused by mycobacterium tuberculosis, which results in high mortality and morbidity around the world. And with the emergence of multi-drug resistance and extreme drug resistance, it is really becoming important to understand the host pathogen interactions at the cellular and molecular levels to come up with better therapeutic interventions. So from our previous data, we have found out that MTB and antigen increases the expression of this anti-apoptotic molecule BCL2. And this we have done this study in macrophages. Another group from our lab also showed that stimulating the cells with the MTB antigen RB2463 also increases the expression of BCL2. So therefore, in our study, we wish to look into the role of BCL2 in modulating the host defense responses in immune cells by using a BCL2 specific inhibitor, which is EM2025 in our study. So moving on to the results section, firstly, to begin with, we first monitored the expression of BCL2 in murine BMDCs, and we found out that the expression of BCL2 increases upon stimulating the cells with MTB antigen RV3416 by flow cytometry, Western blotting, and confocal microscopy. We then monitored the growth of BCG in BMDCs following BCL2 inhibition. So we see that when we inhibit BCL2 expression using BCL2 specific inhibitor, there is a down regulation in the bacterial burden. We have also done this inhibition via knockdown of BCL2 siRNA mediated knockdown, and we also got a similar kind of result. We also wish to find out the role of BCL2 in regulating host defense responses like reactive oxygen species generation, co-stimulatory molecules expression, and cytokine milieu. So to begin with, we found out that when we inhibit BCL2 expression using BCL2 specific inhibitor, there is increase in the generation of protective immune response like reactive oxygen species generation, either via BCL2 specific inhibitor or by knocking down BCL2 expression. We also found out that when we inhibit BCL2 expression, there is increase in the process of autophagy, which is also a defensive response, as there is more of the LC3 puncta formation. LC3 is a very good marker for detecting autophagy. So we see that there is more of the LC3 puncta and less of the BCG. Moving on, we also wished to look into the expression profile of the co-stimulatory molecules like CD40 and PDL1. So we see that in case of PDL1, there is a decrease in the expression of PDL1 expression when we inhibit BCL2 in RV3416 stimulated cells as well as BCG stimulated cells. And there is more of the expression of co-stimulatory molecule CD40 when we stimulate the cells with MTB antigen upon BCL2 inhibition and BCG stimulation upon BCL2 inhibition. We also looked into the profile of the pro-inflammatory cytokine IFN gamma, and we found that when we inhibit BCL2 expression using a BCL2 inhibitor, either in MTB antigen stimulated cells or BCG stimulated cells in BMDCs, this data is in BMDCs and this data is in PBMCs. And we got a similar kind of a profile in both of these cases, that there is more of the pro-inflammatory cytokine IFN gamma expression. We then further wish to look into the effector memory phenotype expression in case of T cells, whether these activated BMDCs have the capacity to activate T cells or not. And for that, we looked into the expression of CD62L, CCR7, and as we know that in our hormone receptors, and if the expression of these molecules. So we see that when we inhibit BCL2 expression using this BCL2 inhibitor, there is down regulation of CD62 and CTR7 expression, but yes, there is more of CD69, which is a T cell activation marker. So here also we get a positive response upon BCL2 inhibition. Finally, the concluding experiment of our study was to see whether this BCL2 inhibition has the ability to modulate T cell activation and function. So for that, what we did was we had infected BMDCs with BCG, and then cultured these infected BMDCs with T cells that were infected with BCG for 48 hours, and then these T cells were added onto GFP BCG infected macrophages, and then we have taken these confocal images where we see that activated T cells have the capacity to reduce bacterial burden in cells that were treated with BCL2 inhibitor. And we got a similar result when we reproduce these results with siRNA mediated knockdown. And moving to the conclusion part, we see that there is more of a decrease in the intracellular bacterial burden in disease, and there is generation of protective immune response, which includes the expression of co-stimulatory molecules and IFN gamma expression. We had finally co-culturing, when we co-cultured these BMDCs and PBMCs with antigen primed T cells, there is more of the proliferation and activation of these cells. And finally, to conclude, we say that mycobacteria upregulates BCL2 not only to enhance its survival in the host, but also to downregulate the protective responses that were mounted by the host. Thank you so much. Any questions?
Thank you, Ayushi, for explaining the role of BCL2 in very short and crisp presentation.
Yeah, thank you so much, sir.
So, so we don't have any question from the other listeners,
Okay.
viewers. So,
No, okay,
thank you.
sir.
We are now moving on to our next presentation.
Okay.
So, Renu Darra.
Yes, sir.
Are you ready for your presentation?
Yes, I'm ready. Am I audible?
Yeah, you are clearly audible. So please share your screen.
Okay. Are my slides visible?
Yeah, your slides are visible.
And they are moving?
Right now they are not moving.
Now?
No.
They are not moving?
No. Reshare your slides. Hopefully they are
Okay.
moving.
I'll reshare it. Is it visible now?
Not yet. Your slides are visible.
Okay. And are they moving?
So I don't think so. They are moving actually. Just try to change. So don't play your slides. You just enlarge your window. So I think then it will move.
Okay. I'll just escape it.
Yes. So yes. You just play in the
Is it fine now?
Yeah, it's fine, but you just show if they are moving or not. Your cursor is moving, so I hope. Yes, they are moving. So please continue.
Okay. So, hello everyone. I'm here to present part of my work done during my PhD. That is virtual ligand screening as a tool to identify drug-like inhibitors against Mycobacterium marinum love protein. So, with the increased incidence of MDR, XDR, and TDR TB, there is a urgent need to design new drugs. And since for that, we had here used M. marinum as a surrogate model to study MTB pathogenesis. The advantages with using M. marinum is because it shares a close genetic homology to M. Tuberculosis, and it has a faster generation time, and it can be handled at BSL-2 facility. So therefore, we used M. marinum as our model system. An interesting thing here is that the target that we used is the Malto locus, the mycobacterial enhanced infection locus. It is specifically present in M. marinum and M. tuberculosis. It was identified by Sahar and her co-workers in year 2004, and it was found at that time that 6 genes in this locus, they are close genetic homologs of prokaryotic luciferase like monoxygenase gene, and these genes are associated with to withstand the enhanced ROS and RNS stress response that is elated by the host. So similar responses were found in M. marinum and M. tuberculosis, and wherein it was found that melton expedites the detoxification of ROS and RNS produced by the activated macrophages, and it was also found that melton glucose displays resistance to ROS and RNS in laboratory media as well as iron gamma plus LPS activated neuron macrophages. Interestingly, the mutation in melapho melphalucus, which is the first gene in melphalucus, it casts a polar effect on the downstream genes, and the increased susceptibility of melamutant to ROS RNS could partially be retrieved by melaph alone and completely by the entire melphalucus. So, keeping that in mind, we designed a hypothesis that targeting the MELF protein activity expressed by this MELF gene of melphalucus would render the M. tuberculosis and M. marinum more susceptible to the ROS and RNS stress response. So, we employed computational techniques in conjunction with conventional laboratory techniques to identify antagonists that can inhibit the MELF protein activity. And we have examined the antagonistic effect of these drug-like molecules on purified MELF in an enzyme-based assay, and the respective MIC and MBC of those compounds were derived. So, in brief, the methodology was to determine the structure of MELF protein and its pocket identification for virtual ligand screening to identify putative drug-like molecules. This gene MELF was cloned, expressed, and purified using MBP-tagged MELF protein in Escherichia coli, and then the drug-like molecules that were screened using virtual ligand screening. They were screened in an enzyme-based assay, and the MIC and MBC and also the cytotoxicity of the top scoring compounds was determined. So the structure was modeled based on 2WGK. It is a structure that was available at PDB with 36% sequence similarity and 97% coverage. The loops were built using Loopy and Loop Builder module of Yasara, and using AMBER-3 force field, the molecular dynamics simulation was done for the structure, and the flexible and rigid parts of the protein were identified, and the protein was a homodimer, and the Ramachandran plot was derived to see the quality of the structure, and it was found that the structure derived was a good quality structure with an RMSD value of 1.5 angstrom. The multi-step VLS protocol included use of Cambridge compound collection libraries, which had approximately 8 lakh compounds that were a diversified set of compounds, which was essential for a VLS to run successfully. We removed the non-drug-like molecules using FAFDrugs2 software and using soft drug-like filtered, and the 5 lakh compounds that were filtered were docked in using Autodock AHITs and dock, and 178 compounds were selected based on their binding energy scores and manual curation. Later on, we identified the largest pocket in the MELF protein using 3 different methods: Inca site finder, Q site finder, and pocket finder, and we using a consensual approach, the best pocket was identified. And in an enzyme-based assay, the putative monooxygenase activity was putative flavin-dependent oxidorotase activity was established using NADH, and the reduction of NADH to NAD at 340 nanometer was measured, and the Vmax and Km values were determined. And later on, after screening the 178 compounds with a whole cell lysate and purified MELF protein, we finally shortlisted 20 compounds with good activity against the purified as well as the whole cell lysate in the enzyme-based assay, and the MIC and MBC of those compounds was determined using conventional CFU assays. And of these, top 6 compounds were selected based on the based on MIC and MBC values, and Dixon plots were plotted where inverse of Vmax value were plotted against the inhibitor concentration using different substrate concentrations, and it was found that 4 of these 6 compounds were competitive in nature. That is, they competed with the flavin adenine dinucleotide for the active site, and of these 6 compounds, 4 were found to be non-cytotoxic. These results were further validated in vitro assay using raw and THP1 macrophages. These macrophages were infected with M. marinum in an in vitro cell culture system, and it was found that the 517552 and 6513 745, these 2 compounds, they were found to inhibit the replication, intracellular survival and replication of Mycobacterium marinum inside the activated host macrophages. The results were further substantiated with transmission electron microscopy. However, due to the restriction of slides and time, I could not incorporate those results into this presentation. With this, I conclude my work. Thank you. Any questions?
Yes. So, do we have any questions? So, Renu, I think we don't have any questions. So, thank you for your presentation. So,
Thank you.
So now we are moving on to our next presenter. We have Deepika here from ICT.
So good evening everyone. I am Deepika Jaiswal and I am going to present my work on implication of promiscuous antibodies and viral immune escape. So promiscuous antibodies or polyreactive antibodies are those antibodies which can bind to multiple antigens. Generally 20% of antibody populations are polyreactive antibodies, but in case of viral infection these percentage increases to 70 to 80%. So here the question arises, why these polyreactive antibodies percentage increases in case of viral infection. Possible explanation is that these polyreactive antibodies might be helpful in countering the immune evasion by fast mutating virus like HIV and influenza. So what is immune escape? Whenever an antigen comes in contact with a B cell receptor, it produces soluble antibodies which can bind to antigen and neutralize it. But when this antigen mutates, then these already produced antibodies are not able to neutralize the mutant antigen and immune evasion occur. This immune evasion phenomena is well known in case of fast mutating viruses like HIV and influenza. So to understand this puzzle of polyreactivity and immune evasion, we have selected HIV as our model system. HIV incorporates mutation in their surface protein or envelope protein, which is a trimeric protein. It consists of two parts, GP120 and GP41. Mainly antibodies are produced against this exposed part of the GP120 and 41. We have selected two critical epitopes in the GP41 region. One is the ELPKW and another is GER-TR-TR. Another epitope is selected inside the membrane because this part is exposed after the attachment of virus to the host genome. And antibodies are produced against this. So against these two epitopes, we have screened 3 antibody fast display library by the biopanel method. We have done 4 rounds of biopanel and we have found that the maximum finding is in the 4th round. So we have carried out monoclonal individual phages screening from the 4th round. So we have selected 400 phages from each peptide and checked for their binding, check their binding by monoclonal ELISA and found that many of the antibodies are showing more binding. So we have selected those phages and check their cross reactivity. Since our next step is to find the promiscuous antibodies, we have selected first the variants of the epitope. These variants are natural escape variants of HIV which are selected from the HIV database on the basis of their percentage abundance in the population. So there are mutations such that there are single mutation, double mutation and triple mutations. Since we have already selected high binders, we have checked their cross reactivity with the other variants and we have found that in both the cases many of the antibodies are able to cross react with other variants also. So we have selected those as shown in the red dots. We have selected those antibodies which can cross react with most of the variants also. After that we have purified these antibodies like in case of GER-DRDR we have purified 5 antibodies and checked their cross-reactive profile by BLI. Here the KD value were in 10 power minus 6 or minus 7 range. And we can see that in first epitope there is a cross-reactive profile, means that each antibodies can bind to different variants of the epitope. And in case of ELTKW, some of the antibodies were able to bind to variants and some were able to not bind to one or two of the variants also. So there is a difference in the cross-reactive profile due to their different hotspot. As here
promiscuous antibodies which can bind to most of the variants, and all antibodies were showing affinity in the physiological relevant affinities. Since one of the epitope was showing less cross-reactive profile, as we can see that this epitope is inside the membrane. It is exposed for very less time, only when there is an attachment of the virus to the host. So this could be the possible reason for the less cross-reactive profile of this epitope. And our study shows that the promiscuous antibodies can be obtained and it might play an important role in countering the immune evasion in case of HIV. Thank you.
Thank you, Devika. So, any questions for Devika? So, I think thank you, Devika, for your short presentation and on role of promiscuous antibodies on HIV. So now we are moving on to our next presenter. So, do we have Hazrat Ali here? Hello. So now we move on to our next presenter if Hazrat Ali is not around. So Shazib, are you around? Can you listen to me?
Yes, sir. I'm here.
So can you share your screen? And
Yes, sir. Is it visible, sir?
Yeah, it's visible.
Thank you. Good evening, everyone. Myself, Shazib Ahmed from Dr. Dinesh Gupta lab. Today, I will be presenting a topic entitled SARS-CoV-2 spike glycoprotein double mutant variant, that is leucine 452 to arginine and glutamic acid 484 to glutamine. It is a structural and dynamical studies. As we all know, the epidemic spread of COVID-19 caused by SARS-CoV-2 has led to a worldwide devastating effect on economic and health system. In this recent time, several SARS-CoV-2 variants have been reported globally and lead to increased infection. The SARS-CoV-2 receptor binding domain, it is located to the S1 subunit of spike glycoprotein, which play a vital role in the virus binding to the host cell receptor and its entry into the host cell through the ACE2 receptor. A recently identified variant of SARS-CoV-2 known as double mutant, which is present on RBD domain. In this regard, the study was aimed to employ computational method on wild type and 3 mutants, which is involved individual mutant, that is 452, 480, 484, as well double mutant on the same structure. We use SARS-CoV-2 spike glycoprotein crystal structure retrieved from PDB database. The RBD variant mutants at respective position and used for the further study to understand the effect of mutation, protein dynamics, destabilization, and damaging role of various prediction algorithms were employed on both wild type and mutant. Consequently, molecular dynamic simulation studies are employed using GROMACS and analyze various parameters. The figure elucidates in yellow, it is RBD domain, and it is highlighted in magenta, it is a mutant. The RMSD analysis revealed that the mutation were unstable. The instability caused by a mutant confirmed with higher deviation of fluctuation in RMSD plot, which lead to conformational change in instability of in RBD. Furthermore, the overall flexibility of The residues were determined by RMSA plot of each residue. The result found that the minimal fluctuation on wild type, that is in black color. However, in the mutant, that is green, red, and blue color, it is showing that the higher fluctuation as compared to wild type. So it means it is unstabilized the protein. Further, the RG, that is radius of gyration, the result from RG suggests that the mutant showed a high devastative impact on native structure, including structure destabilization, which could be responsible for loss of compactness, suggests a cumulative effect on core structure of RBD. Furthermore, the solvent accessible surface area, we can see that the wild type mutant structure were further subjected, and the system of core hydrophobic region were examined, and we found significant decrease in SASA upon mutant. The fluctuation of SASA result justified a high loss of hydrophobic contact between residues on RBD domain, while substitution of amino acid. Furthermore, we analyze intramolecular hydrogen bond of each wild type and mutant structure. The investigation revealed the alteration in hydrogen bond of mutant have less number of intramolecular hydrogen bond, and we can see that the projection on eigenvector 1 and eigenvector 2, the analysis showed that the essential subspace examined where the protein dynamics are noted with eigenvectors associated with eigenvalue, the dynamic behavior of wild type and mutant were compared with a clustering parameter, and results showed that the cluster are well defined in wild type structure by covering a minimum region, whereas we can see the mutant occupied maximum region with a wider cluster which indicate significant conformation change in mutant, leading to a protein instability. Furthermore, it is a pre-energy landscape plot of wild type and variants were explored. This plot is plotted between PC1 and PC2. It is principal component 1 and principal component 2. The comparison studies indicated stable global energy minima confined with one basin in wild type. However, mutant including 452, 452 and 484 and double mutant, we can see that the more expansion basin and numerous meta-stable conformation as associated with mutant. The conclusion of my study, the mutant of RBD established in study, all the three mutants showed a significant effect on portrait, a deleterious impact on SARS-CoV-2. Various studies and advancements revealed the involvement of mutation play a role on the dynamic effect of a spike glycoprotein. The combination of predicted algorithm and molecular dynamic simulation studies facilitated understanding the effect caused by mutant on RBD, contributing to spread and infection in the host. The molecular dynamic simulation study investigation strength are studied to confer the unstable conformations of mutants compared to wild type. Overall, the study opened up a direction for further investigation on deleterious mutation present on RBD function domain of spike glycoprotein. Thank you. Any question?
Any questions? Thank you, Shazir.
Welcome.
Shazir, for your short presentation giving glimpse into the molecular dynamics of receptor binding domain in SARS-CoV. So now we move on. since we don't have any questions for you, so we can move on to next speaker. So Kingsley, are you around, Kingsley?
Yes, sir. I'm ready, sir. Am I audible?
Yeah.
Okay. I'll share my screen.
Yeah, sure.
Is it visible, sir?
Not yet. Now it's visible.
Okay.
So it's not playing actually.
Yeah, just 1 second, sir. Is it moving, sir?
Yeah, it's moving. So please.
Okay. Okay. So good evening to all. So I'll just present as part of my PhD work, and it's on the effect of naciclacin on inflammatory response and outcome in neonatal sepsis rat models. So actually, sepsis is one of the leading causes of morbidity and mortality among newborns and neonates. So antibiotics and supportive care are the mainstay of treatment at present. However, the microorganisms which are causing neonatal sepsis are increasingly becoming resistant to the antibiotics that are currently in use. So we thought of using anti-inflammatory drugs which could reduce the excessive inflammatory response, and that actually could improve the outcome in neonatal sepsis. So actually, calprotectin is an acute phase reactant which is present in the cytoplasm of the innate immune cells, and it consists of two subunits, S100A8 and S100A9 subunit. Now, this calprotectin levels are really elevated in neonatal sepsis. So this causes a lot of cytotoxicity and apoptosis inducing properties. And this calprotectin is also an agonist of TLR4, and it possesses pro-inflammatory properties. It activates the TLR4 NF-kappa B, that pathway, and a lot of adaptive proteins such as MID88 are also upregulated during this activation. So naciclacin is an isocarbosteryl alkaloid which belongs to the amaldiaceae family. That several studies has been done, but in sepsis it has not been tried. So we tried to create a neonatal rat model with E. coli. So we used E. coli to induce sepsis in the neonatal rats, and our main objectives was to assess the anti-calprotectin activity of naciclacin, whether the levels of the S100A8 and A9 are elevated or suppressed after the treatment with naciclacin. We also decided to study the local and systemic bacterial clearance, whether the bacterial load was reduced after treatment with naciclacin. And we decided also to study multiple inflammatory cytokines, because in sepsis, the cytokine levels are increased. So we decided whether naciclacin could reduce the cytokine levels along with calprotectin. Then we did a lot of histological studies and immunohistochemistry, and also the organ damage parameters such as AST and ALT. And we studied the expression of various proteins in this pathway, LPS, which is the important component of all this gram-negative bacteria, which triggers the TLR4 NF-kappa B pathway, which results in the upregulation of cytokines. So we thought, what is the mechanism of action of naciclacin, whether it could suppress the adapter proteins in this pathway. So these I'll just share a short thing. I can't tell you everything, but in 5 minutes. So I'll just share a short of what results we got. So this is the survival. We measured the survival for 30 hours after treatment with naciclacin, and we found that naciclacin at 3 doses we used, because we didn't want to use a high dose of naciclacin. We used very low dose, 0.1 mg per kg, 1 mg per kg, and 3 mg per kg. We found that at 1 mg per kg, it could significantly improve the survival. When untreated rats, neonatal rats, were all dead by 24 hours. So we observed them for 30 hours and found a significant improvement in survival at 1 mg and 3 mg per kg naciclacin. And we also studied the clinical parameters such as piloerection, lethargy, the activity of the rats, and the response to touch. And we found that naciclacin treatment improved the activity of the rats and their response to touch over a period of 30 hours. So we also decided to study the bacterial load, and we found a significant suppression in the bacterial, not only in the blood bacterial load, we also found in the liver and lung homogenates when we plated out. We studied the bacterial load. The naciclacin could suppress the bacterial load. So these are the treatment. This is the sepsis group. You can find a significant elevation of the bacterial. And these are the cytokines we studied. We studied a number of pro-inflammatory and anti-inflammatory cytokines. We used the cytokine B-direct to study multiple cytokines, because the samples we get are very low in neonatal rats. So we also studied, this is the calprotectin. The second column in all the graphs is the sepsis untreated group. So we found the elevation, IL-2, TNF and IL-6, we found the elevation. And these three are the 0.1, 1 and 3 mg in the nasiglacin treated, suppression of pro-inflammatory cytokines and suppression of calprotectin levels in the plasma. So we also studied the immunohistochemistry, as I previously told you. In the liver tissues, we used Abcam kit actually to study the IHC and the binding of the primary antibody. We used S100A8, A9 primary antibodies were visualized by DAB, DAB and counterstain with hematoxylin. So this is a sepsis group. The second one is a sepsis group, and these three are the treatment groups. We found significant reduction of the expression in the tissues as well of calprotectin. So these are the Western blot images. So you can see the A8 and A9 protein levels. The second one is a sepsis group, and there's three other treatment. We found at 1 mg and 3 mg, there was significant suppression of S100, A8 and A9. We also studied number of adaptive proteins. We studied the phosphorylation of NF-kappa B. We found the phosphorylation of IK beta alpha and MYD88. And these are the densitogram, the relative levels and the densitograms. So that is the Western blot picture. And this, we studied all the 6 organs. We studied liver, lungs, heart, kidney, intestine, and the heart. We studied histology, HND staining of all the organs were done. We found a lot of inflammatory cell infiltration in the sepsis untreated group. In the treatment group, this was 0.1 mg. We found a slight reduction in inflammatory cell infiltration. In 1 mg and 3 mg, we found significant reduction. This is the heart. This is the waveform arrangement, which is a symptom of sepsis. Waveform arrangement, increased inflammatory cell infiltration in the untreated group. In the treatment groups, we found significant improvement at 1 mg and 3 mg of nasiglacin. So these are all the histological images. We studied AST, ALT, and also the hematological parameters such as packed cell volume, hemoglobin, and RBC. We found the nasiglacin could normalize this blood hematological parameters and also reduce the AST, ALT levels, which is a symbol of organ liver damage. So it could reduce the plasma levels. So in conclusion, I would like to state that nasiglacin improved the outcome in our study using neonatal rats. And we think that it's through the suppression of the activation of the LPS NF-kappa B pathway, which happens by the inhibition of calprotectin, which is actually an agonist of TLR4. So these findings establish a novel role and mechanism of action of nasiglacin in improving the outcome in neonatal septic rats. Thank you.
Thank you, Kingsley.
Thank you, sir.
So any questions? So thank you, Kingsley, for explaining the role of nasiglacin.
Thank you, sir. Thank you, sir.
Okay, so I think we don't have any questions. So with this, so we have one speaker still left. So Hazrat Ali, are you around? So I think Hazrat Ali is not around. So with this, we are closing our today's session. And thank you all presenters for your patience. So as we were a little, slow on timings. So and please do join tomorrow. We have eminent speakers from around the world, and we do also have a workshop in a second session tomorrow. So see you tomorrow. So good night and good evening. So thank you very much. See you tomorrow.